mechanisms for abiotic stress tolerance in plants, jointly organized by the Indian Society for Plant Physiology, South Zone, and the Department of Botany, University of Calicut. Ladies and gentlemen, let's start our third technical session comprising the lead lecture and a special inviter lecture. I wholeheartedly invite our beloved Professor Dr. Manju C. Nair, Associate Professor, Department of Botany, University of Calicut, for coordinating the session. Ma'am, please.
Very good morning to one and all. So it's my great pleasure to invite the chair, co-chair and rapporteur for this session. So today's first technical session is Dr. Anil Kumar. So I happen to know this person from 1995 itself. So it's a great pleasure for me to introduce Anil Kumar sir. So he is now working as senior scientist in Gen TBJRI, and since 1987 he is actively participating in the conservation biology division, and he is concerned with he is actively doing his research on species management and proactive eco-restoration. And his major interest also lies on seed biology, and his academic pursuits in his credit is with 6 PhDs, 56 conference presentations, and more than 50 research papers, and 2 books in his credit. With these words, I welcome Dr. Anil Kumar sir to this session. So today's co-chair, Dr. Pavadi M. Srikumar, she is Assistant Professor of Department of Plant Physiology, so Kerala Agriculture University, Trivandrum. And her research focus is on plant abiotic stress physiology and plant nutrition physiology. And she has published more than 10 research papers and 5 book chapters. And she is interested in plant biotechnology also. And her achievements include she is awarded with the Namaste Consortium grant under the Namaste India EU Mobility Project, and awarded with the GRC called Storm International Diversity Award at US dollar of 1,250, and awarded the B4 fellowship also. So today's rapporteur is Dr. Danya Thomas. She is an alumnus of our department, and under Dr. Jose T. Putusa. She has taken her PhD, and now she is Assistant Professor of Department of Botany, Kama College, Mala. With these words, I welcome the chair, co-chair, and rapporteur for the session. Thank you.
So good morning to respected teachers, distinguished participants. Thanks for the nice introduction to us, and a special thanks to Jose Putu sir also, and I am already extended wholehearted cooperation to his good venture. So with these words, I invite my co-chair as well as rapporteur to the session. And okay, now coming to the business. So the session is going to start with Dr. Cecilia Z. Toth talk on sustainable and efficient algal hydrogen production. And a few words, sir, shall I introduce her? Yeah. So she is a group leader presently and scientific advisor at the Laboratory of Molecular Photobioenergetics and Biological Research Center, Zwickard, Institute of Plant Biology. And he took his PhD in biology from the University of Geneva, Switzerland in 2006. And she is a guest lab scientist at Max Planck Institute of Molecular Plant Physiology, Germany from year 2012 to 2014. And to her credit, her thrust area of research, it is, I learned that includes algal hydrogen production. That is a chosen topic. And role of ascorbate in photosynthesis and chlorophyll A fluorescence. And in this topic, she has awarded and recognized with many awards and honors, including Strobe Award for Biological Research Center, and UNESCO Women in Science and National Fellowship in 2017. And she is a member of the committee of Joint's Biology Research Program. And it went on like that. She has already published more than 50 research papers to her credit, and 2,027 citations and in H index of 22. Without much interruption between the talk, today's talk speaker, I welcome Dr. Cecilia Isaac to this wonderful seminar topic.
So thank you again very much. I'm really happy to be here. Thank you for the invitation for JOS. And this is my first time in India, and I really enjoy it, so I'm happy to be here, so thank you again. So I will talk about our main topic, which is hydrogen production in green algae, and I will also say a few words about electric current production by algae. So I come from Szeged, which is located here at the southern part of Hungary, and this is our research institute. Got a problem with this? It's working. I don't know what's going on. Anyway, so about renewable energy sources. So we have a problem that the energy demand is continuously increasing, and the fossil fuels are unevenly distributed on Earth. Sorry, I can hear an echo. Is it okay? Can you hear me well? Yes? Okay. It's fine. Okay, I just hear. Sorry. So we know that the fossil fuels are harmful for the environment and also for our health, so we need to find alternative solutions. So there are many possibilities, like all kinds of renewable energy sources, like wind energy, geothermal energy, hydropower, and biomass. But we have a problem that many of them are unevenly distributed on Earth. And there's also solar energy, which is nice because we have available it worldwide. So one possibility is to use hydrogen as a fuel, because most renewable energies produce electricity or heat, but we also need transportable fuels, and for that hydrogen would be quite an ideal option, because during its combustion there is no CO2 emitted, so we have only water as a final product. So it's very promising, but it is very challenging to produce it in a sustainable manner. And actually we use hydrogen for many purposes, like for petroleum refining, for producing nitrogen fertilizers in the food and cosmetic industry, and also in the machine industry. But we don't have it, we don't have a system in which we can produce it in a sustainable manner, because currently 99% of the total production comes from natural gas, and of course that produces an enormous amount of CO2. So there are alternative options, like electrolysis of water. We can also produce hydrogen by bacteria, that is fermentation of sugars, and these sugars could come from agricultural or food industry waste, which is a nice thing. And we can also use water splitting, but this is in research stage. We can also have systems of artificial photosynthesis. This is again quite in an initial stage. And we can also produce hydrogen by green algae. So this is our main research topic. So you know, algae do photosynthesis, and we have oxygen emitted. This is the source of oxygen in the atmosphere along with the plants. But interestingly, green algae can also produce hydrogen. So here, this is our favorite model organism called Chlamydomonas reinhardtii. So in general, green algae are outstanding in terms of ecology and biotechnology. And this Chlamydomonas reinhardtii is a unicellular green alga, and it belongs to the Chlorophyceae family. It is a widespread organism, and it can live both in soil and water. And it lives photoautotrophically, so only using photosynthesis, but it can also use sugars, and it can even grow in the dark. And it's also a haploid organism, and it's very easy to transform, and we have several transformation techniques, and there are also very good systems like the CLIP mutant library and the CRISPR-Cas technique that makes it an ideal model organism. So this is why we use Chlamydomonas to study photosynthesis, chloroplast development, and so on, and they produce hydrogen in anoxy, as I said. So in general, about hydrogenases, there are 3 types of them. So the iron-iron hydrogenases, the nickel-iron hydrogenases, and iron hydrogenases. The first 2 of these are true redox catalysts, and iron hydrogenases catalyze the reversible heterolytic cleavage of hydrogen. So among them, iron-iron hydrogenases are the ones that are found in Chlamydomonas. They have 2 types of them called high day 1 and high day 2, and high day 1 is the more important one. So this is the photosynthetic electron transport chain, as you may know. So we can find hydrogenase at the acceptor side of photosystem 1. So it receives the electrons directly from photosynthesis. So the electrons are transferred from water through photosystem to the plastoquinopool, cytochrome B6 subcomplex, PS1, and ferredoxin to hydrogenase. So green algae are often found under random conditions in nature, like in moist soil, where in symbiosis with bacteria, etc., they find themselves in anaerobic conditions. So, and then this enables hydrogenases to be expressed because they are very oxygen sensitive. So when there's line, when the light is shining, so at the beginning of the day, then there's a few minutes when the Calvin-Manson cycle is not active yet, but this is quite a dangerous situation for the photosynthetic electron transport chain because the excitation energy cannot be used. So actually hydrogenase acts in this case as a safety valve. But when the Calvin-Manson cycle is fully active, then it takes up the electrons, and then the hydrogenases are kind of outcompeted by the Calvin-Manson cycle. And then on top of that, when the Calvin-Manson cycle is fully active, then oxygen evolution is also highly active at that time, and oxygen inactivates hydrogenase, and it also inhibits its expression. So this is why in nature hydrogen production lasts only for a few minutes. So there have been attempts to so there are the pathways for hydrogen production. There are 3 of them. So this is the main one, the photosystem 2 dependent pathway, and then we have the photosystem 2 independent pathway, which is actually cyclic electron transport around photosystem 1. And there's also the dark fermentative pathway in which starch is degraded and the reducing power is used to be taken up by the photosynthetic electron transport, and then finally the electrons go to hydrogenase. So in theory, the photosystem 2 dependent pathway is the most efficient one because in this case, the electrons are directly used for hydrogen production. And this actually is quite an efficient process. So the theoretical maximum is about 13%, which is at least 10 times higher than in the case of like biomass-based biofuels. So these are the protocols that have been developed to prolong the process of hydrogen production. So this one was developed about 20 years ago by the group of Anastasios Melis. And it is based on sulfur deprivation, which means that we subject the cultures to sulfur-deprived, we place them in sulfur-deprived medium. And then, as a result, we'll get an arrest of cell division, and the photosynthetic electron transport chain will get partially degraded, and then we have a decrease in oxygen evolution, and then hypoxia is established. So then, under these conditions, hydrogen production lasts for several days. And this is how it works. So first we have an oxygen production phase. This is phase 1. And then we have a lag phase. And then hydrogen production starts after about 2 days. And it lasts for another like 2 days, more or less. And then we have the termination phase when hydrogen is not produced anymore. You can see that during this sulfur deprivation protocol, starch accumulates and then it degrades. This is what you see in the bottom slide. And then photosynthetic activity goes down quite strongly. And then this is a very severe stress effect. So even the morphology changes a lot. So after about 1 day of sulfur deprivation, we find that large spherical cells, and then they become smaller, and then you can see that their morphology is completely changed. So this is a method that has, of course, very strong consequences on on the physiology of the algal cells. So this is about the oxygen evolution. So you can see that oxygen evolution goes down by day 2. This is the white circles. And then at the same time, hydrogen production starts after about 2 days. And then if we add ECMU, which is a photosystem 2 inhibitor, then you can see that, hydrogen production doesn't occur. So it means that the photosynthetic electron transport chain is absolutely required for the start of hydrogen production, even though part of the electrons would come from starch degradation and the starch was accumulated in response to sulfur deprivation. So, as I said, this is a very severe stress effect, and most of the photosynthetic subunits are degraded on the time scale of a few days. So, for example, PSB gets degraded, so that is a photosystem 2 reaction center subunit, or CP43, interestingly, even RBCR, so the Rubisco subunit is getting degraded, and then Fe over MFM value, which is an indicator of photosynthetic efficiency, that is also very severely affected. So these are the features of sulfur deprivation. Now looking at a biotechnology point of view. So the procedure itself requires several washing steps, which is a time-consuming and expensive process if you think about biotechnology. Then it works relatively efficiently only in the presence of acetate, which again is expensive, and there is a risk of bacterial contamination on an industrial level. The electrons are derived from water oxidation and also from starch degradation, and as I said, it's a very severe stress effect, and the photosynthetic apparatus gets degraded. The people try to regenerate the cultures after these a few days of hydrogen production, but the regeneration was incomplete. And then the maximum light conversion efficiency is in the range of 0.1 to 1%. And yes, these are the attempts that were tried. So like nitrogen, phosphorus, and magnesium limitations, they all work in a similar way, but the problems remain the same. And then people also try to change the culture conditions to optimize the process. And then there was also attempts to downregulate photosystem 2 activity. But the problem is that if you downregulate photosystem 2, then of course it means that there is a limitation in the rate of electron transport, so there is less hydrogen production. So that's why we need new solutions. So this is one of them that was developed in my institute, not by my group, but another one. So it is based on algal bacterial co-cultures. So here they placed E. coli together with Chlamydomonas, and then they observed that in this case, without any sulfur deprivation, there is hydrogen production. And this hydrogen production was pretty efficient. And as I said, in this case, there was no sulfur deprivation, but actually if they combine it with sulfur deprivation, then the rate of hydrogen production increased. So they get about 20 microliter hydrogen in per milliliter culture in about 2 days. And one problem is that E. coli, of course, requires a carbon source, so this cannot work in a photoautotrophic manner. Then 2 methods were developed that are based on keeping the Calvin-Manson cycle inactive, because actually the main role of the hydrogenases is to dissipate the excess energy before the Calvin-Manson cycle gets fully active. So then that's the idea. So let's keep the Calvin-Manson cycle inactive so that the hydrogenases can receive electrons for a longer period of time. So there are 2 methods that are pretty similar. So hydrogen production under pulse elimination that I will explain in a moment, and then photoautotrophic hydrogen production with substrate limitation of the Calvin cycle, and this is our method that I will describe in detail. So under pulse elimination, the method is very simple. So we have the cultures grown in top medium or TP medium, so without acetate. And then the cultures are directly put in this cuvette, and then they receive pulses of white light of this medium light intensity. So they get dark and low light pulses. And the idea is that this treatment prevents the activation of the Calvin cycle. There is no oxygen evolution, and actually the procedure works at quite a low cell density, so very thin cultures. So this is the results. So you can see that oxygen evolution, that is the blue line, so the amount of oxygen decreases very strongly after a few minutes in the culture, and then hydrogen production starts, that is the red line. Here they use the train of 1 second light and 9 second dark periods, and then this is how they could achieve that there was no oxygen accumulating in the headspace of the culture. And then this works also on a time scale of a few days. So you can see that if they use this like dark pulse treatment, then there's continuous hydrogen production lasting for about 2 days. And actually, if they use the acetate, then the efficiency of hydrogen production was increased. And then they get about 55 microliter per meter in 3 days, which is similar to the previous method. But here, the main important thing is that we don't need sulfur deprivation or special treatment, so this can work in a quite a sustainable manner. And finally, this is our protocol. So this is also very simple because we have, well, first we grow the algae, and then we transfer them to a closed bottle, and then we incubate them in the dark for 4 hours. That enables the expression of the hydrogenases and their activation. And then we put them to continuous elimination, 4 days in this case, and we use also relatively high light intensity, 350 microeinstein. So we sample the headspace regularly to see how much hydrogen we have, and we also flush the headspace in order to remove the produced gases. Yes, so this paper was published in 2018, and we also have a European patent on this procedure. So this is how it works. So first of all, we compared top medium with TP, so without acetate. You can see if we don't have acetate in the culture, then we get continuous hydrogen production here for 24 hours. And then at the same time, the oxygen level remains very low. That is the green line on this side. Yes. So here, this is just saying that if we don't have acetate, then we can get continuous hydrogen production, and the oxygen level remains low. So here we show that electrons originate from water oxidation, so this is why we employed DCMU. So you can see that if we have DCM added to the cultures, then hydrogen production stops immediately. And also if we add CO2, so in this case, just to activate the Calvin-Manson cycle, then the hydrogen production starts. So that this really shows that first of all, there is a competition between the Calvin-Manson cycle and hydrogen production, and the electron transport, the photosynthetic electron transport, is absolutely required for the maintenance of hydrogen production. So we checked what are the limiting factors, so how could we make this process more efficient. So we could see that if we add the ionophore FCCP, then hydrogen production goes up very strongly, but of course it's not sustainable. We cannot remove the delta pH in a living culture. But anyway, it shows that the proton gradient is an important limiting factor. It works via photosynthetic flux control. Then oxygen, it's obvious that if we have oxygen, then the hydrogenases are getting inactive. And normally we have very low amount of hydrogen, so it's about 0.1% in the headspace. But if we remove it completely, like with a glucose oxidase ascorbate system, then actually the hydrogen production efficiency increases even more. So there's still some space for further improvement. And then this is what we found. So we used a very simple oxygen absorbent. This is shown on the slide, on the photo. So this oxygen absorbent is simple. It's an iron sodium chloride based absorbent. Actually, it's used in the food industry. And with this one, we could decrease the oxygen concentration in the headspace even more. And then along with this, we get increased hydrogen production. And the good thing is that when we add the oxygen absorbent, then the hydrogenases are preserved even better. So you can see it here, that even the in vitro activity is more preserved. This is showing that the cultures don't get degraded during the 4 days of hydrogen production, so the chlorophyll content remains stable. The photosynthetic efficiency is also quite good. It's about 0.4 after 4 days. And then all the photosynthetic subunits are very nicely preserved, so we don't have this degradation anymore as we had with the sulfur deprivation method. And then after like 4 days of continuous hydrogen production, the cultures can be efficiently regenerated. And then if we put them to a second round of hydrogen production, then we basically get the same efficiencies. So this is a process that can be repeated several times. And then thinking about, again, further improvement. So this was our very simple bark culture study. But then we thought to change the geometry of this photobioreactor, if you call it like that. So because we decided to increase the gas to liquid ratio and surface, so this would help the gases to get out of the culture so that they would be less inhibitory for the hydrogen production process. So this is what we call thin cell layer culture. And then we did the experiments at 2 different chlorophyll contents because we thought that light absorption is very important, so if light gets fully absorbed or not. And then we subjected the cultures to 2 different light intensities, like this 350 that we used earlier, and very high light intensity, 1000 microeinstein, which is more or less the light intensity of full sunlight. And here you can see that just by using these thin layer cultures, we could get quite a high increase in hydrogen production. And then this worked also very nicely at 1000 microeinstein. It's important to emphasize this because in the case of sulfur deprivation or with any of the earlier methods, it was not possible. So the cultures were killed like within a day if you subjected them to this light intensity. So here we can see that the efficiency of hydrogen production was not decreased when we subjected the cultures to this really high light. Yes, and then we got about 600 microliter per milliliter hydrogen, which is more or less 10 times more as with the earlier methods. And then, yeah, this is just some more details. And then we used the cyclic electron transport mutant called PGR5 and PGR1. Here I'm showing on the lower slide for PGR5 because it was really nice. So you can see that we get a 2.5-fold increase in hydrogen production. So now we get to the range of over 1 milliliter hydrogen per milliliter culture. And you can see that this PGR5 mutant is really nice because even at this very high light intensity, its photosynthetic apparatus doesn't get degraded. The chlorophyll is fully maintained. And also its activity, the activity of hydrogenase is kept at 100%. And it's really important to emphasize this because with the early methods, maybe about a few percent of hydrogenases could be kept active after a few days of hydrogen production. So to summarize this part, so the keys to hydrogen production is to keep the Kelvin-Benson cycle inactive, just to limit the competition with hydrogenase for electrons, and we have to remove the evolved oxygen because hydrogenase is still very sensitive to oxygen. So the advantages of our method is that hydrogen production starts immediately upon elimination, so not after like a few days. And hydrogen is produced from water. We don't need any organic carbon sources, so no acetate requirement. And it's possible to produce hydrogen at the intensity of sunlight. It lasts for several days, and it has a high productivity. The results on the PGR5 mutants were published last year in Bioresource Technology. That's the reference here. Yes, and then we think that alga cultures can be regenerated and also used in downstream processes. So there are also, of course, more possibilities to improve hydrogen production, so by different genetic methods, such as that fusing ferredoxin with hydrogenase, that is shown at this side, or we can just modify the docking of ferredoxin to FNR, so to decrease the competition between ferredoxin, between hydrogenase and FNR. This is already shown, so we can downregulate photosystem on electron transport, and we can also think about producing oxygen-resistant hydrogenases. There are some progress in this respect, but we don't have a wonderful hydrogenase that is not oxygen-sensitive at all, unfortunately. So we think that is open vistas to bioindustrial hydrogen production. And then we would like to design photobioreactors with effective gas removal based on this simple system. So we would like to build such a model photobioreactor that can be used later for industrial purposes. And this is a circular bioeconomic concept. So first we would like to produce biomass using agricultural food industry waste. Then we have the hydrogen production mostly from water and sunlight. And then the spent biomass could be used for other purposes. And then just a few words about electric current production by algae. Actually, it's based on the same process, which is photosynthesis, and that the electrons are used for something else, and in this case, current production. So it has some advantages, is that sunlight is used by photosynthetic organisms. It's a self-repairing system. We don't need organic carbon source, and CO2 fixation can be done in parallel with current production. Yes, but currently we have a low energy conversion efficiency, so we would like to improve on that. And these are the microalgae-based photovoltaic devices that we use. So we have an electromediator called ferrocyanide, and this is the mediator that receives the electrons and then transfers them to the anode, and this is how the current is produced. So this is just a system that we're using. It's quite in an early stage. Yes, so we are trying to find the right algal strains to improve the efficiency of these devices, and of course we are also working on revealing the exact mechanism of current production. And it's similar to hydrogen production, so we need to eliminate the competing processes, and there is also a need for new electrode materials that could also improve the efficiency. Yes, this is my research group in Szeged. So these are the people who were mainly involved in the presented projects. And we have also collaborations with Aarhus University, Tel Aviv, and Turku University. And thank you very much for your attention. It was great. Thank you.
Hello, madam. See, your talk was very interesting, and some practical implications are also there. What I learned by humble knowledge is that you have discussed a very complicated electron transport system in a blue-green algae Chlamydomonas reynoldi. But at this, what I learned is that you have evolved a strategy to six-fold increase in the electron transport system, hydrogen production. So I think this is the thing which I have imbibed. Now I think this is the house is open for the discussion, and the distinguished participants who want to know more from you may interact and questions. But only thing is that time is a constraint. So within the time limit, maximum interaction is invited. Am I right, madam?
Thank you.
Yeah, Sylvia, it's a very, very good work, and I appreciate your the work. It's not a question, but it's a clarification. You said thin cultures, the hydrogen production is more, right? So if you put them in reactors, what will be the hydrogen production? That is my first question. And the second question is, what you can also see the over-reduction at the acceptor side of photosystem 1.
Yes.
I mean, when you use 1,000 microinstants. Okay. So what is the role of the H2O2 there? Because there will be over-reduction.
Yes. Okay. I will repeat the question. So first about thin layer cultures. So if there are any photobioreactors that are suitable for this kind of system, yes, there is one, and then we have that was developed at the University of Nantes in France, and we have a collaboration with them. So now right now we are building a photobioreactor for greenhouse purposes, and then if this model is really promising, then they would upscale it for semi-industrial scale.
No, but why it is only till culture you can see the more hydrogen? That's my
Yes,
question.
so
any mechanical reason?
Yes, so the thing is that oxygen has to get removed from the liquid culture as soon as possible, because otherwise on its way it would inhibit the hydrogenases. So we need a very efficient oxygen removal, and then if we have a large surface, so with a relatively thin culture, then this can work more efficiently. And on the other hand, if we have a dense culture, then the light absorption is also more efficient. So these are the two criteria that we considered when thinking about this system. And also actually we have tried, if we have a very small headspace, then the efficiency of hydrogen production really goes down. It's really a tremendous effect. Yes, about H2O2, so about oxidative stress. So first of all, we have very low amount of oxygen present in our system. So that's why oxidative stress in general is not such a problem.
No.
And you can see it with the PGR5 mutant, that even after like 4 days, but we have also 6 days experiments, even if we shine this very strong light, then we don't have any degradation of the photosynthetic apparatus. So in theory it could be a problem, but we are lucky that we could really remove the oxygen very efficiently, so this problem is eliminated. Thank you.
Thank you very much.
Dr. Sylvia, your talk was very thought-provoking, and being a person of cyanobacteria, I may ask one question related to your second part, where you planning to produce some electricity, because the electron transport system which is found in algae, they have a certain capability to transport, because it's not like a circuit that you just put the thing and it will pass on the next level. So what do you think that how you avoid the process of the saturation, as Professor Raj Gopal has asked, that if you put a light, high light, and it will start saturating. So how you how do you avoid this process of saturation of these complexes?
Do you mean photosynthetic flux control?
Yeah.
Yeah, that's a problem. I mean, we know that this is the main limitation, so somehow we have to solve this, and of course we cannot dissipate the delta pH, we cannot use ionophores. So I think the only way I can think about this is to further increase the rate of electron transport to hydrogenase, because this could actually mitigate this problem of the slowdown of the electron transport. So we have to also eliminate the other competing processes, so everything would go to hydrogenase. But I'm sure that the capacity of the electron transport is much higher than the capacity of hydrogenase to accept electrons. And this is the key thing. This is what we have to work on. Thank you.
Yeah, my questions are very fundamental. Like you said, when you remove carbon dioxide from the system, then electron transport or hydrogen production is more.
Yeah.
So how the cell will sustain over a period of time? That is first.
Excuse me?
In the absence of carbon dioxide, how
Yeah.
the system will sustain
Yes.
over a period of time? That's the first question. And second is that you mentioned that H2O2 will be ultimately produced in the process, one. And second thing is over reduction will also cause some of the damage to these pigments.
Yes.
So then again the production of hydrogen will decline over a period of time. So how these two problems you going to overcome if you have to have some
Yes.
continuity in it? Thank you.
Yes. So about the CO2, so first of all we have to grow the algae, like in CO2 replete conditions, so we add either CO2 or we can also supply some organic carbon sources to have a dense biomass in large quantities. But then we can just simply transfer the cultures to dark conditions to induce the hydrogenases, and then we have continuous hydrogen production for a few days. And of course, we have to regenerate the cultures. I mean, nothing can live from nothing, so. So then we can have a regeneration phase in which we again supply the cultures with carbon dioxide, and then after like 2 days we can start a new cycle again. This is how it will work, and under laboratory conditions it's quite promising. I think it could work in the industrial level as well. And yes, about degradation of photosynthetic apparatus, well, I can only see that our results show that it is not a problem. At least using this BGF5 mutant, they can really support, they can really survive this very, we can call extreme conditions, since we have no CO2 fixation and we don't have, so we have only hydrogen production. There is no oxygen in the system, so it's quite a particular situation, but they seem to survive quite well. So I think it can work, yes. Thank you.
Thanks very much for your comments and the discussions.
Thank you very much.
Distinguished participants also, thanks.
Thank you. Ma'am. Thank you, Dr. Sylvia, for your inspiring talk. Now, I invite Chair of the session, Dr. Anil Kumar, to present the memento to Dr. Sylvia as a token of our gratitude. Sir, please.
Yeah, may I request Dr. Rajesh Mehrotra from Birla Institute of Technology and Dr. Sujata from Acharya Ranga Agriculture University to provide their presentations for the next session. I think that can be collected in the desk outside. Please, otherwise they cannot arrange for the next sessions. Thank you.
Good morning all. So we are up here for the next presentation by Dr. Dinakar Challabatula. So let me take this opportunity to invite the speaker for this session. So Dr. Dinakar has obtained his PhD from the University of Hyderabad, India, in the field of molecular plant physiology. So he was also a postdoctoral fellow at the Institute for Molecular Physiology and Biotechnology of Plants, University of Bonn, Germany. His research focuses on molecular physiology of drought and salinity stress responses, and he is also currently a reviewer for many international national funding agencies, as well as peer-reviewed journals across the globe. He was also a visiting scientist at IMBio, Germany, and he's also a visiting scientist to the Swedish University of Agricultural Sciences, Uppsala, Sweden. He has published to his credit more than 25 research articles in international peer-reviewed journals and has more than 1,500 citations and an H-index of 15. So he's today here with us to speak on the wonder that resurrection plants do to tolerate desiccation. So thank you, sir, for this wonderful talk. So over to you.
Thank you, madam, for the introduction. And first of all, I thank Professor Jose Couture and Calicut University for giving me this opportunity to share some of my research ideas and results. Yeah. As the title says, I have been working on this resurrection plants from past 12 to 13 years. So here what we are trying to do is we are trying to understand the mechanisms that are involved in tolerating this high stress, what is called as desiccation. It's not working. Yeah. So I am from Central University of Tamil Nadu, which is in the southern part completely. This Tiruvarur is called as the temple city. There are a lot of temples here. And then we have two campuses. One is the residential campus, which is on the right side. Then I have some problem. Yeah. So we have two campuses. One is residential campus. The other one is the teaching campus, academic campus. And then which is in between, we have the river Vettar. This is our university. Coming to my lab, so we have been working on two different aspects which are related, I say. So its major focus is on abiotic stress tolerance. So we are working on the first one, which is called as chloroplast mitochondrial interactions, where we are deciphering the role of alternative oxidase, a mitochondrial enzyme, and then we are correlating that with the stress tolerance and the chloroplast mitochondrial interactions. And the second part, which is going to the main topic today, where I will be discussing more about the desiccation tolerance in some of the selected resurrection plants. So we have been using, the first plant is Oropicium. The second one is the Craterostigma. The third one is Lindenia brevidens. And for comparative studies, we use a sensitive plant, which is also belonging to the same family like Craterostigma and brevidens. Then that is called as Lindenia subracemosa. So the question arises why we need to work with the resurrection plants. So the picture itself is self-explanatory, where when the plants experience drought, you see the leaf curling along with loss of pigmentation, and automatically if you don't water these plants, they may die. Now if the prolonged dehydration happens, the second picture where you can see, that is called as desiccation, where when you water them, then they cannot restore back or they cannot come back to the live state. So what we want is the plants which can tolerate drought stress, and even they can survive under drought stress. When the rainwater comes or when the water is supplemented, they can grow back to the normal plants, as a normal plants. So now when we are talking about drought and desiccation, there is a fine limit in between these. So drought is experienced by many of the plants, and then they can revert back. If there is a mild stress, mild drought stress, many of the plants can revert back and they can grow back easily. But when the drought stress is prolonged for a longer days, for example 30 days, 40 days, that may lead to irreparable damage. So generally what happens is, that is called as desiccation. So when the relative water content in the leaf or a cell decreases to less than 20% or 5%, then automatically majority of the plants, majority of the crop species, they cannot survive. So automatically what happens, they undergo the never return phase, that is called as desiccation. So here in this slide, the cell size, when you take it as one of the parameter, then automatically when the water is lost from the cell during dehydration, the cell shrinks and the relative water content will decrease. So majority of the crops, when you see, their relative water content, when it comes to 60% or below, they may not rehydrate back, they may not grow back. But what we have is, here on the right side you see desiccation tolerant plants, they can tolerate up to 5% relative water content, and then when the water is given back to them, they can rehydrate back. So these can be used as models to understand the mechanisms of dehydration tolerance or desiccation tolerance. So in our lab, we use Arabidopsis thaliana as a sensitive variety for normal salt tolerance, and then Eutima salcigenium, which is both drought and salt tolerance. So we use these as models to understand the drought tolerance behavior and salt tolerance behavior. And we use Lindenia seborrheicosa and Lindenia brevidens as drought sensitive and desiccation tolerant organisms. So to identify the mechanisms that are responsible for drought or desiccation tolerance. Now, majority of the crops, as I told you, they are completely either drought sensitive or desiccation sensitive. Automatically what happens is when the relative water content goes lesser than 40%, then automatically these cells will shrink and then there is no way go back. So they cannot return back to the normal life. So automatically when these resurrection plants in the below one, when they are desiccated, they can rehydrate back. So if you can identify the mechanisms that are responsible for their desiccation tolerance, then we can integrate those pathways or genes into the modern day crops, and then we can at least expect some drought tolerance. That is the hypothesis and objective which we are working on. So now generally when we look at these resurrection plants, generally when the water content decreases, automatically if you take either transcripts or proteins or metabolic changes that occur at different relative water contents. So on the x-axis we have 100%, 80%, 60%, 40%, 20 and 0. When you look at these different relative water contents, the plants that are experienced to different drought conditions, when they are having relative water content at these levels, automatically the genes or the proteins or metabolites, the changes that we observe, they can be categorized into 7 different types. For example, when you look at type 1, the down one, all the genes these genes are constitutively expressed. That means when the plant is experiencing drought at different levels or different days, when the relative water content is decreased periodically, then the genes, particularly for example actin, tubulin, GAPDH, these are constitutively expressed. That means these are called as housekeeping genes. So they don't vary with the relative water content. But when it comes to type 2, then automatically what happens, initially when the water is surplus or when the plants are turgid, when the leaves are turgid, their expression levels are lower. But when the stress is increased, they are increased. So up to desiccation, they will be increased. So they can be stress responsive genes. So they can be induced during stress, and then these are called as stress responsive genes. Normal heat shock proteins, they can be taken as stress responsive genes. When it comes to type 3, majority of the photosynthesis related genes, what happens is they are highly active when the water is in surplus. When the relative water content is above 90%, then photosynthesis will be actively growing. But when they dehydrate slowly, then photosynthesis gradually decreases. Then in these plants, the astonishing feature is, even though the photosynthesis decreases, then when you rehydrate them for 24 hours, the photosynthesis goes back and it comes back to normalcy. So the majority of the photosynthesis genes, they come under type 3 category of genes. When you look at type 4 category, then these genes are slowly induced during dehydration. But as the dehydration crosses 40% or 50%, or less than 40% or 50%, then they will be completely downregulated. So these kind of genes are dehydration inducible genes. In majority of the non-resurrecting plants or drought sensitive crops, so these kind of genes are seen. So automatically when the stress increases, dehydration comes, the genes will express. Then automatically when it is prolonged for a longer duration, these genes will downregulate. So these are common for normal non-resurrecting plants. When it comes to type 5, then if you see, they will be expressed lower than the type 7. But what happens? They decrease like photosynthesis genes, but during desiccation, when the drought process is increased, they will upregulate. Then when it comes to type 6, these are also like photosynthesis genes. Initially they will be higher, but automatically they will be at a very lower phase. They will be completely decreasing. But what we are interested is desiccation induced genes, which are not present or their regulation is not present in the drought sensitive crops or desiccation sensitive plants. So in this category, what you can see is type 7 category genes you can see where here they are not at all induced under control conditions, they are not at all seen, but only after the relative water content decreases below 40% or 30%, then they will be induced. So majority of the LEA proteins or dehydrins, so they come into this category, and then they can be used to to consider as markers for understanding the mechanisms of desiccation tolerance. Yeah. So now, till now, whatever I was telling is about the vegetative desiccation tolerance. It is not new. In regular everyday life, we see it. So we see it in the form of seeds. We see it in the form of pollen. So seeds and pollen are generally desiccation tolerant because the seed completely dries off. So when you add water to it, it germinates, and then the desiccation tolerance is lost. So automatically, now what happens in these resurrection plants is, now the pathways that are present in the seeds, they have been evolved or they have been transferred into the leaves, vegetative tissues. So this we call it as metabolic wiring, rewiring. So the pathways that are present in the seeds, these pathways are active and are modified in the leaf tissues. So that is what is generally observed in the resurrection plants, in the leaves. Now see this is a very classical example of resurrection plants. So Cratostigma is used as a dicot model, and Europicium thomaeum is a monocot model. One problem with Cratostigma plantaginum is it is an octoploid plant. So hence genetic transformation experiments are very difficult. The genome sequence is not available. So we need to rely on the transcriptomic or proteomic data. Whereas Europicium thomaeum is a diploid plant, it's a grass. So it's easy to transform. Now we are standardizing the transformation protocols. So these two can be used as models to understand the mechanisms of desiccation tolerance. So as you can see, fully hydrated plants, now when you dehydrate them for 21 days, they can be completely desiccated. The chlorophyll is completely lost. But when you add water, within 24 hours, they can rehydrate. Within 4 days, they can fully rehydrate and they can photosynthesize. They can do well. Now, opposite to it, seeds, when they are desiccation tolerant completely, when water is added, when they start to germinate, for example, when testa is ruptured, or radicle is protruded, or root length is seen, or appearance of root is, generally the desiccation tolerance is lost. So the seed, when it germinates, the desiccation tolerance strategy is lost. But upon treatment with abscisic acid, where you can see on the pink line, upon treatment with abscisic acid or osmotic stress, automatically this desiccation tolerance strategy can be revived. But until when it is revived, only unto stage 3. When it goes to stage 4, when the plant is completely germinated with radicle and plumule, the desiccation tolerance strategy cannot be restored. Even in desiccation tolerant seeds like Xerophyta viscosa, the below one, the same strategy is being followed. So the plant is desiccation tolerant, but seed, when it germinates, so initial stages of germination, there is no tolerance, but in the later stages, once the leaves are completely formed, they will be completely tolerant. So as I told you, we have been working on this. For today's presentation, I will be using Cratostigma plantaginum, Lindernia brevidens, and Lindernia subracimosa. Europicium, I will do it later. So these 3 plants are used in our lab to understand the mechanisms. So just a video to show how it is resurrected. I hope it comes. Yeah. Is it working? Yeah. Just a small video where it's the desiccated plant, if you keep it in water, then what happens is within 24 hours, it can rehydrate and it can grow back. So the photosynthetic values can be easily recorded, and then chloroplasts can be isolated for functional studies, and many other experiments can be performed. So this is 6 months old plant. When we keep it in water for 24 hours, within 24 hours it got rehydrated. What? Finished? Oh. Yeah. Okay. So then these plants can be used as models, I was telling you. So what I do, how do I test this? So the initial simple experiments that can be done in the lab are simple relative water content measurements, where you can see as they dehydrate, the relative water content decreases completely, and then when you keep it for rehydration for 24 hours or increase that to 48 hours, the relative water content will be increased back or restored back. So whereas in sensitive plants, so this relative water content will not be restored back. The plants are completely dead. So relative water content measure, simple relative water content measurements can be done to check whether the plants are desiccation tolerant or not. The next important thing that can also be done is after 24 hours rehydration, you can check the photosynthetic activity. So once we can check the photosynthetic activity, then as you can see in the first pink line, the photosynthetic activity at different light intensities that is increasing. Then when they are desiccated in the second stage, they are completely in the yellow line, they are at a basal level, very base level. But when they are rehydrated, automatically they are going back. Since they are only 24 hours rehydrated, we are getting the photosynthetic activity here. If we rehydrate for 48 hours or 72 hours, automatically it will go back to the normal stage. So what are the different mechanisms? We have been working on different kinds of mechanisms. For today's talk, I will be talking only about LIA proteins. So now when we club all these mechanisms that are active in these resurrection plants, we can see the first thing is changes in cell wall properties. So there are some proteins which are localized in cell wall. They increase the metabolic flexibility of the cell wall. We have identified some glycine-rich proteins which are binding to the cell wall, and then these are aiding in cell wall flexibility. Then leaf rolling and anthocyanin accumulation, and then some phenotypic morphological changes are observed where the plants are completely they avoid themselves from the excess light mediated reactive oxygen species or oxidative damage. And the third one, some novel antioxidants are seen, like cysteine peroxidinoxins, polyphenol oxidase, and these kind of novel antioxidants are seen along with the commonly observed antioxidants to protect them from reactive oxygen species mediated oxidative damage. Some unusual carbohydrates like octulose, stachyose, verbascose, these are accumulated in these resurrection plants so that the the osmoticum can be maintained so that the cellular membrane damage is decreased completely. Then along with that, we have seen some lipid moieties which are being increased, lipid remodulation we have seen, some small RNAs are there, some long non-coding RNAs we have seen which are generally expressed but they didn't code for any proteins. Now the strategy what we have used is, since the genome sequence is not available, it is an octoploid plant, we thought that okay, we will go for the transcriptome sequencing at different stages. So what we have done is we have taken the relative water content of 100%, around 90-95% we got it, then we partially dehydrated them for 48 hours, then we desiccated them for 15 days and then rehydrated. At each stage we have extracted the RNA, then we have made the CDN library and then we have gone for the transcriptome sequencing. Similarly, we have done the transcriptome sequencing from the two other plants, all belong to the Lindanaceae family, Cretostigma plantagenum and Lindana brevidis. These both are desiccation tolerant, and Lindana subracimosa is desiccation sensitive. Now one unusual feature that we have observed is 33% of the transcripts that we see during the transcriptome sequencing are taxonomically restricted genes, which we call as orphan genes. So their sequence similarity is not matching with any of the genes that are sequenced until now. So these can be the reservoirs, or these can be the candidates for gene identification, which we have not explored till now. So we need to understand the mechanisms, or we need to understand the importance of these genes for the stress tolerance. But what we have considered now is the 67% annotated genes. Out of these 67% annotated genes, whichever gene is abundantly expressed on the type 7 category of genes, whichever is abundantly expressed during desiccation, that we have considered to be compared with sensitive plants. Now this is what we have used for Lindana brevidens plants. Also we have used 100% relative water content, 50 to 55% relative water content for dehydration with 2% relative water content, and rehydration for 48 hours where we get 80% relative water content. In sensitive plants, desiccation sensitive plants, we have not used desiccated and rehydrated. We went till late dehydration because we know that after late dehydration they will not be rehydrating back. Now, one of the most abundantly expressed gene from the transcriptome sequencing were found to be late emergence abundant proteins. So these genes, now when we looked at the literature, they are not only present in the plants, they are present in vascular plants, they were in non-vascular plants, they are in fungi and bacteria, they are seen in worms, they are in arthropods. So initially they have been identified in plants, late emergence abundant proteins are identified in plants, but when we look at the literature, they are present everywhere. So they are dehydration inducible, they are salt inducible, they are osmotic stress inducible, they are majorly stress inducible genes. So generally when we look at the literature, these dehydrins or these LIA genes, they are generally induced during drought stress, high salinity, cold stress, and then when these stresses are imposed, automatically their gene expression is induced. So under control conditions, their gene expression is not at all seen. Now, when you look at the biochemical mechanism of how this activation happens, it is an ABA regulated gene. So the ABA that is increased during the stress conditions travels to the xylem, and then when the stomata is closed, automatically it activates the transcription factors that are present on the LIA genes, and then LIA genes get activated. So it is ABA mediated regulation of LIA genes. Now we have identified the most abundantly expressed genes and we have categorized them. Out of these, as I am showing only two to three genes, later in later slides I will show that all the genes which we have identified. CP1124 was abundantly expressed during DL is dehydrated leaf or desiccated leaf, and DR is desiccated root. So it is selectively expressed during desiccated conditions in both vegetative tissues of leaves as well as roots. In the similar way, CP619, Keratostigma plantagenum, 6th colony and then 19th plate, they have also expressed during desiccation as well as desiccated root and desiccated leaf. Similarly, when you look at a western blot, in the western blot also, as you can see, in the fresh leaves, you don't see any protein expression. In 4 hours dehydration, you don't see. But in 4th and 5th slot, when you have these 24 hours dehydrated leaves or 100 millimolar sodium chloride, you can see the expression of 1124 as well as 619. So when we compare these with the sensitive plants, now in the sensitive plants, when the desiccation is given, so the below slide shows desiccation tolerant plants as well as sensitive plants. When we see in desiccation sensitive plants, here you don't see any expression of it. In this one, you don't see any expression of it. Whereas in tolerant plants, you see the expression of 1124 as well as 619. So only in tolerant plants, we can see the expression of 619 as well as 1124. In the sensitive plants, generally during desiccation, you don't observe any expression of it. Now, in order to see whether this 1124 can be accumulated for a longer time, when we give repeated stresses and then repeated recovery phases. So in order to do that, what we have done a simple experiment where we have taken the fresh plant, we have given a stress for 6 hours, desiccation, dehydration stress for 6 hours, then rehydrated for 18 hours, and then that we simultaneously we did it for 3 to 4 cycles. Like we have S1, stress 1, then recovery phase for 18 hours, R1, then stress 2, then again stress 2 for 6 hours, again recovery for 18 hours. Like this we went till R3. Then when we went for R3, then when we look at the gene expression of 1124, this 1124 at every stress, it is induced. It is induced at a higher level. Then when it is induced at a higher level, we wanted to see when we recover the plants completely for 24 hours. Well, during the recovery phase, whether the gene expression is still there or not. So we have checked the gene expression is still active. And when we look at the protein here in the S4 condition, so in the 24 hours, then 2 days, 4 days, until 4 days during recovery, the protein is still accumulating. That means it acts like a memory for the cell so that it is accumulated and it is protecting the cell membrane from dehydration mediated injuries. Now in sensitive and tolerant plants, when these kind of differences were observed, we wanted to see whether is there any phenomena that is happening in between sensitive and tolerant plants to protect the RNA or to degrade the RNA. See here, when we checked it, we have used some of the housekeeping genes like actin, like GAPDH and EF1 alpha, which are known to be constitutively expressed under all conditions in normal plants. So when we look at their expression under untreated leaf, DL is desiccated leaf, RL is rehydrated leaf, they are constitutively expressed in tolerant plants, desiccation tolerant plants. When we look at sensitive plants like Suprasymosa, generally in untreated leaf they are present, but when they are desiccation and dehydration, they are not seen. There is no concept of rehydration in desiccation sensitive plants, but during desiccation, these transcripts are completely lost. Now we wanted to see whether this is the mRNA loss or mRNA not, there is no transcription of mRNA or the polyribosomes are dismantled. So when we isolated the polyribosomes and we have extracted the RNA from polysomes, then when we checked at polysomes, automatically we see there is no polysome at all. That means there is no active translation. There is no mRNA that is being formed from the desiccated leaf of the Lindenia suprasymosa, the sensitive plants. This tells us that there is some phenomena of mRNA protection or RNA protection in tolerant plants, whereas that protection mechanism is lost in the sensitive plants. Now this is a simple experiment which we have done where we wanted to see the yield of RNA per 0.2 gram of tissue. So in sensitive plants as well as tolerant plants during different dehydration conditions. When we do this for different dehydration conditions, as you can see, the % changes in RNA yield are shown on the right side. So where you can see that the RNA yield is more or less similar in desiccation tolerant plants, whereas during dehydration, different days of dehydration, the RNA yield is decreasing. So here, pictorially, when you can see here, this here in the right side of your slide, in the desiccation sensitive plants, during desiccation, automatically majority of the RNA is degraded. Here in these slides, the RNA is degraded. So RNA degradation can be one of the simple mechanism which these plants adapt in order to what you call kill the cells. So the mRNAs are all completely selectively degraded in sensitive plants, whereas in tolerant plants, these RNAs, selected RNAs, they are protected, and then when the rehydration phenomena goes on, the photosynthesis genes can be activated back because polyribosomes are still active. Now, as I told you that we have isolated many of the genes which are completely induced during desiccation in resurrection plants. When we saw these genes and when you try to categorize them, almost all of these genes, when they are categorized them, they all belong to the same family, which we call them as dehydrins, group 2 Lea genes. So these group 2 Lea genes can be identified based on the presence of K segment, S segment, Y segment, and the nuclear localization signal. So when we categorize all these genes, almost all these genes are induced during complete desiccation in both leaves and roots, and then these, all these genes belong to the same family, which we call them as dehydrins. Now when we check their expression during dehydration and rehydration in both leaves, roots and callus, almost all genes are induced during dehydration. They are all induced during dehydration. But one specific phenomena that what we have observed is in the callus, this desiccation phenomena is not at all seen. So these callus tissues that are generated from either seeds or leaves, they lose the desiccation tolerance strategy. So they are here on the right side, you can see these callus tissues, when they are desiccated, they don't rehydrate, they are non-viable. But when they are treated with ABA, automatically they restore their resurrection capability. So the callus tissues that are generated from the leaves of desiccation tolerant plant, they are completely losing the desiccation tolerance. But when the ABA is given, these genes are induced and then desiccation tolerance is revived back. So the same thing is shown in the below plot, where in the control and dry, you don't see any dehydrins, but when you give them with ABA and ABA dry, then automatically the dehydrins are induced. That means the dehydrins are selectively induced when you have ABA in the medium. So in order to decipher, in order to know more about this ABA induced activation of the dehydrins, what we have done, we have generated transgenic plants with CP Lea-like 1124 gene, a promoter with GUS, and then we have transformed the Arabidopsis plants. Once we have Arabidopsis plants, under control conditions without ABA, the gene induction is not seen, which indirectly we have scored for the GUS activity. The GUS activity is not at all observed, but when you treat the seedlings with ABA, automatically the GUS activity is seen. So now we have so many dehydrin genes. For all the dehydrin genes, we cannot generate the transgenic plants. So what we have done, we went for a simple approach where we do a transient transformation with CP Lea-like 1124 with GUS, and then we can transiently observe the GUS activity in the leaves. So what we have done is, we have isolated a 1 kb promoter, then we have decreased that 1 kb promoter to the active promoter size, where we got 307 base pair as the active promoter region, which is driving the GUS expression. And then what we have done, we have cloned it in PBIN, and then by co-cultivation as well as by transient transformation using particle bombardment, we have bombarded into the leaf, and then we have seen the activity of the GUS. As you can see now, this 307 base pair promoter from Cretostigma is able to drive the GUS expression in the leaves of Cretostigma plantagenum. Now the question arrived, when we submitted the paper, the question arrived that is it because of agrobacterium infection or is it because of the real promoter activity? So in order to answer that, what we have done is, we have taken the small leaves and the larger leaves of Cretostigma, and then we have infected them with the agrobacterium, and we seen the activation of CP Lea-like 1124. So we know that during desiccation stage, the Lea-like 1124 is activated, but during agrobacterium, there is no activation at all. So it selectively states that the Lea-like 1124 is activated selectively during dehydration, not because of wounding, that is because of agrobacterium infection. Now in the simultaneous using the same strategy, we have identified the promoter regions from Lindenia brevidens, then Cretostigma plantagenum and Lindenia subracemosa plants. In Lindenia subracemosa, since we have only 2 ABA responsive elements in the promoter region, we have gone for extension of the promoter to 488 base pairs, where we got 4 ABA responsive elements. then what we have done is when we compare these promoter regions, we have seen the differences in the promoter, promoter sites, these cis-acting sites in these promoter regions different. Then we fuse these promoter regions to the Gus gene, and then we transiently express these in the Cratostigma leaves or Lindenia brevidens leaves or Lindenia subracemosa leaves. In the first figure, as you can see, when you have taken this 307 base pair promoter region of CP1124, then in the CP, when you bombarded and treated them with mannitol and ABA, we have a higher induction of Gus. In the similarly, then when you transform that to Lindenia brevidens, then also we have higher Gus activity. And again, in Lindenia subracemosa also, we have higher activity. So that means CP Lea-like 1124 promoter with ABRE elements and a DRE element, he is able to drive the Gus expression in all the three leaves. In all the three leaves, both sensitive and tolerant, it is able to drive the expression. Now when it comes to second case, LB Lea-like 1124 promoter, then it doesn't have a DRE element, the element which is shown in yellow color there, it doesn't have, it has only ABRE1, ABRE2 and ABRE4 elements. Now when these elements are taken, now the activity, promoter activity is lesser than Cratostigma plantagenum. So here it suggests that some of the transcription factors maybe are missing in Lindenia brevidens. Now when it comes to subracemosa, when you take in the subracemosa promoter, which is completely sensitive promoter, and when we transform that to Cratostigma plantagenum, which is desiccation tolerant, the promoter activity is less, the Gus expression is less. Similarly, in Lindenia brevidens also, the promoter activity is less. In subracemosa also, the promoter activity is very less. This suggests that the promoter, LS-Lea promoter, is unable to drive the expression in either desiccation tolerant plant as well as sensitive plants. So this whole slide says that promoter activity can be scored to check whether this, the gene can be active in the plant in resurrection plants or non-resurrection plants. So as you can see in the last slide here, the CP Lea-like 1124 promoter, when it is taken and then when it is fused with Gus and expressed in transiently transformed in Cratostigma plantagenum, the promoter activity is more, where you can see the blue activity. Then in the Lea-like 1124 also, the promoter activity is seen, whereas in Lea-like 1124 promoter, the promoter activity is very less. The next question is, now, if you mutate that DRE element, the DRE element, the D element that is there, if you mutate the DRE element, whether the promoter is still active or not. So what we have done is, in this stage, we have taken a wild type 1124 promoter, and then selectively we have mutated some of the ABRE elements and DRE elements. So when we take this ABRE3 element, when you mutate it, the promoter activity is not decreased because ABA responsive elements are more. A2 and A1 are still there. So A1, A2, A3, these elements, they can drive the expression of Gus. But selectively when DRE element, which is drought responsive element, when you mutate it, then automatically what happened? The promoter activity is completely decreased here. So similarly in CPD A3 mutant also, when D and A3 are mutated, automatically the promoter activity is decreased. So in CP Lea-like 1124, now you have DRE element as well as ABRE elements. Even if you mutate one ABRE element, still it can drive the Gus expression. But when DRE element, any DRE element, if it is mutated, automatically the Gus expression is completely stopped. Now in the second phase, where you can see CP wild type, CPA3, CPD, now when you treat them with ABA, in the ABA also, you can see the similar kind of response. That is with mannitol, the top one. Here is it with ABA. In the ABA also, similar response is seen, which suggests that drought responsive elements are key features for these Cratostigma promoters to be active. So if you mutate these drought responsive elements, automatically the 1124 gene activation cannot be seen. So this is a water control. The last one is a water control, which we can see. So now recently what we have done, so we wanted to do a comparative analysis, but we have published with Cratostigma. So what we have done is we have taken the transcriptome and proteome and we correlated with the physiological studies. So to simplify everything, what we have done is we have taken untreated plant, we have taken a partially dehydrated one, desiccated one and rehydrated one, and we have done a comparative transcriptomics, comparative metabolomics and a comparative proteomics. So generally what happens is when these plants are partially dried, the photosynthesis is completely shut down, and then automatically the catabolism is mostly going on. So the plants go to the catabolic phase, then anabolic phase. But when they are completely desiccated, their Oxidative stress parameters are not increased, but what happens is they have protection mechanisms are more induced. Like for example, production of more carbohydrates, production of any heat shock proteins, these kind of mechanisms, production of more antioxidants, these kind of mechanisms are seen. Then automatically when they are rehydrated, 24 hours rehydration is seen, then automatically they go back to the photosynthetic phase where the synthesis of chlorophyll, then other pigment molecules, and then photosynthetic machinery is being seen. So to conclude, here in this study, what we have shown is these LIA genes are abundantly expressed during desiccation in resurrection plants, and then these LIA genes can be used as markers to see whether during dehydration, whether the plant is undergoing any dehydration or not. Then desiccation induced genes show similar properties of gene expression in desiccation tolerant plants. We have compared that with 5 resurrection plants. In all the 5 resurrection plants, the dehydrins are abundantly expressed during dehydration, whereas in sensitive plants, they are selectively degraded. They are not seen. Then the presence of promoter cis elements explains the differences in transcript abundance in the desiccation tolerant and desiccation sensitive species. So here in this study, we have identified a DRE element. In some other promoters like 00250, we have not identified DRE promoter, but we have identified only ABRE cis element. That's the ABRE cis element. When we mutate it, automatically the activity of the promoter is lost. So it is not a universal phenomena. Having a DRE element in the promoter of these genes is not a universal phenomena. Then RNA expression analysis indicated degradation of mRNA in sensitive species. This is universally observed. We have taken rice plants, we have taken some grasses which are growing outside, we have taken sensitive varieties, many sensitive varieties, and we try to dehydrate them for a longer duration, and we have seen selective degradation of RNA. This is a universal phenomenon. Then dehydrins are abundantly expressed in both leaves and roots during desiccation, whereas in callus tissues, treatment with ABA acts as a signal for dehydration expression. So in vegetative tissues, although the dehydrins are expressed, callus tissues require the external signal of ABA or osmotic stress. And now we are doing some experiments with some unusual carbohydrates. These looks like these unusual carbohydrates are also driving the expression of dehydrins, and then desiccation tolerance is induced. So still we have not published it, but this is under communication. So what we are doing now? So what we are doing is we are majorly focusing on the comparative studies where we use the salt sensitive and salt tolerant varieties of rice, or drought sensitive and drought tolerant varieties of Pisum sativum, or here desiccation sensitive or desiccation tolerant plants to unravel the mechanisms that are important in tolerant plants for withstanding the stress. So these are the strategies which we are using. For this, my student is working with valedamycin A, which is a trihalase inhibitor, and he is trying to accumulate the trihalose inside the plant cells, so and see whether the dehydrins are induced or not. Luckily, he got a result where he was able to show that trihalose is inducing under normal conditions, not under stress conditions. Trihalose is able to induce the dehydrin gene expression, and then dehydrins are upregulated under normal conditions without imposing any stress. Generally, trihalose is accumulated under stress conditions, but in the normal conditions, he was able to show the trihalose accumulation leading to the activation of dehydrins. So he is working on trihalose metabolism. We have another student where we are working on some plant growth promoting rhizobacteria, whether they are able to induce salinity tolerance in plants. So we have communicated this paper, it's already accepted. We are able to show that these bacteria, which is halophilic bacteria, halophilic plant growth promoting bacteria, they were able to promote the plant growth under normal condition as well as salinity stress conditions. Now we are looking at how the plant is being protected under stress conditions, the mechanisms of protection. Another student who is working with Arabidopsis and Telungella comparison. Here we are studying the regulation of dehydrins in this salt tolerance. So till now with the dehydration we have a clue that dehydrins are involved, but in these sensitive and tolerant plants we are looking at dehydrin regulation and expression. We are able to isolate the promoter regions, we are isolated the genes. Now we have identified, classified the genes of dehydrins in these plants. Now we are looking at the expression analysis of these genes. So now I have, I need to, that's all for this slide, for the talk. I need to acknowledge some of the people who are really helping me in this, pursuing this kind of research. Majority of the desiccation tolerance studies were from Professor Dorothea Bartels. I was a postdoctoral fellow there, and I was an INSA DFG visiting scientist, and I was a visiting scientist in Germany for quite a long time. So majority of the resurrection plants are with Professor Dorothea Bartels. So she introduced me to desiccation tolerance biology. And then I am a student of Professor A.S. Raghavendra in Masters, and then after that during my PhD and postdoctoral training also, I worked on chloroplast mitochondrial interactions. So he is my teacher, mentor and collaborator now. And then Professor Padmashri also, I have been involved in working with chloroplast mitochondrial interactions. And I am taking help of Professor Rajgopal sir for thylakoid isolation and the 2D electrophoresis and some biophysical studies. Professor Jose Putur and myself, we are collaborating with the priming, particularly halopriming. And then Professor Rajkumar is preparing bioformulation with different bacteria. And then he is giving to me so that we can give it to the plants so that these plants can tolerate extreme salinity tolerance. Now my past and present students also have tremendously helped me. The funding is from SCRB, CRG we have. And then inside EFG, I visited Professor Dorothea Bartels in 2017. And then we got the seeds, and then we did some experiments with dehydrins there. And this is my current lab, and this was my lab during my visit. I'll be happy if I can answer your questions, if you have any questions. Thank you.
Hello, Denkan sir. Your topic was so extensive, but it was very interesting. In the sense also, you have identified certain issues which is mimicable and simulatable in terms of the stress withstanding ability to be adapted to certain plans of our choice. And I am in connection with, I just remember that Professor Jose Putur sir's lab is also doing wonders in the seed technological aspects, especially in identification of poikilohydric orthodox seeds in terms of quantifying these Lea proteins, probably in a limit of 2000 kilodaltons in the initial stages, so that the conservation strategies may also be standardized in future for the conservation of the plants of our choice, especially endangered species. So this is a very interesting topic, and now it is open for the discussion. So please.
Very nice topic actually, not being studied in India.
Thank you, sir.
The question which I had in mind, but you answered that one of your students is now taking up.
Yes.
Because you mentioned a lot of things about Lea and dehydrin and all that thing. But major tolerance about desiccation is from the complex sugar, like trehalase and all that thing. So I was just wondering whether you are going to mention anything about it, and you did mention. But would you like to explain a little more about that?
So actually what it is doing is, this generally not all resurrection plants accumulate trehalose under stress conditions. So what happens is some unusual carbohydrates are generally accumulated under stress conditions. So generally plants, when the water is lost during dehydration, automatically sucrose is the major molecule that is being accumulated. Some resurrection plants, what they do, they accumulate unusual carbohydrates. So generally what they have done, they accumulate it and they store it as a carbon source. Automatically when rehydration happens, then automatically what happens, these sugars are metabolized and energy is being used for synthesis of new proteins. So this is the mechanism that is generally going on. So trehalose for many years, we have not understood, we never understood the importance of trehalose in plants. So in mosquitoes, this trehalose acts as an energy source for ATP generation during flight. But in plants, that's, the mechanism is not at all understood. So it is accumulated during stress. Now recently what happens, yeast work has been, the work with yeast has been started, where they started working on yeast trehalose metabolism, and then they identified that when the trehalose is accumulated, yeast is becoming desiccation tolerant. Then started again going back to the plants. Now when we looked at the plants, not all plants are accumulating trehalose, but trehalose metabolic genes are present. Trehalose is present in very traceable amounts, very minimal amounts. That means this trehalose that is accumulated at a minimal amounts is important for conferring the stress tolerance. But what we are trying to do in the lab is, we are trying to increase the trehalose content in the cell. What we are doing is, trehalose is metabolized by trehalase. So we try to stop the trehalase activity in the cell by using a validamycin. So when trehalase is completely inhibited, automatically trehalase gets accumulated. Because trehalose generally it's a non-reducing sugar, it is metabolized by trehalase and glucose molecules are formed. Once the glucose is formed, automatically it will be utilized for energy. Then automatically trehalase will never be accumulated. We are trying to stop under control conditions trehalase activity. Then trehalose is actively accumulated inside a cell. When trihalose is actively accumulated in the cell, the plant, what it is thinking is, it is thinking that is under stress conditions. Generally under stress conditions, these carbohydrates are accumulated. Then what it is doing, it is signaling, we are not sure whether it is signaling ABA, it is through ABA or not, but what we have identified is, this trihalose once it is accumulated, then the stress responsive pathways are activated. So dehydrins are induced. The dehydrins which you are seeing under complete extreme stress conditions are induced under normal conditions when the trihalose is accumulated. But when you have trihalase automatically, when trihalase expressing plants are there, then automatically what happens, dehydrins are never observed. So when trihalase overexpressing plants are there, the dehydrins are never observed. So now what it does, there are many reports, but evidences are not very clear. What they say is, it binds to the membranes and protects the membranes from damage. Some studies say that, okay, it is holding the water, and then some water is always there in the cell, so that what happens is, the cell is never devoid of any water. So vitrification, they say. So now these kind of studies are all there, and then there is also a report where they say it is protecting some enzymes from damage, oxidative stress induced damage. So these are all reports. I think recently one author has shown that citrate synthesis activity is being protected by these trihalose. So these kind of studies are going on, sir. Yes, sir.
Dinekar, it's a very good work. So I have, clarification,
Yes,
but Lia gene, right? The
Lia genes.
Lia gene and
Yes.
trihalose, is there any interaction
no.
or, let's take if you knock out Lia gene, whether trihalose still can be accumulated?
No, actually it's, I think it's only one way. So when trihalose is accumulated, Lia genes are activated.
Yeah,
Lia genes overexpressing plants never accumulate trihalose.
okay.
So when trihalose is pumped into the cell, the dehydrins. So trihalose is not accumulated in plants at all, sir. What happens is, only under stress
No,
conditions
that's
this
what
is
I know.
accumulated.
But in stress condition, Lia also is accumulated.
Lia also is accumulated. So these dehydrins are group 2 Lia genes. There are 7 categories of genes in Lia. So group T are selectively dehydrins. So these are accumulated when trihalose is inside the cell.
Oh.
So now trihalose acts as a signal, but whether it is through ABA, because till now we know that it is through ABA. But here trihalose is directly activating dehydrins. Whether it is through ABA or not, that we are not sure.
That is some.
Some connections are missing there. That we need to identify.
Dr. Dinekar,
Yes, sir.
I'm here.
Yes, sir.
Yeah, I'm Dr.
Yes,
Abba. It's very interesting. I
Yes,
think
sir.
while you were presenting, I got a very general question.
Yes, sir.
It has nothing to do with your presentation. I'm working on coconut.
Yes, sir.
In coconut, especially breeders now, they select, see, you said that desiccation, drought and everything. Wherever drought is there, breeders, they select whatever that plant which is surviving there.
Yes.
From that, they will select the seeds, collect the seeds, then they develop the seedlings. They say that it is drought tolerance. Now, how your study could explain the whole mechanism in coconut, whether anything can be extrapolated? Because I was wondering for the answer for this question. If you can throw some light, I think it would be very, very interesting.
That will be good. But actually, what our emphasis is here, we wanted to identify the mechanisms that are important for tolerance. So whether the tolerance strategies are universal or not, we don't know. So what we have done, we have taken some model plants, and then we have checked in the model plants, Tetrastigma, Plantagidium or Orapecium, Linden and we have checked this. In Arabidopsis also we have checked. Arabidopsis there is no activation, whereas in desiccation tolerant plants there is activation. Now the next stage is, we need to extrapolate these to all the plants, whether this will work in all the plants or not. Whether dehydrins are there, in all the plants dehydrins are there, but selective activation happens to them or not, that is the question that is seen. So in these plants, when you give them a stress, when you challenge them with a stress, automatically activation is happening. But in all other plants, whether this happens or not, that we don't know. Dehydrins maybe can be used as markers for stress tolerance. Yes, sir.
May I suggest that all the discussions may be continued during the tea session
Okay,
also,
sir.
because it's a very interesting topic.
Thank you very much. Thank you. Thank you all for your questions. Thank you.
Thank you, Dr. Dinekar, for an engaging talk. I invite Dr. Parvati M. Srikumar, co-chair of the session, to present a memento to Dr. Dinekar Chalabatula as a token of appreciation. Ma'am, please. Thank you, ma'am. Thank you, sir. I invite Dr. Sunoj Kumar P., Associate Professor, Department of Botany, University of Calicut, to present the memento to Chair Dr. Anil Kumar as a token of appreciation. Sir, please. Dr. Parvati M. Srikumar, co-chair of the session. Dr. Danya Thomas, session rapporteur. Thank you, sir. Thank you, ma'am. There is an announcement. Dr. Thadikonda Sujada is requested to present the short lecture PPT at the mic desk. Thank you. Moving on to the next technical session, I invite Dr. C.C. Harilal, Professor, Department of Botany and Head of Department of Environmental Sciences, University of Calicut, to conduct the technical session 4. Welcome, sir. There is another announcement. We'll be breaking for tea after the first lecture of the session. Thank you.
Hello. Hello. Distinguished delegates, researchers and dear students. We are on the second day of the International Conference on Physiological and Molecular Mechanisms for Abiotic Stress Tolerance in Plants, hosted by the Department of Botany, University of Calicut. At this outset, I would like to congratulate my colleague, Dr. Josti Puttur, for taking up the courage to initiate this program, spanning for a week's time in the Department of Botany, University of Calicut. And we are successfully on to the fourth section. And to chair this session, we have Dr. Hebar KB, an eminent physiologist, biochemist, and post-harvest technologist. And presently, Dr. Hebar KB is the head of the division of plant physiology, biochemistry, and harvest and post-harvest technology of Central Plantation Crops Research Institute, Kasaragod. I will not be getting on to his full credentials that you can have from various online platforms. And he is a reviewer of manuscripts of the following journals: Indian Journal of Plant Physiology, Journal of Plant Biology, Indian Journal of Agricultural Sciences, GeoBios, Current Science, Experimental and Economic Botany. He is selected as Global Research Alliance and Borlaug Fellow for the year 2010 under the head Climate Change.
I welcome you, sir, to this August gathering. We have with us the co-chair for today's technical session, Dr. Krishnakumar Ji. He is assistant professor from the Department of Plant Physiology, Kerala Agricultural University, a College of Agriculture, Vallanikara. He has got advanced skills in root phenotyping, bioinformatics, gene cloning, gene editing, and generation of transgenes. He is NISA Young Scientist of the, he has bagged NISA Young Scientist of the Year 2020, which is awarded by the National Environmental Science Academy, New Delhi. I welcome you, sir, to this gathering. We have with us the rapporteur of the session, Dr. Sruthi P. She is a former student of the Department of Botany, and presently she is an ad hoc faculty with the Pioneer College, Kannur. And I welcome you to this gathering. So I wish all the best for this technical session, and I request Dr. Hebar KB to chair this session in a less stressful way. Thank you, sir. Yeah. It's up to you, sir. You can do it from here or.
Professor Jose Puttur for having organized this conference here. I think the climate is very good right over here, and today we have very eminent speakers, I would say, on photosynthesis. The first speaker is for today is Professor A.S. Raghavendra. In fact, he doesn't require any introduction. I still remember when I was the student, when I was doing my MSc program at US Bangalore with Professor Uday Kumar, I always used to look forward for listening to his talks. If there is one authority on photosynthesis in India, I think it is Professor A.S. Raghavendra. I think he has seen, it is like a factory. I think he used to explain in such a wonderful way. I think anybody who is not knowing on photosynthesis also would understand so easily. So I think we are very fortunate to have him here. He has come all the way from Hyderabad to here because, see, when it comes to Kerala, mostly we will be working on plantation crops. They are very difficult to work. Understanding the plant itself is difficult. Photosynthesis is furthermore difficult. It is a great opportunity for us. I have been given a brief CV of Professor Raghavendra. I'll just read it out for those who are youngsters here. Professor Raghavendra is currently working as the Institution of Eminence Research Chair Professor in Life Science, University of Hyderabad. The thrust of Professor Raghavendra's research has been on photosynthetic carbon metabolism and its interaction with mitochondrial respiration and nitrogen metabolism. His group has also contributed significantly to the topic of bioenergetics and signaling components of guard cells in relation to stomatal function. In fact, in today's relevance of climate change, what we have been talking about, I think this is very, very significant. Many of the papers from the group on the topic of photosynthetic carbon metabolism and signal transduction in stomatal guard cells are highly cited. He is an elected fellow of the Indian National Science Academy, National Academy of Sciences, Indian Academy of Sciences, National Academy of Agriculture Sciences, AP Academy of Science, Third World Academy of Sciences, Italy. He was awarded and recognized with INSA Young Scientist Medal, AP Academy of Sciences Young Scientist Award, UGC Career Award, AP Scientist of the Year, INSA Professor T.S. Sadashivan Award, DST J.C. Bose National Fellow, Humboldt Fellowship, Germany. Then JSPS, Japanese Society for Promotion of Science, senior visitor, Japan, and he was also a corresponding member of the American Society of Plant Biology. A total of 9 books, special issues of journals, and 226 papers in peer-reviewed scientific journals and chapters in books have been published. In fact, this is a very brief one. I had seen his CV in the net also. More than I think this CV, he has guided so many students. In fact, they are doing wonders. That itself is a great achievement of Professor A.S. Raghavendra. I think everybody is keenly awaiting to listen to him. Now I request Professor A.S. Raghavendra to deliver his lecture. Thank you, sir.
Yeah, let me try.
This is a pointer, and actually the
Point.
pointer is not too, screen is too bright, that's why the pointer is not clear to that.
It's not clear. Yeah.
Digital TV, that's why. Then
Let's
this is
move
the
on,
next
yeah.
slide moving.
Yeah, I will try if they can't hear. Before I move to near the screen, I think I should thank Dr. Hebbel for a flattering introduction. We try to do as much as we can, but whatever we do is all because of training from big people like Professor Ramdas and Dr. Sinha, who are now no more, but they were the people behind plant physiology progress in India. First of all, let me thank Dr. Jose Puttur for bringing me over here, and also Dr. Rajgopal and Ashwini Parekh. They were the people, they informed me and said we should meet. Some of you may be knowing, I have a special attachment to Kerala. My first job was in ICR, Central Plantation Crops Research Institute, but at a regional station in Vitla. Then I went back to university. Then I came to Kottayam as a deputy director. So I have quite a few friends and quite a lot of nice experiences, first as a bachelor and as with family. So I always remember, I think my wife also jumps whenever we say we come to Kerala. Anyway, thank you so much once again. And if we go to today's topic, well, photorespiration is a very common term, as all of you know. And then the title itself indicates that it's a respiratory process where you have CO2 being evolved, CO2 being consumed, and oxygen being consumed and CO2 being evolved, but happens only in light. Okay. For a long time, there was a lot of discussion. Everybody said, why we need photorespiration? Because it's, it actually takes away a considerable amount of carbon at the level of Rubisco. Rubisco has two kinds of activities. It fixes carbon and functions as a carboxylase, but it also functions as an oxygenase. So we have the problem with the oxygenase actually. I will not say problem, I will try to emphasize and try to convince you that it is not evil or disadvantage or drain of CO2. It is an unavoidable consequence, no doubt, because Rubisco is made to do that, but it has evolved to be an important part of abiotic stress responses. Yeah, that will be the summary of what I am going to convey. It is essential actually under our oxygen atmosphere. We have actually 21% oxygen and only less than 1% or 0.04% of CO2. So extremely oriented towards oxygen-rich atmosphere. And so when we have this, it has also a lot of other problems. So photorespiration is designed to
optimize photosynthesis and also to protect against the oxidative stress, which leads to photoinhibition. Well, this is my favorite kind of statement. Photorespiration is not an avoidable evil, but actually it's quite fascinating both in terms of cell biology, biochemistry, physiology, and so on, and serves an important purpose of removing one of the toxic products of Rubisco. I will show you in a moment, and tries to give plant a lot of benefits back. Actually, this is too much of information in one slide, but let me just go through quickly. As I said, it is a fascinating example of organelle interaction. You know, plants are known to have an efficient crosstalk. When I say crosstalk, it is not real, just signaling. It's a lot of metabolism, then try to protect each other like a community. You know, if you think plant cell as a community, all these organelles, they work together. And if there is any problem to any of them, the other organelle comes to the rescue. So they try to work together and also try to protect each other. We work a lot with P and Arabidopsis, and then you can see respiration has two components. One is the dark respiration because of mitochondria, and then photorespiration. And of course, both of them they can protect, but photorespiration is a major protective mechanism against photoinhibition. And it responds to biotic and abiotic stresses. And the reason why we are talking is that photorespiration is not alone in doing this. As I said, all the organelles, they come together and they try to protect. It is also complemented by chloroplastic system, which also tries to keep the energy acquisition, and too much of energy is dissipated by chloroplastic cyclic electron transport, and then mitochondrial alternative pathway. I think Dinakar is working, but he has focused on another component of his work. All of them, they work together to protect photosynthesis. Why we talk of protecting photosynthesis is because chloroplasts need light to be active and to do photosynthesis. But the moment you have light and the moment you have chlorophyll excited, this oxygen becomes very, very reactive to these excited electrons. So you start with the superoxide, and then superoxide is converted into hydrogen peroxide. And these two are bad. You know, they combinedly are called as ROS, reactive oxygen radicals. And all of these, like photorespiration, cyclic electron transport, and the mitochondrial alternative oxidase, they all come into picture. Why Rubisco is a problem is shown here. When you start with the carboxylation, it forms a very active intermediate, and this active intermediate, once it is formed, it cannot stop reacting with either CO2 or oxygen. And remember, we have a lot of oxygen rather than CO2. Well, in fact, the value you see here is old. Now it should be about 0.041% of CO2, but still way below oxygen. So once it is reacting with CO2, there is no problem. We have two molecules of PGA, but if we have oxygen taken into the reaction, it forms PGA, okay, one molecule of PGA, and the other molecule is a problem. This phosphoglycolate is quite toxic. It has to be removed quickly and efficiently. And so this one photorespiratory pathway is beautifully designed to do this job. As you can see, phosphoglycolate is taken out of chloroplast. I think I have a next slide showing some reactions, and it is converted slowly back into phosphoglycerate. That means the toxic product is removed and carbon is recovered back. And so this recovery of carbon can be as much as 50 to 75%, which is losing after the oxygen level. Yeah, when it is recovering, there is also another purpose being served. If you see now, we have what we call as the Calvin cycle, consuming NADPH and ATP, and this consumption is quite important because the energy is being used and re-assimilated, and but if you have only phosphoglycolate, you are losing energy. So phosphoglycolate now is metabolized into photorespiration, again gaining the same thing, NADPH and ATP are used, releasing CO2, that's what I said. So photorespiratory CO2 becomes what you call as some kind of substrate for also carbon fixation. And another thing is ammonia. Ammonia is re-assimilated. So we have re-fixation of CO2, we have re-assimilation of ammonia, and both are quite important. Yeah. Okay. We use mostly leaves and protoplasts for this system. If any of the students are interested, we use like Arabidopsis because it's a model plant, and P, and both of them can be used to also isolate a lot of mesophyll protoplasts. Yeah, this is what I was mentioning. If you see carefully, actually this is a classroom kind of slide. Chloroplast is the place where you have this rubisco-oxygenase. So when we have oxygenase activity, phosphoglycolate is formed. This is quite toxic. So chloroplast sends it into the peroxisome and in the form of glycolate. So we have phosphoglycolate phosphatase. And glycolate is now oxidized to glyoxylate. And during this process, glycolate oxidase produces a lot of H2O2. And this has its own function. Glyoxylate converted into glycine goes into mitochondria. Glycine is decarboxylated with the 2 steps to serine. And this is the place where you have a lot of CO2 being generated and a lot of NADH being produced, and also ammonia being liberated. So this ammonia becomes important for chloroplast. And what is interesting is that now you have considerable carbon coming back in the form of serine. Serine, hydroxypyruvate, and then glycerate. Glycerate enters chloroplast, converted into phosphoglycerate, which means that whatever carbon you lost at the oxygenase level, now it is coming back to chloroplast again in the form of glycerate and converted into phosphoglycerate. So not much is lost. Carbon is now recovered, what you call as conserved. At the same time, there is also some production of a lot of energy in the form of glycine oxidation. NADH can be the source for several things in the cell. Ammonia is conserved and re-assimilated and becomes important for nitrogen metabolism in chloroplast. So, photorespiration is a classic example of mitochondria, chloroplast, peroxisome, cytoplasm interaction. And in fact, this is backed up by the cell cytology. This is what is shown already. What you have here on the left is a cross-section of a cell showing up chloroplast, and then at the top corner you have mitochondria represented by M, and chloroplast, mitochondria, and peroxisome is in between. Physically they are arranged. It's a very consistent feature in many, many leaves. And it's not only biochemistry what you see, even physically they are close. So this on right we have another species, same situation. You have chloroplast, mitochondria, and peroxisome, which is having that crystal rhomboid shaped matrix they call. And so they are physically in touch with each other and biochemically in touch with each other. So it's a kind of super example of physical proximity. I don't need to really read all of this. I think I will just go through the next set of slides. Yeah, this is an important point. When chloroplasts are active, they cannot stop from functioning as a photochemical system. So photochemical system produces a lot of ATP and NADPH, and both of them combinedly are called as assimilatory power. But this assimilatory power of ATP and NADPH they have to be recycled. Unless you have ADP, unless you have any ADP, the system cannot go on. So this recycling is very important. In addition to that, if you don't do that, the chloroplast gets over-energized, and that's where you get the problem of photoinhibition. What normally happens during carbon fixation, that is ATP and NADPH are consumed during carbon assimilation, and then the balance between production of ATP and the regeneration of ADP, it is maintained. So recycling and supply of ADP and NADP are essential. But a point to be emphasized at this is that this recycling is not only because of assimilation of carbon, it's also because of assimilation of nitrogen, assimilation of sulfur. So chloroplast does a lot of things. And interestingly, peroxisomes are also quite important in providing the nitrogen in the form of ammonia. Yeah, when this balance is affected, then there is a lot of problem. On the left, you see a normal system. You have a light which is optimal, and when it is optimal, the leaf starts producing ATP and NADPH, and it is consumed in the carbon assimilation. So chloroplast is quite happy, and we have a normal and efficient photosynthesis. But then you can have stresses. For example, you can assume there is too much light or high light. When you have too much light, the chloroplast is so much energized that it will not be able to come back to the ground level. So we have excess energy, and this is excess energy causes problem. But chloroplasts do have protective mechanisms, but still sometimes it is not enough. So what happens is, if chloroplasts are over-energized, then later photosystem 2 starts getting damaged, probably followed by photosystem 1, and then you have this phenomenon of inhibition of photosynthesis by light itself. That's why it's termed as photoinhibition. And this can happen also under other kind of stresses. For example, we are talking of balance between photochemical system and biochemical system, where you have a good recycling of ADP and NADP. Suppose you have, say, like low temperature. Enzymatic reactions are slow if the temperature goes down or temperature goes, say, like supraoptimal. So when the temperature and things like that, then the carbon metabolism is affected, and then also you get photoinhibition. That's why photoinhibition occurs quite commonly in temperate countries because of this low temperature rather. As I said, chloroplasts are having multiple systems to protect themselves. For example, if we have supraoptimal light, thylakoids are getting overexcited, and this energy is dissipated through several means by cyclic electron transport, Xanthophyll cycle, as heat, thermal dissipation, and trying to protect themselves by turning over a lot of D1 protein and so on. But a lot of this dissipation of excess energy happens through oxidative electron transport and photorespiratory metabolism. So I put these two as the crucial ones, much more than others, and that's what we know. Okay, now one small thing which establishes the importance of photorespiration, when this pathway was discovered, naturally people try to locate mutants which are deficient, and if you see on the top, wild type plants, they can grow happily, but if you have a mutant and you try to grow at normal levels of oxygen and CO2, they don't grow. And in fact, many of them, they die over a period of few days. They cannot survive, which means that if you don't have photorespiration, plants will not survive in our normal atmosphere. So in the lab, of course, what they do is, you try to grow them at high CO2. And you can see on the top. So instead of having, say, like 0.035, they used, in some cases, they have used even 1% CO2. So you need high CO2 for these mutants to survive. And in the bottom, you have the enzymes which are listed. I think you have seen these different organelles where we have any enzyme which is involved in photorespiration If it is deficient, all these plants, they don't survive. Everything, every enzyme is important. And that's what is summarized here. This recycling of glycolate by photorespiration becomes essential for even survival at our normal atmospheric conditions. So mutant is a good evidence that photorespiration is essential. Yeah, this is what I already said. A lot of work has been done in different mutants, and just for comparison, the scheme is shown on the top. So we have the experiment on 3 types of mutants down below. So we have wild type represented by WT, and then we have this glycerate kinase followed by pyridoxine GOGAT, pyridoxine glutamate synthase, serine hydroxymethyl transferase, and dicarboxylate transporter, which actually exchanges malate and glutamate across the chloroplast. So whenever they have this what you call as deficiencies, if you look at CO2 fixation, all these mutants, they try to photosynthesize, but they cannot survive continuously. They lose the capacity, it goes down. So as you can see, within about 40 minutes or so, they all come down in their photosynthetic rate. At the same time, on the right, it's a kind of complementary measurement. The chlorophyll gets into non-photochemical quenching. If you have photochemical quenching, that means the energy is being used. If we have non-photochemical quenching, it means that energy is being lost without getting into carbon assimilation. And you have a reverse. If photosynthesis is bad, non-photochemical quenching goes up. So the wild type is down below, and then the mutants, all of them, they are much above the wild type. So the whole photochemistry and biochemistry balance is disturbed. And that means photorespiration is helping in dissipation of excess redox. And that it does, both directly and indirectly. Same thing, to emphasize once again, like when you have photochemical activity, the photosynthetic electron transfer produces NADPH and ATP. And so this reductant can be also in the form of reduced pyridoxine. So this is something which goes at a very high rate the moment you have light. But the regeneration of these substrates to keep this photosynthetic electron transfer going on, you need systems. So Calvin cycle is not sufficient to have this recovery. It has to be what you call as complemented or supported by also the photorespiratory cycle, which is shown as C2. So we have a carboxylase activity favoring Calvin cycle, and we have oxygenase activity which keeps photorespiration running, and only when both of them then come together, the plant is able to perform well. Yeah, this is an indication why it happens. Now you can see the theoretical requirement of ATP and NADPH for photosynthesis. When photorespiration is going on and ammonia recycling is going on, the chloroplasts, they can consume more ATP and more NADPH, and as a result you have ADP coming back and NADP coming back. So photorespiration is quite essential to have this optimal recycling of ADP and NADP. Yeah, this is a summary of whatever I have shown. Like you have photosynthetic electron transport, which will go on at a very high rate the moment you have light. It's like a switch. And this ATP and pyridoxine or NADPH reduction can go on only when they come back. So the recycling of ADP or pyridoxine or NAD or NADP is helped in several ways. In addition to that, photorespiration, I have already mentioned that it generates CO2, and it has been found that in all these conditions, they all help. For example, photorespiration is it gives CO2, and it's believed that it is quite significant for carbon assimilation in chloroplast, and it's almost like a CO2 concentrating mechanism. It gives out glycine, and when glycine is oxidized, it produces NADH and subsequently ATP in mitochondria. It also releases ammonia, and this ammonia is reassimilated and sent into chloroplast. Over and above all this, glycolate which is formed, which is toxic, it is detoxified. And then hydroxypyruvate reduction and glycerate formation for chloroplast. All of them, they try to keep the H2O2 levels at a normal level. This H2O2 is needed as a signal. Only when it becomes excess, it creates a lot of problem. And then keeping this ROS levels low is in fact done by not only photorespiration, but also the chloroplastic cyclic electron transport and mitochondrial alternative oxidase. That's why I said photorespiration is complemented by also this mitochondrial alternative oxidase and chloroplastic cyclic electron transport system. Yeah, the proposal is that photorespiration actually it tries to keep this tempo. It's not directly what you call as protecting, but it tries to keep the cell poised for adjustment. So when we have a normal system, this poising and adjustment against excess light is done by several things, cyclic electron flow, and then there is also a myelite valve in the chloroplast. So on the top what you see is a green kind of box is chloroplast, and here at the bottom you have what we call as the other parts of cell. So this overexcitation is brought down to a limit, ROS accumulation, ROS is scavenged, and this acclimation keeps ROS levels at a signaling level. And this ROS at a signaling level will help plants to acclimatize and protect what you call as against different kinds of stresses. Any mild disturbance in the regulation of photorespiratory activities will create a lot of problem. I think. Yeah, I will just tell you only one case. See, for example, here we have a mutant of Arabidopsis deficient in alternative oxidase. So if you see the mutant of Arabidopsis, when it is exposed to cyclic electron transport inhibitor like antimycin A, so when you try to play and block the electron transport, the AOX becomes important. And during this process, photorespiration seems to be also parallelly increasing. So we have an increase in the glycine to serine ratio, and we have an increase in the expression of glycine decarboxylase, all of them indicating that under this AOX deficiency or promoter, we have also the parallel kind of stimulation of glycine decarboxylase has 4 proteins, so P protein is one of them which responds quite well, and that's what is represented here. Maybe I just read it. Glycine to serine ratio increases to indicate an increase in photorespiration, and this is reflected also in the level of GDC, and aminoacetonitrile, an inhibitor of GDC, decreases photosynthesis in wild type, but not in alternative deficient mutant. Yeah, as I said, it dissipates excess redox by what you call as direct and indirect effects. It's a direct utilization of ATP and NADPH, utilization of reduced peridoxine, and of course keeps H2O2 levels at an optimal level. Then indirectly, photorespiration promotes photosynthetic cyclic electron transport, promotes alternative oxidase, and it also provides CO2, which is coming from glycine decarboxylation. So all of them, they become important. Yeah, this is an example. Suppose you try to decrease one of these key enzymes of photorespiration. So this is a potato mutant, which has reduced P protein. That means it has reduced activity of glycine decarboxylase. And then what happens? In these, the plants, they try to compensate by overexpressing alternative oxidase, and that is what is shown up. So there seems to be a kind of balance between mitochondrial alternative oxidase and the photorespiratory glycine decarboxylation. Well, the explanation is very simple. The plant tries to keep ROS levels at a minimal level by modulating either chloroplastic electron transport or oxidative electron transport, because these bioenergetic membranes are major sources for production of ROS, and that's what you see here. If you have this electron transport going on, you have a tendency to produce superoxide radicals, H2O2 and so on. This is because of the chlorophyll here, and if you use a cyclic electron transport system around photosystem 1, this reaction will be reduced. So you can see, if you don't allow this overexcitation and keep electron transport in a cyclic way. And similarly, in the mitochondrial electron transport system on the top, reactive oxygen species is produced a lot under normal conditions like cytochrome oxidase. But if you happen to have alternative oxidase tries to minimize the ROS levels. So we have two different systems in the cell which tries to keep the reaction oxygen species at a lower level. And that's what happens. All of them, they come together. So you can see that photorespiration gets what you call as stimulated under stress conditions, tries to help plants to adapt to abiotic oxidative stress. And this is mainly by keeping the cellular redox, that's particularly ROS, at optimal levels. And during this process, photorespiration interacts strongly with the chloroplastic cyclic electron transport system and mitochondrial oxidative, alternative oxidase. Yeah, these are quite a few references, and I will not go into the details, but the point is, if you modulate photosynthetic cyclic electron transport system, photorespiration also is modulated. Modulate alternative pathway, you have already seen, photorespiration tries to, what you call as increase and try to keep the balance. And if you have mutants which are deficient in both alternative oxidase and CEF, it normally, is also affected in photorespiration. So there is a very strong crosstalk between these three, and that's what is represented in the next slide. The factors on the top, they're all under the stress conditions. So drought, highlight, or low temperature, or low CO2. Under these conditions, you have high ROS, high ATP requirement, and high ratio of NADPH to NADP or NADH to NADP. Under these conditions, invariably, photorespiration is stimulated, and along with that, alternative oxidase is also stimulated, and cyclic electron transport also stimulated. And all of them, they try to protect the plant by keeping the redox state at an optimal level, protect against photoinhibition, and also provide additional ATP required for the cell. And what is not clear is whether there is, photorespiration can modulate alternative oxidase and cyclic electron flow. It's not clear if alternative oxidase can regulate photorespiration back, and similarly, cyclic electron flow can it modulate. So that kind of work is still going on. So then another point is, after all, why this system has evolved and started showing up photorespiration. It seems to have evolved as a result of the atmosphere. At one time, as all of you know, there was no oxygen at all. Then oxygen started increasing. Then later, CO2 started increasing with industrial production and so on. And then photosynthesis started building up. And but then in the recent years, of course, we have more of CO2, but oxygen is still quite high. And I think this next slide will show you. this is what happened with the levels of CO2 and the levels of oxygen in the atmosphere. So what we have now, CO2, is very, very low. You can see oxygen is in percentage and CO2 is in points, less than something like 0.1 now. At the beginning, the CO2 started coming down. So number one is before land plants, concentration of CO2 was high, and as the land plants appeared, as you can see in the phase of 2, the CO2 concentration started coming down because photosynthesis is going on. Then at the same time, since photosynthesis is going on, oxygen has started going up. Then suddenly there was a disturbance in the CO2. People believe that asteroids hit to the Earth and there was volcanic activity and so on. And when this has happened, then the CO2 started going up. And there was a disturbance in oxygen also. Then oxygen came down, but it went back. Oxygen kept at very high level. So at 4, what you have is very high levels of oxygen, 21%. And currently we have also very low levels of CO2 because after that burst, which happened in phase 3, we have now. And this imbalance between oxygen and CO2 seems to have made the photorespiration to be active. Yeah, and this can be something like an example of taking plants. Photosynthesis has evolved with cyanobacteria and algal system. They were all in water, and at that time photorespiration was, it is not that it is not there, it is low because Rubisco is always having that glycolate. And in fact, some of the algal cells, they secrete glycolate into the medium. On the other hand, once this land habit has started evolving, now we can see oxygen concentration has started increasing, and because of this very high increase in oxygen and is maintained at very high levels, so we have this photorespiration also started increasing to protect the plants. So the take home message is photorespiration is actually a fascinating phenomenon and protects against photo inhibition and helps against stress, but it interacts very strongly with chloroplastic cyclic electron transport system and mitochondrial alternative oxidase to optimize the ROS levels, and seems to have evolved as a result of an increase in the atmospheric oxygen much more than that of CO2 during the course of evolution. But I don't want to leave you all with a notion that photorespiration is everything for plant photosynthesis. People tried to handle photorespiration and tried to reduce. No, this is 2 of our papers. Attempts to improve photosynthesis have been made without going for manipulating photorespiration, because the first effort is, if you try to decrease photorespiration, can productivity increase? First point is whether you can play with Rubisco itself. Rubisco is such that it is evolving or reacting very highly with CO2. Suppose you can identify a form of Rubisco which reacts with more with CO2, not much with oxygen. It didn't happen. For example, here you have the relative specificity from different type of plants. So you have higher plants, cyanobacteria, and then some of this other bacteria, red algae, which are in sea. Now you can see the affinity for CO2 over oxygen. It's almost like 100%. And so the 100% will indicate that affinity to CO2 and oxygen are the same. And if you have a higher figure like 230, 238, which is in special red algae, it is capable of reacting more with CO2 than oxygen. That means CO2 favoring reaction of Rubisco. But then this is present only algae. So people tried to take it out and express in higher plants, but it didn't work. Rubisco is a very, very complicated enzyme. It has 2 types of subunits and it has to be assembled and so on. The point is, scientists could not locate a good source which can be used for plants. Then the other approach is whether you can make a short circuit. We have already seen in the normal type of figure at the beginning, so that Rubisco oxygenase activity produces glycolate, and it's metabolized through peroxisomal reactions. And what you have here is, glycine serine decarboxylation, and serine is converted back to glycerate. So all these black reactions are present in the normal system. Suppose you have a short circuit, don't allow glycolate to be sent to peroxisome. Can we recycle within the chloroplast itself? Yes, it is possible. They identified certain enzymes. So glycolate is converted to glyxylate and then to tartanic semialdehyde and CO2. And tartanic semialdehyde can be converted to glycerate. So we have glycolate, CO2 coming back, and glycerate coming back. So we have some kind of recycling of carbon. But then unfortunately it works well in theory, it works well in Arabidopsis. But the moment you take into the what you call as field, it doesn't work. So what you need is three different enzymes, which is something like synthetic biology. They are taken from bacteria and introduced into the chloroplast. The reason why this doesn't work is because you are not considering ammonia reassignment. So you only carbon, but what about ammonia and what about the energy status? It doesn't work. So that's what I said. You have carbon being considered, but we are not reconsidering the ammonia recycling, which happens in case of photorespiration. Then a couple of examples. You can try to overexpress some of the rate limiting steps like pseudoheptulose 1,7 bisphosphatase. This is actually rate limiting and is feedback inhibited, but you can take a bacterial enzyme which is not, and they try to do it. So that's mostly in what you call as tobacco and wheat. It works for a while, again in the field, in the lab. But if it goes to field, then it doesn't really work. This is something which happens with plants, genetic manipulation and so on. Then people now think that there are a lot of ways to improve, optimizing Rubisco itself, optimizing Calvin benzyl cycle, then introduce a carbon concentrating mechanism. Like you can take carbonic anhydrase from another plant, and then you can try to introduce carbon transporters into the chloroplast. Then there is also the pyrenoid-based carbon concentrating mechanism, cyanobacterial type CO2 pumps. Ways are present, but putting them all together is something like, it will be a marvel. So the idea is to have an increased growth and increased yield from several of these crops. This is, I will say right now, theory at this point. We need a lot of work to implement it. But science progresses. What appears not possible today may happen tomorrow. I think I have completed what I wanted to say. So I should thank all of you for having listened to me. I have been fortunate to have a good set of students. I have 4 of them here. I will show in the next slide. PhDs and postdocs, quite a good collaborators. I'm also lucky to have got money from different sources from India as well as abroad. Yeah, these are the people, our team or my team at this conference. So my wife also is a part of it. So Bharat and Shashi. Bharat is working on something like a signal transduction stomata, Shashi of them. Incidentally, they have a similar field, so they stood together. On the right, we have Deepak and Ishwar. They concentrate on carbon metabolism. I think all of them, they are giving oral presentations, probably today afternoon. So thank you once again for your patience.
Sir, as I said in the beginning itself, it would be a wonderful talk. In fact,
say that my pen drive, which I brought with the slides ready, it gone kaput, something happened.
But I think, but you explained so elegantly, sir.
So I tried to build up, and maybe Some of the slides I have already shown last time when I was here.
Otherwise, we thought that it is a photorespiration means a will, but you said that it is a very fascinating.
Yeah, it is.
And in fact, it is very relevant for us, sir, especially those who are working in these Western Ghats, especially Kerala area, because we grow crops like coconut, arecanut, and spices. In fact, these are all shade-loving plants. Under the present circumstances of climate change, we have been facing a lot of problems, especially as you rightly shown that photo inhibition and everything.
Yeah.
So under that circumstances, because we were not able to do much work on these aspects, otherwise I think this is an eye-opener for especially to the students and other people.
No, actually the message is plants are quite fascinating. They know how to protect themselves. And in fact, when you come to this, shade-loving plants, we only talk of light, high light, low light and all that. But what happens in the undercover, if you have say like coconut or arecanut and cocoa in between, it is not having either low light all the time, either high light all the time. So you have this movement of sun, and when the crown moves, you have this
flecks of light,
intermittent light. They call, sun specs is a bit different. So you have this intermittent variation. And this is even more interesting.
But one small question, sir, because between this C3 and C4, C3 has high photorespiration, whereas C4
No. That's actually misnomer.
Yeah.
See, C4 has a special kind of anatomical feature. So we have a mesophyll and we have a bundle sheath. And this photorespiration, in case of C4, it is confined deep in the cell. So we have photorespiration going on. And the anatomy is made in such a way that if any CO2 tries to come back and gets back into the atmosphere, mesophyll pepcarboxylase is a like absorbent. It just puts back into sulfur acid. At the same time, a lot of CO2 is present on the top. Since this bundle sheath cells are very thick, their oxygen concentration locally is minimized. So if you calculate the amount of CO2 present, in case of mesophyll cells, the chloroplast level concentration of CO2 will be, I think I will put something like 0.07%, very low. If you go to the bundle sheath, because of this limitation of photorespiratory reactions within the bundle sheath cell, the concentration of CO2 is something like 70, at least 10 times more. So what C4 plants they have done is, they have concentrated CO2 in the bundle sheath cells. And of course supplemented with other C4. But basically it is again Calvin cycle at the end. So if you say C4, you have to say C4 plus C3, because CO2 has to come back and refix it in Calvin cycle again. But that microenvironment is made optimal for Calvin cycle.
Okay. Any specific question, or shall we have the discussion while taking tea? Everybody is telling yes. Okay. Thank you, sir.
Thank you again. Thank you again.
Thank you so much, sir, for such an engaging session. Now, I would like to invite the chair of the session, Dr. Heber KB, to present a memento to Professor Raghavendra as an expression of our love and gratitude. Sir, please. Thank you, sir. There is an announcement. The participants for oral presentation who have registered today are requested to submit their presentations at the help desk immediately. Now, let us break for a tea. We will be resuming the rest of the technical session after the break. So all are requested to assemble here by sharp 12 p.m. I repeat, sharp 12 p.m. Thank you.
Botany, University of Calicut was founded in 1968 and has a long and distinguished history created by eminent faculty and researchers. It is one of the leading botany departments in India, offering MSc in Applied Plant Science and MPhil and PhD programs in botany. It has been funded by the Department of Science and Technology, Fund for Improvement of Science and Technology Infrastructure, and Kerala State Council for Science, Technology and Environment, Society for All-Round Development Programs. The university has recognized this department as a center with the potential for excellence during the Golden Jubilee year 2018. Currently, the department encompasses 8 divisions: Angiosperm Taxonomy, Cell and Molecular Biology, Cyanobacterial Diversity, Environmental Science, Fungal Diversity, Genetics and Plant Breeding, Plant Biotechnology, and Plant Physiology and Biochemistry. Many faculty members have been awarded with prestigious fellowships including Nuffield Foundation Fellowship, Smithsonian Fellowship, Alexander von Humboldt Fellowship, Janida Fellowship, and Nehru Centenary Fellowship. Several distinguished teachers have served the department in the past and contributed immensely to its growth and development. So far, more than 230 PhDs were completed from this division. The tissue culture methods developed in this department are now being widely used in the cultivation of banana, vanilla, and a variety spices. 2 genera and more than 200 species of flowering plants, 4 genera and 224 species of fungi are discovered as new to The Department of Botany, University of Calicut was founded in 1968 and has a long and distinguished history created by eminent faculty and researchers. It is one of the leading botany departments in India, offering MSc in Applied Plant Science and MPhil and PhD programs in botany. It has been funded by the Department of Science and Technology, Fund for Improvement of Science and Technology Infrastructure, and Kerala State Council for Science, Technology and Environment, Society for All-Round Development Programs. The university has recognized this department as a center with the potential for excellence during the Golden Jubilee year 2018. Currently, the department encompasses 8 divisions: Angiosperm Taxonomy, Cell and Molecular Biology, Cyanobacterial Diversity, Environmental Science, Fungal Diversity, Genetics and Plant Breeding, Plant Biotechnology, and Plant Physiology and Biochemistry. Many faculty members have been awarded with prestigious fellowships including Nuffield Foundation Fellowship, Smithsonian Fellowship, Alexander von Humboldt Fellowship, Janida Fellowship, and Nehru Centenary Fellowship. Several distinguished teachers have served the department in the past and contributed immensely to its growth and development. So far, more than 230 PhDs were completed from this division. The tissue culture methods developed in this department are now being widely used in the cultivation of banana, vanilla, and a variety spices. 2 genera and more than 200 species of flowering plants, 4 genera and 224 species of fungi are discovered as new to science, and 2 patent applications has been filed from the department. Our rich tradition of classical taxonomic research has brought laurels to this department in the form of coveted awards. Padma Shri, Dr. Janaki Ammal Award, Professor Puri Award, Chair of Taxonomy, Y.D. Tyagi Gold Medal, Professor P. Maheshwari Award, Dr. V.V. Shivarajan Gold Medal, Excellent Scholar Award, and Todas Tusi Gold Medal. Professor Joski Mangali, Professor M. Sabu, and Professor Santosh Thambi have been recognized by the Linnean Society of London by awarding them with its honorary fellowship. The scholars from this division have bagged 9 awards instituted by International Association of Plant Taxonomy. The department is the headquarters of Indian Association for Angiosperm Taxonomy and Governmental Foundation. The department has an internationally recognized herbarium with over 1 lakh specimens of vascular plants from India. The department is proud to have an excellent botanic garden with an area of 33.5 acres with diverse collections of zingiberas, gesneriads, orchids, aquatic plants, ferns, bromeliads, spiderworts, cacti, medicinal and aromatic plants. A touch and feel garden for the visually challenged people is another major attraction. Redia and Eco Chronicle are the 2 journals published from the department. The state government funded Inter-University Center for Plant Biotechnology is also associated with the department. The department has undertaken research projects in all major areas of plant science, for which it has received generous funding from the Department of Science, Science and Engineering Research Board, Ministry of Environment, Forest and Climate Change, Department of Biotechnology, the University Grants Commission, Council of Scientific and Industrial Research, National Medicinal Plants Board, and Kerala State Council for Science, Technology and Environment. The granted facilities from various funding agencies are also used for interdepartmental research. For the last 5 years, the total national and international research funding for the department was around 3 crores and 40 lakhs. More than 60 books and over 2,000 research papers in reputed journals with high impact factors were published from this department. Many faculty members have active collaborations with institutes in and outside India.
Ladies and gentlemen, welcome back to the International Conference on Physiological and Molecular Mechanisms for Abiotic Stress Tolerance in Plants, jointly organized by the Indian Society for Plant Physiology, South Zone, and the Department of Botany, University of Calicut. I would like to hand over the mic to Chair Dr. Heber KB to resume the technical session 4. Sir, please.
Let us presume this post-tree session now. I welcome all the delegates back. Now we have one more special invited lecture by Professor Rajgopal Subramanian. I request my co-chair, Dr. Krishna Kumar, to introduce the speaker.
Thank you, sir. It's a great honor to introduce Professor Rajagopal Subramaniam. He's a professor and head of Department of Plant Sciences, School of Life Sciences, and director and coordinator of Aspire Bionest at University of Hyderabad, India. His research arenas include photosynthesis, bioenergetics, acclimation of chloroplast, structural and organizational dynamics of photosystems and light harvesting complexes, then state transition phenomenon, nutriphysiology including ion and photosynthetic apparatus, and in addition light accumulation, lipid accumulation and the profile under abiotic stresses. He has earned a lot of honors and recognitions, and he's visiting our adjunct professor in various universities at USA, Canada, Australia, Japan, Israel, Russia, Greece and China. To date he has around 2,800 citations with an H index of 34. Welcome you.
Yeah, thank you for a nice introduction. Before starting, I would like to thank Jose Potur for inviting me. And also before starting the talk, I would like to thank or dedicate this work to my late PhD supervisor, Professor Prasanna Mohanty, who did my PhD at JNU. Okay, today's topic is a little different, and Dr. Sylvia is here, from Hungary. It's most of them what she spoke about hydrogen production, but I'm going to talk about a little more, the biomolecule, which is how lipids can be synthesized using these two mutants. That the mechanism is here, the autophagy-induced accumulation of lipids in two mutants, that is the PGR1 and PGR5 from Chlamydomonas. Okay, so as Dr. Sylvia said that the Chlamydomonas is a model organisms for plants as well as in any cell biology it can be used. And here, this is easy to grow. When I joined in University of Hyderabad, I was working in Arabidopsis, which is a very, it's a model system, but it took, at least it takes about 2 to 3 months to get some results. But Chlamydomonas is easy to grow. Within, the multiplication time is only 6 hours, or you can get the log phase within two days. So, and now the sequence is available, and you can do a lot of mutation studies, and with recent technology can be used, CRISPR-based technology. One of my students is here in the audience. Jerome is here, he's working on crystal technology in the same, the field to accumulate more lipids using that one. So, okay. So general photosynthesis, you could see a linear photosynthetic electron transport, where you can see this linear electron transport. Ultimately, you get the chemical energy, that is ATP and NADPH. So, and we have a cyclic electron transport around the photosystem 1, where it is also very important in photoprotection. So how this photoprotection occurs through the two mutants that I'm going to talk about it. So in these situations, the membrane complexes of photosystem 2 and photosystem 1 and cytochrome B6 is very important in highlight stress conditions. So now, if you look at this, the tightly bound, also the carotenoid constituents can deactivate the excited triple chlorophylls. That is the one, it's called, singlet oxygens, can be reduced if you produce more of the carotenoid. Role of carotenoids are very important in photoprotection. That's we are going to look at in a few slides later. In the chlamydomonas, we have it is because aquatic algal system can grow in fresh water, and they have a mechanism is developed differently than the higher plants. The photosystem 1 has a, this is the photosystem 1 complexes, where on higher plants, you have one half moon shaped the quarks reaction centers, and one outside peripherally 4 light arresting complex are there. Whereas in chlamydomonas, so they have evolved. We also we discovered this one way back in about 20 years back. So the outer ring, another 5, this altogether 9 light arresting complex are there, which is very, very important in photoprotection, and also for harvesting the light. The photosystem 1 light arresting plays a major role. So what it does, that's what I'm telling. I think that Dr. Sylvia is here. I'm also going to talk about PGR1 and PGR5 here. The two mutants here, as I said, PGR5 and PGR1. What they do here, you can see here they play a major role in photoprotection. And if you can look at the PGR5 play essential role in transferring the electrons from paradox into the PQ pool, down in the somewhere in that cycle here. So now also another one is a cyclic electron NDH dehydrogen-like complex dependent pathway that mostly prevails in higher plants. However, in the algal system, it is not at point, but now recently there was one paper, the NDH dependent also is playing a major role in cyclic electron transport. So now, so all these during the one side photo protection. The other side also, you can see this, the metabolic pathway, whatever after the photoprotect for the chemical energy which generated, so you get the NADPH and ATP and enter into the Calvin-Benson cycle. So you get, through all the anabolic pathway, where you can get through gluconeogenesis, you can get all the other components. The main element, the process of any biomolecule synthesis is the primary is, the photosynthesis. Without photosynthesis, they cannot produce all fatty acid and carotenoids or amino acid. So we are going to look at all those components, how when in the highlight conditions, whether anything advantages are there, in any synthesis of any biomolecule. That's what we are going to see here. So now, during this mechanism, during this process, the non-photochemical quenching plays a major role. What is non-photochemical quenching? If you look at the thylakoid, architecture or schematic representation, the luminal side of the thylakoids, whereas here the acidification will increase, the delta pH increases. So when the delta pH is increased, so there is a this is called a non-photochemical quenching. It's increase. This is a photoprotective mechanism. Through and non-photochemical quenching and the cyclic electron transport, the proteins which I said here, PGR1, PGR play major role. So what it does here under these conditions, there are plants get adapted through the short-term and long-term responses. As Professor Raghavendra was telling, and also the chairman of the session was talking about the shade plants and the plants which is highlight exposures. In the short-term responses, they go to the fluctuating light kind of thing, or even in the short exposures, where you can see these are the components the ROS generation can activate it. There is a kinase mechanism that is called, which plays a major role in adjusting the energy between photosystem 2 to photosystem 1, that is called state transitions. I'm not going to talk about, we did a lot of work on state transitions, significant work on state transitions, where the LHC2 kinase plays a major role to phosphorylate LHC2 and to migrate those LHC2 to photosystem 1. That way it's getting photoprotecting in the plants. And also try to get a formation of delta pH and other things. And long-term exposures, the plants are getting acclimatized through the non-photochemical quenching. That's what I wanted to say here for the students. Through non-photochemical quenching, the plants can get adapted or acclimatized. However, if the stress is becoming more and more, even non-photochemical quenching also can be, cannot be, it can be again, reactive oxygen species will be too much, and it can be damages the system. So this is the one I'm talking about, the non-photochemical quenching, QE. You can see the components QE, QZ, and QT and QA. They're all non-photochemical quenching related. QA is nothing but photoinhibition. QT is again inter-state transitions. QZ is nothing but zeaxanthin dependent, and QE is non-photochemical quenching components. So what we did, we have taken these two mutants, PGR1 and PGR5, and NPQ mutant also. We grew them in various light conditions. That is 50 microinstance is the control, and 250 and 500 the microinstance are you can say that various light conditions. We grew them as the mutants. It takes longer time. They grow little slower, but after some time, they can, after 4 days or 5 days, they can reach to the normal control conditions. We grew them in acetate medium as well as in the phototropic medium. This photoheterotropic and phototropic both medium, HSM medium, where they can photosynthetically grow. So they also grow very well in all of in two conditions. They grow very well, and we also seen the photosynthetic. I'm not showing any of the PAM measurements, but only since we are focusing more on non-photochemical quenching, we are just showing the non-photochemical quenching is significantly increased in wild type, whereas the non-photochemical, the second figure, the B figure, the non-photochemical quenching is suppressed in PGR5, whereas the non-regulated energy, which is also considered a non-photochemical quenching component, which is increased in the mutant. You can see the black line, and they have significantly increased, and this is in the HSM medium. So through one is in the wild type, this is, I think, my student Ranay Mohan Yadav is going to talk about more in detail about these components. The QE and the is nothing but delta psi and delta pi, where it is both of them are in the delta pi is increased more in the mutants. Thereby it is balancing the photoprotecting to the highlight. So further, what we did, we just recently published this paper in BBA Bioenergetics. So we wanted to characterize what is happening into the cell inside biochemically or a molecular level. What we did, I mean, this here, we just measured the ROS components. The ROS is significantly increased in the mutant, that is the PGR5, and PGR1 also is a little bit there. And whereas, in wild type, we could see only little ROS. And this is because, again, since it is a mutant of PGR1, the excited molecules at the acceptor side of photosystem 1 is overexcited, where you can get the photooxidation. Also, it will be increased in burn. But however, there are advantages that we are going to discuss later. We also did all these fluorescence measurements and little biophysics here. We deconvolute them where you can various components. This, those who are students, fluorimetry, the 680 nanometer array originates from photosystem 2, and 700 is originated from photosystem 1. When you see these mutants in the top one panels are the control light conditions. The bottom is 500 microinstance, you consider as a little higher light, where the photosystem 1 in mutant is significantly reduced. That means, the stoichiometrically they are different, but by the energy transfer levels are little different in the mutant, also because I said the previous one because of maybe due to ROS production. And we wanted to know what is happening to the thylakoid super complexes, called the super complexes, where we use a mild detergent, beta-DM, and we can separate all the complexes. Those are the complexes, mega complex, PS2 super complexes, PS1 monomer, dimers, and LH2 trimers and LH2 monomer. Whether there is anything, change is there. Of course, surprisingly, for our surprise, the wild type, there was no significant change. Whereas in PGR1 and PGR500, these are the main super complexes on the top band. They have, it's not a degraded, it's there is a change in the complexes. That means pigment interaction is doing. We did a lot of other studies to prove this one. And very interestingly, the trimer of LHC2 trimers, which is a major component to harvest the energy, this is been increased. That is because to adapt or acclimatize to the highlight. Similarly, in PGR5 also, you could see all of them are changed. This trimer complex has been increased because to adapt to this the highlight. Further, we did want to go deep into what is happening inside the complexes. So we put them, isolated them on mild detergent, 1% beta-DM we used. And we went ahead with the sucrose density gradient. Sucrose density is about, it takes about almost 16 to 18 hours to spin down all these complexes. The band 1 is originates attribute to the LHC2 complexes, the trimers. And band 2 is a photosystem 2, band 3 is a photosystem 1 complexes. And we did, SDS gels to see all of them. And there are, these, in PGR5, they significantly changed all the super complexes. Further, we did circular dichroism spectrum to those who are, aware of the biophysics. This circular dichroism can tell the polarized light can be pass on to the biomolecules, and where you can see the the pigment interactions and pigment protein interaction, how their changes can be to the any biotic stress or So where the light arresting complexes, as I said in the previously, the sucrose density gradient, there is a, or in the blue native gels, the light arresting complexes are even much increased. That means the conformationally they're almost stable, the first one, the top band. And where the photosystem 1 and photosystem 2, the second band and this one, this is a PGR5 and PGR5, you see the PGR1 and PGR5, this is significantly changed. This is the last band, that is a band 1, band 2, band 3. So which shows that there is the interaction between the photosystem complexes, especially the photosystem 1 and photosystem 2 is being changed. So further, we wanted to see the pigment level changes where the carotenoids has been significantly used. This was used HPLC quantification using standards where you can see the blue one, the blue bars, all the carotenoids. In the highlight condition, that means you can see that 500, and we also used NPQ where you want to see that whether NPQ have anything, any change is there. But NPQ, because NPQ is derived, there is a knockout between the zeaxanthin dependent gene where we don't see any zeaxanthin increase in that one. So we can see here the pink color, there is no zeaxanthin increase, which is indication that our results are accurate. Further, we did all this one, deep into the analysis of pigments, how the pigments are present in all the super complexes. That from the one which we have isolated from the sucrose grade. So here, what you can see fraction 1 and fraction 2. So the fraction 1, you can see clearly that all these one of the chlorophyll to B ratio, anything, yeah. And fraction 1 and fraction 2, this is the fraction 1 and fraction 2 and fraction 3, where you can see, you can maybe can see that in the down, the lutein content is increased in trimer, which is very well agreed that lutein have a more of more present. The lutein is presented more in LHC2 complexes. Whereas the super complexes, beta carotene content increase significantly under all the conditions. That means in all 3 complexes. And lutein and beta carotene content were less in PS1 complexes because the lutein is always less in PS1 complexes because, as I said, it will be more present in the PS1, PS2 and LHC2 complexes. So these are all because, again, the highlight, what we wanted to say that, previously I said, non-photochemical quenching is a very important, that is a zeaxanthin dependent quenching. The zeaxanthin is increased significantly in our study. So we also did with RT-PCR to see that whether the specific genes are enhanced or, as I said, lutein is increased and zeaxanthin is increased, beta carotene is increased, and what happens to the, the gene. So accurately, whatever the data we presented the previous slide, so the gene, their RT-PCR data also matching with that. You can see that all these one, the bottom, they're all controls, and whatever the rising bars, which are all the genes, you can see in the right panel, lycopene to, let's say, beta carotene, LCYB gene, and ZEP gene, that is the zeaxanthin dependent genes, they're all also been increased. So that is which is our, whatever the data, the biochemical data is matching with the RT-PCR data. So I want to skip because I don't have time, how many minutes I have further? So we wanted to, again, want to show that why all this happened. These events of, whatever the events happen, this is all because of, there is aggregation. The aggregation is due to, again, the ROS components that will come to the next slides sometimes bit later. some of the specific proteins are aggregated in the, especially in the mutants. That is a PSA-A, it is a photosystem 1 core complexes, one of the significant protein, and D1 protein from the donor side of the a D1 protein of donor side, and some of the LHC proteins are getting aggregated. Hence, there is a lot of changes in the photochemically. There is a lot of changes we have seen. So, to summarize only this chapter, the light arresting complexes, trimer complex abundance, absorption and pigment interaction by circular dichroism and blue native gels and other things was seen that mostly the PS1, PS1 LHC1 is mostly, changed. That is also, I have said, the fluorometry results where the photosystem 1 is reduced. That is because of this, reduction in the proteins. And xanthophylls and lutein and beta-carotene were less in PS1 LHC complex, which is, again, I said, they will be lower in all these complexes. And most of the pigment interaction has been changed in the complex. Now, I just want to go to the next story. Why, in these mutants, the autophagy is, just want to give you a brief overview of this one. Autophagy can induce through the ROS. ROS can be generated in all the complexes, mitochondria, peroxisomes, vacuoles, as well as endoplasmic reticulum. The autophagisms can degrade into lipids. That's what I wanted to say here. So the same thing happening in our studies. So what we did, we just tested in the highlight where you can see significantly the ATG8 is significantly increased in highlight conditions in control, wild type, and PGR1 and PGR5. And through, you can also see here the increase of ATG8 is very, very significant. So that means the autophagy is increased significantly in our mutant. We also wanted to see that if autophagy increase, as we expect, there is a lot of vacuoles will be accumulated. You can see the big vacuoles in these mutants. That is because now you can see that autophagic bodies accumulates in cell vacuums. So now further we wanted to see that, what are the, all these autophagy is one of the mechanisms. I am not telling that autophagy alone can induce the lipid production. Some of the proteins are accumulated. Those are, lipid, that is a triacylglycerol is the one. Synthesis of triacylglycerol, you have a PGAT and TGAT. So these are the ones where they will be very involved. In these mutants, the PGR and PGR5, you could see this, a significant increase of the PGAT and DGAT, where we can see the bar diagram also. So which indicates that triacylglycerol accumulation is high in these mutants. And we also, I will skip the fatty acid biosynthesis. We also can see the TAG, that means triacylglycerol is significantly accumulated in PGR1 and PGR5, the top band. And how they can, they accumulate, there is something, if there is something accumulated means biologically, there will be some kind of, some conversion should be there. The membrane lipids, that is the MGDG, DGDG, SQDG, and the PE, these are the membrane lipids. The membrane lipids quantity has been significantly reduced. This you can see here. So this is significantly reduced. So the membrane, the lipids are being converted into TAG. That's what we have also done a lot of further experiments to identify them. So further, we did all this metabolomic analysis. All the metabolites were quantified. You can see with, we did a lot of, the analysis. Just want to present here the whatever the scale 2.5 fold, which the PGR5 has accumulated. You can see this one, the green line, wherever the green spots, all these metabolites, carbolic acid and amino acid metabolism, fatty acid metabolism, they have been, the green or the one, it is significantly increased in PGR5 mutant. So we also did, the entire, I'm not showing entire data, to summarize entire metabolic pathways. So you could see here, a lot of, now the green, whatever, they can, the wild type versus 500 microinstance, PGR1 500, PGR5 500. We analyzed with the various directions. So where you can see here the PGR5, PGR5, 1, 2, 3, third, the third one, wherever is the third one, actually the green line. So those are all the metabolites which has been significantly accumulated in mutants. So what we wanted to conclude from this study, and because it's too much, it requires at least another half an hour to explain to you. Maybe I can share these slides, you can see this and all these metabolites. So what I wanted to summarize here, the increased ROS levels in PGRS cell could play a dual role in signaling to activate lipid biosynthesis and autophagy inducers start recycling cellular components to fuel lipids. So now again the membrane lipids have been decreased, that's what I said, the membrane lipids have been decreased in order to synthesize the TAG, that means triacylglycerols. So with this, we are studying, looking at the reactor level, whether the PGR5 can be used as a potential, feedstock, organisms for, industry of, lipid production as well as, the agriculture, you can use for feedstock for many things. So I'll just only one slide. Similarly, we did iron stress where we have seen very significant accumulation of lipids. Yeah, where you can see this, the iron stress is a severe iron stress where you can see large bodies of lipids. A large part of lipids are increased. So this also we are looking at the reactor scale, whether you can produce enough lipids for industrial scale. With this I stop here and thank you for giving the opportunity. So if you have any questions and suggestions, I'll be welcome. Thank you very much.
Thank you, Dr. Rajagopal Subramaniam. It was a highly intensive work. Normally people do surface work with the mutants, but you gone through intensively. You used a lot of techniques in that. Flow cytometry, then staining, various staining methods, then microscopy, cell sorting, protein studies, pigment profile, then metabolome, all these. So this is the way we all should follow for studying a particular phenotype. And so with this, I would like to invite our audience to give questions.
So nice presentation, sir. So I just one basic question. In your study, these mutants, they are producing the high amount of lipids, and you showed the growth parameter based on the OD, optical density. So I just wondering if you have tested the biomass production, like because the change in the shape of the cell will interfere with the.
No, I didn't keep that. Yeah, please.
Is produced at the cost of biomass. We all know, sir.
Yeah. Now that's what I initially said. The mutants, they grow slowly, 1 day, 2 day, 3 day, 4 day. Now, by third day, wild type is reaching to the stationary phase. Whereas the mutants, it requires some time to adjust. That's what I said, there are non-photochemical quenching components is involved in photoprotection. After third and fourth and fifth and sixth day, even they grow much better than the wild type. So there is no compromise of biomass, but you need to wait little longer time. And biomass versus lipid production also is high if you look at, sixth day or seventh day. The only thing, wild type, you have a less, within 3 days you can reach to the stationary phase, but that it takes little longer, but we get good enough biomass. Oh, lipid 1 is to 2 times. No, the 2 times in highlight, whereas in iron we get 6 times. Very huge amount of, we have standardized that iron can induce a significant amount of lipids.
A very nice presentation, sir. It was wonderful to listen you. One question related to your B and page data. You showed that there is a accumulation of LSC protein in the PGFPR mutant, and if you look at the gel, there is a PS1 dimer is very in lesser amount. So have you correlated with the activity assay of PS1?
That's what I said. If you see the fluorometry, the fluorescence emission spectra, the accumulation of photosystem 1 and the activity of photosystem 1 is significantly reduced in the mutants. So that is because, as also I mentioned, the over-reduction of the photosystem 1 at the acceptor side, that is the pyridoxine, where it over-reduced and produced more RYS. It can the first one to damage, that is the photosystem 1. Thank you. Yes, sir.
When you are a student of Professor Mohanty and you are doing so well, first of all, congratulations for that. And I just have one question. This is regarding, you said that iron is basically promoting lipid production, its accumulation. Huh? So what could be the mechanism associated with that? Because that's very interesting. Because that's the thing, today we are talking about biofuels. I mean, it's a wonderful thing. What could be the mechanism associated with that?
So the mechanism, if you want to, say that still we are working out. So as I said, we are not sure whether autophagy alone is inducing the lipids. That is one way to say that, but the literature support shows that autophagy is inducing the lipid mechanism. The other one, as I also said, the membrane lipids are degrading. The chloroplast membrane lipids are degrading, Mg, Dg, Dg, Dg. So this is one, we have to do the pulse chase experiment. I am telling you, revealing the experiment to that, to identify whether it is really exactly the membrane proteins are, membrane lipids are increasing, that we have to look at that one. At the moment, I say the answer is yes, this is through the membrane proteins is converting into the lipids. If you look at the KEG pathway, now KEG path, we also did a lot of transcriptome analysis, I think with Ranay is going to talk, where it shows that I also said, especially at the site of, there is also, I didn't tell you that starch accumulation is there. Starch also, one is through the chloroplast. This is the cumulative mechanism. One is through the membrane lipids. The other one also comes from the starch. So we have seen a lot of metabolites as well as the membrane lipids. Through both of them, some of the the acetyl-CoA, basically the primary the precursor for synthesis of all these components, lipids. The acetyl-CoA is entering into the glycolysis, not glycolysis, sorry, the PGAT and DGAT at triacylglycerol accumulation. Among all these lipids are increased, but specifically triacylglycerol. The ultimate goal is to increase the triacylglycerol. So I hope, still we are not yet clear, but we have, through our transcriptome analysis and metabolomic analysis, I would say that through the KEG pathway analysis, KEG pathway is the main playing a role, that is a big DGAT and PGAT, which can convert into triacylglycerol through the acetyl-CoA. Thank you, sir. Thank you for your, yeah. Sir, I'm not able to hear.
Probably you are trying to stress that autophagy, iron is promoting autophagy. Iron is promoting autophagy, and that is the thing which is responsible for the accumulation of these lipids. Do you really expect that high levels of, you are indirectly trying to say that membrane lipids, membranes are getting degraded, and membrane lipids are the things which are getting accumulated. Do you really expect that much? Actually, I was thinking, did you ever try to see whether lipid synthesis, fresh lipid synthesis is occurring or not?
Yeah, that's, again I said, sir, there is some amount of membranes are getting into the lipid, the triacylglycerol. I agree with you, and a very good question. And also I said starch accumulation also we have more. Through both of them, that's what a cumulative effect. This accumulation from the membrane lipids and also through the, the starch accumulation. So we need some further experiments to prove exactly how it is coming. When we submitted this paper also, we have submitted big journal, but the same question. And Pranay is finished PhD, and there is a Nisha is the one who is another girl. She already finished her PhD, and she is in US. So we have to depend on other new students. It will take some time to prove all those. Thank you very much.
Respected delegates, this is a very interesting topic. So in the post session time, we can discuss more on this. So we can continue with the next upcoming sessions. Thank you.
Thank you, Professor Subramaniam. For an interesting lecture, I would like to call upon Dr. Krishna Kumarji, co-chair of the session, to present the memento as a token of our gratitude. Sir, please. Thank you. There are a few announcements. The participants for oral presentation who have registered today are requested to submit their presentations at the help desk as soon as possible, and Dr. Arun Kumar and Dr. Yogesh Mishra are requested to submit their presentations for short lecture at the mic point immediately. Thank you. Now I welcome Dr. Jose T. Puttur, Head Department of the Botany, to share the activities and contributions of Department of Botany, University of Calicut. Welcome, sir.
I am just stealing 5 minutes from the session. I have been asked by my department council to present the department before the elite audience here, so that we can invite more collaborations and work together if possible in any line. So this short presentation about my department. I'm skipping this. Anyway, we have funding from many of the national funding agencies, and also our department has been selected as the Center with Potential for Excellence by Kerala government in 2017, and we have got a fund of more than 1 crore for the development of the department. And already I told our former professor was honored. He was very rarely the professors get Padma Shri, and he has got a great contribution in the field of taxonomy. And this is the faculty strength I have. And in the department, we have 1 senior professor and 5 professors, associate professors 2, and assistant professors 5, and also some supporting staff. And I told in the yesterday's inaugural session, our department is hosting the Indian Association for Angiosperm Taxonomy, just like ISPP is hosted in IARI. We are hosting this office of IAT, and also the 2 journals are being brought about in association with our department. One is REDIA, solely for Angiosperm Taxonomy, and another is Eco Chronicle, for more into ecophysiological and environmental studies. And this is, yesterday you have experienced our garden, which is spread out in 19.5 acres. It started in 1971, and this is mainly for ex situ conservation of various groups, and it is one among the largest university botanical gardens in India. And we have a herbarium, which is, the acronym is KALI, And we have more than 1 lakh collections. Some of you have visited this herbarium. And almost 85,000 collections what we have has been digitized. You can access it from anywhere. And the type species we have 170, and it is one of the largest university herbarium. And soon it is going to be accredited by National Biodiversity Authority as the national repository. So if any sort of studies has to be done in the plant field, and if you want to deposit the plant material, they have only 16 in whole of India. Now ours will be the 17th national repository. And we have an inter-university center for plant biotechnology also associated with our department. And yesterday some of you must have seen the touch and feel garden for visually impaired with the funding of Ministry of Environment and Forest. And even the blind people can enjoy the garden and they can feel how the plants look like, in the sense that they have a braille script. They can just find out how in which family it belongs to, what is the species name and all. And this is being funded by MOEF. And we have an arboretum maintained in 13.5 acres, and it is an evergreen patch of natural vegetation, allowing succession in natural way. And this is a picture of that. And there is a university park, which you will be visiting today again under the strength of botany department. And we will be having the dinner today in the garden if the weather is fine. And this is the laboratories what we have. We have 9 divisions in total: genetics and plant breeding, cell and molecular biology, cyanobacterial diversity, environmental science, bioinformatics, fungal diversity, physiology and biochemistry, angiosperm taxonomy, biotechnology and bryology. This is the 9 specializations what we have in the department. And the research areas are confined to these 9 areas. And the PhDs produced from our department is still, until now, it's 238. The articles produced is 1,620. Books are 52. We have already gathered upon more than 9.27 crores of funds from various agencies. And patent, we are still behind a little bit, only 1 filed until now. And popular articles also. The citations cumulative for our department is 16,403. And the I-10 index in cumulative is 294 for our department. And the down data is of 5 years, last 5 years data. And we have been awarded with the DST FIST grant for 2 cycles repeatedly. And also we are getting an infrastructure grant from the Kerala government also in the name of SARD. And we have also contributed, or our department has mainly worked on these regional floras. You can see the big list of regional floras, where our department has also the revisions of various taxonomy families has been conducted. And this is the list of that revisions being conducted. And the major programs taken up, you know about the GYAN program that is funded by MHRD. That is, we get a foreign faculty to conduct the GYAN program. They come over here and do the 2-3 weeks workshop. So we already conducted 2 GYAN workshops, erudite scholar residence program, frontier lecture series. We have also conducted international conference in the area of ferns, and also botanical congress. All these are being hosted over here. And this is the glimpse of the international workshops being carried out in our department. And this is again erudite scholar scheme which has been carried out in our department. And we have international collaborations. The list over there in the left is visible over there, Royal Botanical Garden, and also with Warsaw University, Professor Kalaji, Dorothea Bartels, we have collaborations. And also we have collaborations with IISc Bangalore, Department of Botany, Delhi University. And we also recently, last month, we have brought about a biodiversity pictorial guide. This book is available in the department right now. And if anyone would like to have, this is a book where we have collections of around more than 550 flora and 250 fauna, which is available in our department, which is being collected from our department. And yeah, that's all. Thank you.
Thank you, sir. I would like to hand over the mic to Dr. Heber KB, chair of the session, to continue with the short lectures of the technical session 4. Sir, please.
Thank you, Dr. Jose Futu, for giving a bird's eye view of the department. I think it is worth, some of the things are unique here. I think it is worth visiting also. It is very interesting. Now coming back to the session, this is short lectures. There are 3 lectures slated here, and 15 minutes for each talk. I would suggest, I think, 13 minutes for the talk and 2 minutes maybe for the discussion. That way, if it is gone, I think that would be better. Without wasting time, I think we will call. So, Dr. Shailendra Pratap Singh, Senior Assistant Professor from BHU, Banaras Hindu University. Dr. Singh.
So, thank you very much, sir, for giving me the opportunity. Especially, I thank Professor Jose, sir, for giving this kind opportunity to present my work. And from yesterday, we were hearing about the plant. Today, we got the algae. Now, I'm bringing you to the cyanobacteria. Basically, I will be presenting my work here related to the photomorphogenesis in cyanobacteria. So we already know from morning start that what is the role of cyanobacteria in shaping the present day life. They produce the oxygen on the early Earth's surface, and now several biological processes have been well studied in cyanobacteria, including photosynthesis, iron homeostasis, and redox regulation, etc. And now we are moving towards the application of cyanobacteria for the biofuel production or other valuable compounds production. But the cyanobacteria, they are easy to grow, and but at the same time there are several abiotic stress factors like light, temperature, salinity, and oxidative stress. They compromise the fitness of the organism, and then we end up with the lower amount of biomass. And I work for some time at the Reliance Industries algae to oil setup, and the industry, they want the good amount of biomass from the biologist, having the good amount of lipid, carbohydrate. Protein is not required for the biofuel production. So anything having good biomass is the major requirement. And then if you get the good amount of lipid, then it is add-on. But nowadays we also have the STL technology, hydrothermal liquefaction, where these industries are using the whole biomass for converting into the biochar. Okay, so with this background, being photosynthetic organisms, we know that the light acts as an energy source to drive the photosynthesis. And meantime, the quality, quantity, and direction of light, they act as a light signal for driving the development and photomorphogenesis. And the best example we know about the classical example from the DETU lesson, where I won't go in detail, but just want to bring the attention here that the elongation and shortening of the hypocotyl is associated with the elongation and shortening of the cell. Okay. And this is because cyanobacteria or algae, they are aquatic organisms. This is the spectral distribution of light that is falling on the top surface of the water column. And we see like the red light is mostly absorbed by the photosynthetic organism, like eukaryotic organism. And at the lower depth, and depending on the water quality also, turbidity, the spectral quality and the depth the light is reaching will be compromised. Then also like for example, photobioreactor density will also affect the light quality. So at the lower depth, we are having the more green light environment. Okay, and then we can also, if we are talking about the cyanobacteria, because they don't, generally cyanobacteria, they don't use blue light. Okay, because of the phycobilisome. So the higher density and or at lower depth, the organisms have to evolve in such a manner that they can utilize the green light to drive the photosynthesis. And this phenomenon has been well studied in cyanobacteria that allow the cyanobacteria to utilize the green light depending on the spectral property. And this is called complementary chromatic acclimation. The cyanobacteria, they mainly use the phycobilisome for the excitation of PS2. That's why generally cyanobacteria can't use the blue light in comparison to the eukaryotic plants. And they change the composition of phycobilisome, the outer rod parts, where they can change the phycobiliprotein composition depending on the light environment. If they are receiving the green light, they will have more phycoerythrin that can absorb the green light to excite the PS2. And if they are growing in high light environment where the red light is more abundant, they have the phycocyanin, and this they can change. So this overall phenomenon is called complementary chromatic acclimation. And there are different kind of CCA, and among these different,
The one that is CCA type 3 is well studied in Fremella diplosyphon. Here they change the outer rod part composition depending on the light environment. So this is the Fremella diplosyphon. If you grow the Fremella in green light, you have more phycoerythrin. So they appear red in color. If in the red light, you have more phycocyanin, so they will appear bluish-green in color. And based on different genetic studies, mutant development, the RCA photoreceptor was identified, which senses the light quality. And if you don't have the RCA, this Fremella diplosyphon become colorblind. They cannot differentiate between green and red light, and they accumulate intermediate level of both phycoerythrin and phycocyanin, and they appear blackish in color. So based on different studies, now we know more about the signal transduction pathway, where under red light, this RCA acts as a photosensor. It gets phosphorylated. It has got kinase activity. And then there are two downstream response regulator, RCA-F and RCA-C. After phosphorylation, they bind to the promoter region of phycoerythrin and phycocyanin. And at the same time, they act as an activator inhibitor for the synthesis of phycocyanin and phycoerythrin. So in the red light, they promote the RCA phosphorylation, promote the synthesis of phycocyanin and inhibit the phycoerythrin, while in the green light, there is no binding because the RCC is not no more phosphorylated. This phycoerythrin is not more inhibited, while there is no activation of phycocyanin. So in addition to this pigmentation, there is also change in the morphology. Under the green light, where that is common with the low light environment, the cells are longer, while in the red light grown cells, the cells are smaller. And we also notice that the RCA mutant, they also have the altered cellular morphology. So we were working to get more detail on this morphological aspect. We started with focusing on the oxidative stress, because oxidative stress also generally alter the morphology. So we grew the cells in the presence of ascorbic acid to see if we can decrease the ROS level, and this decrease in the ROS level can change the morphology. So here in the green light, we have lower ROS level in comparison to red light, and when we added the ascorbic acid, there was decrease in the reactive oxygen species. And if you see this under the red light, we were able to get some elongation in the cell length. So that indicates that in some part, this higher ROS level under red light is associated with the shortening of the cells. In addition to that, the filament length is also higher under green light in comparison to the red light. But when we add the ascorbic acid, which is associated with the decrease in the ROS level, we were getting the higher length of the filament. So this having the higher length of the filament of cell is also beneficial for the industry, because they spend a lot of money on the centrifugation and harvesting of cells. So if you have the longer cells, that will also cut down the cost associated with the biofuel production. So further we investigated if we can change the morphology of RCA mutant, because the RCA mutant was found to have a very high level of ROS in comparison to the wild type. So we thought maybe this defect in the morphology is associated with the ROS level, but we were not able to decrease the ROS level by adding the ascorbic acid up to beyond certain level, and there was also no change in the morphology. So that indicated that in the wild type, this ROS is playing some role with the morphology, but in the mutant, there are some other genetic factors are involved in controlling the morphology. So to further investigate that, we focused on the BOLE gene. These are the morphogene and the MREB, which is the homologue of actin of eukaryote cytoskeleton protein. And we tested the gene expression level in mutant, and we also generated the BOLE knockout. We were not able to completely remove it, but overall, if I summarize this slide, that this BOLE was negatively controlling the expression of MREB. If you have the higher level of MREB, you will have longer cells. That is well established in the bacterial system. So, and this BOLE expression also goes high in the bacterial system during the stationary phase, where you get the spherical morphology. Okay, so we also demonstrated that this bole can bind to the promoter of the MREB region, and so it is controlling the expression of MREB. And then RCA also accumulate lower level of bole. So this regulation is affected in the RCA mutant. So these are just the length and width measurements, so I will skip that. Then because the sequence of bole and MREB from Fremella is highly conserved, so we wanted to test if these MREB and bole have the functional conservancy in the bacterial system. So we overexpressed, and we showed that. So normally the upper panel showed that in the normal condition during the stationary phase, you get the spherical morphology in E. coli due to the overexpression of bole. But if I overexpress the bole even at the earlier phase of growth, you get the spherical morphology. Similarly, MREB during the stationary phase, you get the spherical morphology because of the negative regulation of MREB. But if I put Fremella MREB, even during the stationary phase, we are getting the rod-shaped morphology, elongated cell. So that showed that these proteins are highly conserved and can also function in the bacterial system. So from earlier studies, we got the information that this bole expression is high in red light, and also the RCA, they accumulate lower level of bole. So we wanted to test those things at the protein level after producing the antibody, etc. We did the transplot, and we confirmed the result of the transcription at the protein level, and we also showed that this RCA accumulate lower level of bole. So by that we developed the hypothesis that if we overexpress the bole in the Fremella, maybe we can revert back the morphological defect in the RCA, and also this overexpression of bole can give the spherical morphology or short cell morphology under the green light. So we were able to show those things with the overexpression of bole, and also there was tremendous increase in the filament length. And these things, I am not showing the data here, but these things were also associated with the decrease in the ROS level. So bole is somehow controlling the ROS level. Same thing we did with the RCA mutant. We overexpressed the bole, they having the higher amount of bole, and we got the morphology that was close to the wild type strain. So that said that this RCA having the low level of bole. So based on all those studies, now we have this model where we don't know how these things are working, but RCF and RCAC are definitely not involved in this pathway. But this RCA dependent high production of bole control the MREB, then we get the overall this kind of structure. So we also did notice there is change in the morphology or shape structure of carboxysome in RCA mutant, and we did the protein study. There are several proteins involved in the structure of the carboxysome, and the main thing is that there are the carboxysome structures. There is one protein CCMM, and there are two variants of this CCMM, and the certain ratio of these two variants are required for controlling the morphology, and that was altered in RCA mutant. This is just two more slides. Now recently we have started the blue light at different light condition with the Senecococcus elongators, and we noticed that the blue light and green light increase the length of the cell in comparison to other light, and we also noticed that there are some spaces in the thylakoid membrane where we see the stacking. Okay, so there are, it's showing that the decoupling of the phycobilosome. And these, we tested the different cytoskeleton protein, and we saw that the expression of these genes is altered by the light. So we are getting more into detail about those things, how it is working. So with that, I will stop, and I would like to thank my PhD supervisor, Professor Donat Peter Heather, then my another kind of supervisor, Professor R.P. Sinha from BHU, Beronta Montgomery, CCA work we do with that person, and then Cyril Kerfet, carboxysome, and these are my currently PhD student. Vinod has completed PhD.
Then Somila is working on the monothiol glutardioxin. So with that, I will stop here. Thank you.
Very interesting talk, Dr. Shailendra Pratap Singh. In fact, you had shown that how the different part of this electromagnetic spectrum, which part is more useful for the production of biomass in lower organisms. It's a very interesting talk, and you have tried to understand the mechanism, how it is either elongating or expanding the cell and biomass production is increasing. If there is any specific question on this topic. No, one thing, you said that green light and red light, these two are important. How much difference in the biomass production is there?
So it depends on the cyanobacteria.
Yeah.
So if the cyanobacteria has got the capability to use the green light, there is not much more difference. But not all the cyanobacteria can use the green light, not all the cyanobacteria can use the blue light. So if they have got this mechanism, CCA3, so green light, they are cyanobacteria producing the phycoerythrin, and this phycoerythrin is present in the phycoglycol, they will grow efficiently. And similarly with the blue light, several cyanobacteria, mainly marine cyanobacteria, they can change the chromophore, phycourochlorine or phycoerythroglin, that attach to the same apoprotein. So if they have that ability, you get almost equal amount of biomass in both red light, green light, or blue light. So it will depend. So, but in this case, Frenella, they grow almost equally.
General question. Because when light comes, plants also grow, higher plants or all these things. Any difference in growth in terms of growth kinetics between these cyanobacteria and higher plants? Suppose you have one cell of cyanobacteria and one cell of plant. When light comes, will there be any difference in the total biomass produced?
So in context with the green light,
Whatever be the light.
so cyanobacteria, they have higher productivity. In comparison to, they have more high photosynthetic efficiency and productivity than the, there are, it's not my word, but there are reports like they have more productivity than in comparison to the grass.
That is why industries are interested in this kind of. Okay, thank you.
That's why the people are focusing on the biofuel using algae and cyanobacteria rather than the grass.
Is it related with light intensity? What about intensity? Is there any?
So what I use there, like for green light, blue light, or red light, for this is experiment I use around between 20 to 30, more specifically like 25 microvolt per meter square. And if you increase the light intensity, you get the decrease. So these elongation and shortening, they increase the fitness of the organism by providing the more larger surface area for the better absorption of the whatever the resource is available, and they can also accommodate high amount of thylakoid. For example, in the Synechococcus, we have also got the increase in the lipid content. Normally, cyanobacteria, they have 9% of biomass lipid content. That's why the preference is given to green algae if you are focusing on the lipid. But with the blue algae, we have increased up to 14%, and we have also developed several knockouts where we can get up to 18% of the lipid content. So we have recently filed for the patent. That's why not having those data.
Thank you, Dr. Sailesh. All the best. Okay. Now I think let us call upon Dr. Prabhat Kumar Sharma, Senior Professor, Department of Botany, Goa University. Dr. Prabhat.
Good afternoon, friends. In my presentation, I will be talking about a comparative study of two varieties of rice. One is salt sensitive and another is salt tolerant. And we'll see possible marker for developing salt tolerant based on these varieties, which are naturally salt tolerant. One is salt sensitive, another is salt tolerant. So in general, we all know the salinity is one of the most important abiotic stress after water stress. Approximately 70 million kilometers square of the land is saline land in the world, and out of which about 2 million kilometers square can be recovered for the salinity agriculture. And India also, we have about 1.2 million hectare land which is affected by salt. And in Goa, where I reside and work, approximately 80,000 hectare land is saline land. This is a low level land where during low tide, high tide, the soil is inundated with salt. And there are certain varieties, local varieties, which are adapted to grow in that salt condition, though the productivity is a little less. So we decided to compare to see what are the parameters which is making these varieties salt tolerant. So the process of the mechanism of salt stress is known to everybody. Here I just present that salinity rapidly causes osmotic stress because of the physiological dehydration, and which lead to buildup of ionic stress because of all this salt accumulation. And these two together ultimately lead to the oxidative stress. And those plants, the sensitive plant, are not able to handle these stress factors, generally have a low productivity or may not be able to grow. And now we have to remember here two things, that plant is trying to sustain itself in the stress condition. So they have developed those mechanisms. But that is not sufficient for us, because we want plant to produce maximum even under the stress condition. So this is what we look for. So those are the plants which are able to have protective mechanisms such as ion homeostasis and better antioxidant activities and prevent damage to the membranes and things like that, are able to sustain themselves and give some productivity. So we have grown these plants under the control conditions, and we have provided them saline condition up to 200 millimole concentration. And we have grown them up to 21 days, and then they were harvested, and all the parameters were analyzed after 21 days of the growth. We have looked at the morphological parameter through external and internal morphology, what changes are there under salt conditions. We have looked various physiological parameters, the common which are studied, such as relative water content, pigments, photosynthesis, ion accumulation, oxidative damage, etc. And we have also looked at the molecular parameter, such as gene expression and proteomic studies. And before I go further, let me tell you, this is a PhD work of one of my students. She couldn't be here, so I'm presenting the work. And all the parameters, all the studies were done by using third leaf of the plants. So here is a very nice picture, simple picture. You can see Jaya, which is a salt sensitive variety, and Corgut, which is a salt tolerant variety. You can see the effect very nicely. And here up to 120 millimolar, you don't see much of the effect on the growth in the Corgut. And when you go beyond 120 is mostly what is available in the soil. And artificially you can give as high as you want. But the plant is growing very nicely up to 120, and the root is also very relatively better, not much, but relatively better. And what is most interesting is the pigment level is very high in the Corgut variety. And these are the numbers of the same plants with regard to shoot length and root length. So there is a considerable decrease in the shoot length and root length, 74%, 84% in the sensitive, and 54 and 64% in the tolerant variety. This is regarding biomass, shoot root, biomass, fresh weight, dry weight, more or less similar data, so I will not spend much time. So sensitive variety has a much effect on the biomass, and tolerant variety has relatively less biomass effect on the biomass. Let's come to the morphological data, and these are the first two slides represent the control in Jaya and Corgut. And you can see the trichomes structure, which is relatively small in Jaya and quite large in the Corgut variety. These are the normal plant. And then the third and fourth column, these are the salt tolerant, 200 millimolar. And you can see Jaya, the trichome size has increased, but number remain more or less same. And in the Corgut, you see sizes further increase considerably compared to the control. We also looked at the stomata, and Jaya variety has a large stomata opening compared to the Corgut variety, which is the tolerant. And another thing which we noticed is the cuticle papillae, which you see here, dot kind of structure, which was also seen in much greater number on Corgut than on the Jaya. This is regarding internal morphology, and this is a very interesting slide. What you see is that when you give salt stress to salt sensitive variety, the grain structure is distorted, and there is a slightly swelling also, whereas no such distortion in the grain structure was seen in the resistant variety. So these morphological parameters may have a considerable effect on the plant water status at one point, and they may also have a providing a storage for the salt, better storage of salt in the tolerant variety. We also looked at the RWC, and the parameter match with our morphological data. You see in the RWC, the green one is salt tolerant Corgut variety, and red one is here salt sensitive J variety, and the X scale is the concentration of sodium chloride. So when we see the RWC, that very large decrease in the RWC was there, 73% compared to about 27% in the Corgut. So we understand that if plant water status is distorted, most of the physiological process will also be affected. And better plant water status means better physiological processes. So now we looked at the various physiological processes. One was the pigments. So this was done by using HPLC. Qualitatively, there were not much change. We saw our basic carotenoids and chlorophyll, A and B. But when we looked into quantitatively, we saw a large increase in the pigments in Corgut variety over the sensitive variety, and the chlorophyll A to B ratio was also increased considerably in the tolerant variety. And when we looked into the carotenoids also, the carotenoid content also increased considerably in the tolerant variety. This is a photosynthetic parameter measured using IRGA. So you have normal net photosynthesis, transpiration rate, and stomatal conductance, and internal carbon dioxide concentration. So we saw as much as 81% decrease in the net photosynthesis rate in Jaya, and whereas only about 48% in Corgut. And more or less same kind of effect was seen in stomatal conductance and transpiration rate also. See, internal carbon dioxide was not decreased. So basically the metabolic processes were working, and these was related to the plant water status or stomatal conductance. We also looked into fluorescence measurement, and what we noticed here, you see that FO, which is indicative of energy transfer within the light harvesting complex. So that the increase there is related to the decrease transfer, that means disorganization of light harvesting complex. So in Jaya there is a slight disorganization of light harvesting complex, where you see 40% distortions. I mean increase in the FO, which is related to the distortion in the arrangement and orientation of the pigments within the light harvesting complex. Then we have a photosynthetic efficiency also, which was declined quite considerably in Jaya, 68% compared to only 30% in Corgut. And likewise QP, photochemical quenching, which is light utilized for the photochemical molecule like ATP and NADPS2, which was quite high. Sorry, I think we have gone one more slide up. Yeah. QP was quite stable in the tolerant variety compared to the sensitive variety, 76% vis-a-vis 43%. And QNP, which is a non-productive energy, was higher. I think I will stop. I'm sorry. Because you have started this buzzer now, you should have started from yesterday. Then this situation would not be there. Huh? Yes. So please give me time. I will rush through. I know. Okay. Yes. So QNP was higher in the Jaya variety, which is good actually. So it is trying to survive, but at the cost of the productivity. Whereas the QNP was lesser in the core good variety. Then we looked into accumulation of salt also. So sodium accumulation in the leaves were more or less same in both the variety, 15-fold and 16-fold. In the root, it was much higher in the Jaya variety than the core good. Chloride level was also much higher in the sensitive than the tolerant, but it was not very significant difference. And in the root, of course, there was much difference as far as chloride content was concerned. It was 5.4-fold, whereas in the core good it was only 3-fold. And for the potassium content was very stable in the salt tolerant variety. Actually, it increased 12%, whereas potassium content in the leaves in the Jaya decreased by 56%, and more or less same trend was followed in the root also. So the potassium to sodium ratio increased in the tolerant plant over the sensitive plant. Then we followed that ROS production, and here we see the S2O2 production is almost 200%, compared to the tolerant about 5. Why it keep changing without touching? Okay, so 136%, and more or less same trend was seen for hydroxyl ion also, okay, which is another ROS. Then we also looked at the proline. Proline is a organic osmotica, we all understand, and it is accumulated to cope up with the plant water ratio, to increase the water potential in favor of the plant. So there was a very high increase, 15-fold increase in the sensitive variety to make it survive against the salt stress, at the cost of course, productivity, whereas in the core good it was only 5%. And we looked at the antioxidants such as ascorbate also, which was comparatively higher in the tolerant variety. And we looked at the oxidative damage also to the lipids, membrane, proteins, as well as the leakage. And what we see is all these three parameters were higher, which is indicative of membrane damage, oxidative damage to membrane in the cell. And most importantly, I will like your attention on the lower slide, MSI, that is indicating of leakage in the membrane. So the ion is sequestered, but in the sensitive variety, those ions are released into cytosol, affecting the metabolic processes, whereas in the tolerant variety, they were to some lesser extent. I think this slide I will skip. And then we'll study the antioxidant enzymes also, and those profile you can see. The antioxidant enzymes was much higher in the tolerant variety. Actually, threshold level was higher. In the control itself is very high, and when you give salinity, it is not really increasing. It's just maintaining the much higher level. And in the sensitive variety, though it is increasing, but the level of increase is relatively less than the even threshold level of the tolerant variety. Same pattern was followed for CAT and APX. Then we also studied the expression level of the gene. And these are the genes related to this OS-HKT11, that is sodium uptake transporter. And so you see the sodium uptake is, the channel is increased much greater in the Jaya than in the core good. OS-HKT24 is a transporter for potassium over sodium. And here you see that potassium, the transporter is increased more in core good. And the third one is OSP5CS1, which is related to proline synthase. And the enzymes is working more in the tolerant one, but the accumulation we saw more in the sensitive one. And here we have also seen those antioxidant expression level of SOD and APX, and you can see quite a considerable increase in the antioxidant enzymes in the tolerant variety. This slide I will skip because this lipid also we have done, and protein profile also have done. The lipid doesn't change much, but the unsaturated to saturated ratio was higher in the tolerant variety. We did some proteomic study also, and these are some twofold increase and decrease and the protein which did not change much. And so identified proteins were 50, common protein in all these two plants were that number 248 and 341. Upregulate was much more in the tolerant variety, so were the downregulated protein. And those upregulations were mostly linear upregulated or linear downregulated, and only 4 protein were seen only in the 200 millimolar concentration. This is a PCA plot also to indicate that these two varieties has two different pattern, and these are our frequency distribution and all that thing. And I will just move on to our last slide. And the conclusion of this work is basically in 3 words. One is that these morphological parameters are providing plant-water relationship, okay, improving the plant-water relationship in the salt-tolerant variety, and which is the second one is the tolerant variety have a better ion homeostasis strategy that is also related to the morphology, the more trichomes and higher trichomes, number and size. So that might be sequestering, but we'll further study the sodium content of those trichomes also. That idea we get from the yesterday's lecture by Sabala. Okay, so that we'll do. So better ion homeostasis and better potassium and sodium ratio in the tolerant plant. And third is that they also have a better antioxidant ability. So to prevent the damage to membrane and thereby leakage to ion, and most of the physiological process continues. The only thing is that the productivity is about 1.8 ton per hectare for Corgut under salt condition, and under salt condition to Jaya is virtually no productivity. Of course, Jaya otherwise under normal condition gives us about 5 ton per hectare, where that the maximum it gives the Corgut is about 1.8 to 2 ton per hectare. With these words, I thank you for consideration.
Thank you, Dr. Prabhat Kumar. I think it is quite interesting. You have clearly demonstrated how this salt tolerant and susceptible rise at the level of physiology, biochemistry and molecular level, how they would differentiate. I think you have highlighted very clearly in case of Jaya, at 40 millimolar itself, it is accumulating quite a huge amount of sodium in its leaves. I think because of that, all those further consequences are happening. It could be one of the reasons. Since we have already overshot the time, if there is any specific question, maybe 1 or 2, we can pose on Dr. Prabhat Kumar.
Sir, you also did proteomic studies, right? Any specific markers we could see in sensitive variety and versus tolerant, so that you can manipulate those one in the future?
Yeah, looking into that, we started this proteomics works, and the story is very sad one, in the sense that we see a lot of proteomics studies going on in India, but wherever we approach for carrying out, we don't have the facility. Wherever we went to approach the people to get this work done, they just back off. So this work was done in NCL Pune with Professor Kardu's help. So we have just done one experiment, and that too during COVID times. So we have not been having that much interaction. This is the basic information derived from the basic data and analyzing the software. So we need to repeat that data. That's why I have not really emphasized much. I just shown that there are changes there. I mean, the number of protein which is induced and repressed are much larger in the Corgut. And some of those localization we have seen and expression levels. But to further analyze, we have to at least carry 3, 4, 5 times to come to some conclusion there.
Thank you. I think you can make many more theses from if you get more.
Yeah.
Right. Thank you.
Okay, thank you, sir. Thank you very much.
There is an announcement. Ms. Malini Bhattacharya is requested to report at the help desk immediately. I repeat, Ms. Malini Bhattacharya is requested to report at the help desk immediately. Thank you.
Yeah, with that, we will now come to the last presentation in this session. Dr. Rajesh Mehrotra, Professor, Birla Institute of Technology and Science.
Hello everyone, I am between your lunch and this is a very wrong time to be, but anyhow, I'll try to see. We have heard about promoters since yesterday in many of the talks. I've been trying to learn about the promoter architecture. We go in detail about the promoters, and I'll just browse through some of the experiments which we have done. And it doesn't work. So the general outline of my talk is why we have taken the ACGT element, and the pointer is there? No, it doesn't point. So why we have taken the ACGT element, and in silico analysis of ACGT repeat elements, and characterization of protein phosphatase 2C promoter. I just like to browse a bit, and characterization of recently we have done it in amino acid transmembrane protein and its promoter. So if you look at, the reason why we are interested in this ACGT core motif is, basically ACGT motif is important in multitude of responses. Since yesterday we have been listening about ABA response, we have been listening about the salicylic acid response. So if you look at these detailed architecture, how does a promoter respond to these things? So there is one of the core element today, Dr. Dinakar mentioned about the ABA, ABREs, and DRE. So I'll just go in a little bit detail about those. The ACGT element, for example, if they are in 2 copies present close by, they are actually being induced by the abscisic acid, when the spacer between them is 25 base pair. And when the spacer decreases to 25 base pair, to 5 base pair, the response becomes salicylic acid. So, and there are various, with recruiting and interacting with other factors and other cis-regulatory elements, they give various kinds of response. For example, the oxygen response, the neurobasis in G-box, jasmonic acid. So ACGT is a core element which is present in multitude of promoters. And so if you look at the promoter can classically be defined as, there are multiple cis-regulatory elements which are there. So, and the spacer between these cis-regulatory elements are critical determinants of their biological activity. And we have been trying to work and try to study the motif number, the recruitment of transcription factors at various cis-regulatory elements. So this was one of the articles which initially, when I just joined as a faculty, we started working, and we showed that when 2 ACGT elements are separated by a spacer of 5 base pair, it actually is induced by salicylic acid, while if you increase the distance between them, by 25 base pair, it is ABA inducible. So this was initial studies which we did, and this has a lot of meaning here, because when we say salicylic acid inducible promoter, it means that it is going to be induced by a biotic whenever there is an attack of the pathogens. It's going to be induced. When we say about ABA response, it means that it is normally, as we have learned this couple of days since yesterday, that ABA level tends to build up under stress conditions. So this is an ABA inducible promoter. So we have actually recently got some patent also of these. These all promoters are patent, which we have designed. And so going further, we try to study. I'm interested in the distribution, genomic distribution of these ACGT element across multiple of monocots and dicots. So we studied 2 monocots and 2 dicots. This is a stress condition for us. I just explain it because sometimes when you are starting off your career, you don't have enough money. So there are a lot of data available in public domains, which actually is lying without any purpose. So we exploited those data, which were actually reported by Professor Akhilesh Tyagi's group. So these data were actually lying ideal there. We extracted the promoter sequence of all those genes, almost 1 lakh. This study was done on more than 1 lakh genes, promoter. And then we studied 2 monocots and 2 dicot plants. And we fished up, what we found is that if you look at the spacer length on the x-axis and y-axis, we have the number of occurrences of the ACGT repeat. So what we identified is there are certain conservations, there are certain patterns in the occurrence of the ACGT repeat element in these genomes. Subsequently, we also compared 2 monocot plants, rice and sorghum, and also identified some regions of the peaks, and there's some common peaks. So we could identify. Also, we try to study the evolutionary pattern of these elements in the orthologous genes. All those genes, one of the important findings which we had is all those genes which were induced under stress have significant amount of ACGT repeats. And that was our curiosity. That was we tried to study how they have evolved. So across the organism, when we did the orthologous gene analysis, we identified that there's a parallel evolution of these ACGT elements across the genomes. Subsequently, this is the point I just mentioned. So there's a natural question which comes to our mind is if this element is there, what is the genomic distribution? If you look at any of the tetrameric sequence, by the law of probability, you can explain it very simply, because 1 by 4 to the power raised 4 should be the probability of this sequence to occur. But in a promoter region, which is 1000 base pair, most of studied people have tried to focus on those. So in 1000 base pair regions, if something is occurring in 14 copies or 15 copies, how do you explain that? There's too much of skewness in this particular. So this skewness was again a driving force for us to identify what is happening there. And so, but if you talk about the genomic distribution, we didn't find ACGT as a very high number. But when we talk about the promoters or genes which are induced under stress conditions, in them they were overrepresented. So this overrepresentation means something in biology. That's what we're trying to learn. And this entire set of work which is published in BMC Genomics is patterns and evolutions of ACGT repeat elements across 4 plant genomes. Further, moving from here, we try to study, see, this is a sequence I was trying to say. These are 1000 base pair sequences, if you see from here to here, and you see the repeat, which is occurring on multiple times ACGT element. How do you explain that? Because by the law of probability, you can just explain that this sequence should be 1 by 4 to the power raised 4, that is 1 by 256. So in a 1000 base pair region, it should be a maximum at 4. It should maximum have the 4 times repeat, but there is occurring in such a huge number. So this skewness, what is the purpose of this skewness? We're trying to learn that. This is one of the promoters which we studied. And when we started to study this promoter, so this was our model which we published previously. And we made multiple constructs. We made a deletion constructs. We removed a few of the ACGT elements. We made deletion constructs, developed transgenic plants for all these constructs. And we identified, there are multiple papers we published in this work. I'm just giving a very generic glimpse. So we removed one of the cassettes you can see here is actually AB-inducible. Now this was an interesting cassette for us. So, and when you compare with the full-length promoter, the induction was high. So we identified here a negative element which was present in the full-length promoter, and when deleted that, the induction went further high. So this work is published, so I'm not going to discuss more about that. And this is just the data of the, I think, do I have a few minutes more? 5 minutes more, okay, thank you. So, furthermore, when we went, see, we tried to study how is the MEGJ, what is the response in terms of MEGJ and in terms of the salicylic acid. We could see that we identified, this PP2C-like promoter, when various deletions were made, we identified that its deletion variants are induced by ABA, but not by MEGJ and salicylic acid. And subsequently, we studied another set of promoters. So we are looking for all the promoters where they are overrepresentation of ACGT element. So this is another set of promoters more recently we identified. Is the AT-AV60 promoter. This is a transporter, and this transporter is useful, very useful. I'll show through some of the experiments which we did. So when we did the tissue-specific expression analysis of this promoter, we took the various tissues. We could identify that in celiac the expression is high. We also identified this promoter to be induced, this particular gene is induced under the nitrogen stress condition. We also showed that this particular gene is induced under these are all real-time data, under drought stress. We also showed that this is induced under ABA in 10 hours. And we made various constructs. So you can see here, these are all repeats, are ACGT element repeats. So these are all ACGT repeats. So these were all ACGT repeats. So we keep on deleting until we had the 2 ACGT elements, because as per our understanding, when the 2 ACGT elements are there in close vicinity within a space of 25 to 30 base pair, this is AB-inducible. We went further to do the study of this. We made constructs. We made construct and tried to study the localization of this with the AT-AVD6D transporter with a GFP. We identified that this is actually with a marker lysine K4, which actually is going to the nuclear, is a membrane localized transporter. And further, we went ahead with this gene GFP co-localizes with the plasma membrane marker. Excuse me. We also did the yeast complementation assay and showed that this particular gene is involved in, you can see the data here, please see the last panel. You can see here, this actually confers the osmotolerance. So you can see AT-AVD6 enhanced osmotolerance in yeast mutant. We also did this study in the Xenopus laevis. And for this, during my experiment, my student got EMBA fellowship. She went to Professor Tony Miller's lab to do this experiment. And we could show in this experiment that there's an efflux of this aspartic acid into glutamic acid. We tried to study, and we monitored, and we identified, these were experimental protocols which we followed, and we showed that this AT-AVD6D is involved in the uptake of aspartic acid at pH 5.5. And now at the present stage, we have generated transgenic plants, and there is interesting observation we have. We are still to identify, we can see the overexpressed lines, these have small silics, but interestingly, the test weight of them is around 20%. So we are trying to understand the trade-off between the silic, why there is the decrease in this, but nature tends, we have a lot of discussion, nature tends to resist these changes. So with the overexpression lines, but interestingly, the grain weight has increased in this case. And the set of data which we generated here, we published in this year itself in Plant Molecular Biology, effect of ACGT element in the spatiotemporal regulation. And again, we went further to do the characterization of the promoter, because we are a promoter guy. So we don't want, of course, we would like to have genes as well, but I work on promoter. So we were interested in this promoter as well. And when we studied the promoter of it, you can see all these blues are basically the ACGT element. Again, the same question is, why is this skewness? And we try to decipher the skewness of this. And we did all kind of things. While we were doing this, we identified this particular promoter has more than 65% AT-rich region. And amplifying AT-rich region, which is more than 65%, is difficult. And in that case, we tried to learn about, and we optimized the PCR condition. It's a two-step PCR optimized by my student, Pinky. We published this protocol. And as of now, we have made all the deletion constructs of this particular promoter. And we have shown that these are AB-inducible. It is also induced in salt condition. We have given salt stress. So this, as of now, the latest standing of us is that one of the cassettes which we made basically is a Pro C2. This cassette is also now under patent. We have filed the patent of this cassette. So this is actually, we think, data suggests that this is an inducible promoter. The salt for, and this is my group. Myself, Dr. Sandhya, she is my wife as well, a faculty in BITS Pilani. This is Zeba, my student, she graduated last semester. Pinky graduated this semester. And Sneha is graduating on 7th of November. She has her final defense is there. And I have been thankful to, I received funding, not just so generously, but occasion. And many of the data, which I'm just suggesting for the younger students who are here, many of you can use the public domain data, which are actually lying idle, because a lot of horizontal information is there. But vertically, you can exploit those data to say what you want to say. So even if we don't have money, these dry lab experiments can be performed with less amount of money. So that's all for me. Thank you so much for, I'm sorry, I don't want to be between you and your lunch.
Dr. Rajesh, it is wonderful. It's a wonderful talk, in fact. Even at this lunch hour also, you are so energetic. We can understand you yourself has a very strong promoter, I think. If there is any one or two questions, quickly. Yeah, I think during the lunch time, I think we can have a discussion. Thank you, Dr. Rajesh.
Yeah, thank you.
So with that, I think we have come to the end of this session. So I'm very thankful to all the speakers. In fact, first two special lectures, they have done wonderfully well. And another 3 short lectures, they were also, in fact, very well. I must thank my co-chair and rapporteur for their sincere work. I also take this opportunity to thank organize for having given me this opportunity. Thank you, one and all. If there is anything.
Thank you, sir. As we have concluded the technical session 4, I would like to call upon Dr. C.C. Harilal to present the memento as a token of appreciation to Dr. Heber KB, chair of the session. Sir, please. Dr. Krishna Kumar ji, co-chair of the session. Dr. Shruti P., Session Rapporteur. Let us break for the lunch. Please enjoy the meal and kindly be back by sharp 2:10 p.m. Thank you.
Those who are waiting outside, I kindly request you to get in into the hall. Good afternoon, all. Welcome back to the International Conference on Physiological and Molecular Mechanisms for Abiotic Stress Tolerance in Plants, jointly organized by the Indian Society for Plant Physiology, South Zone, and the Department of Botany, University of Calicut. I would like to invite Dr. A. Yusuf, Professor, Department of Botany, Director, Inter-University Center for Biotechnology, University of Calicut, to coordinate the technical session 5. Sir, please.
Good afternoon, one and all. So we are entering into the 5th session of the International Conference and Workshop on Physiological and Molecular Mechanisms and Markers for Abiotic Stress Tolerance in Plants. And hearty welcome and good afternoon to all of you to this session. My duty is to introduce the chairman and co-chairman and rapporteur of this session, and also to introduce the speaker of the session. On the chair, it is Dr. Abida PS, Professor and Head Department of Plant Physiology, Kerala Agriculture University. She is an eminent scholar who is having 24 years of academic and research experience in Kerala Agriculture University. Her research areas are transcriptome and proteome analysis in abiotic and biotic stresses, molecular markers, immunodiagnostics, and computational analysis. She has published more than 45 international research papers in highly reputed journals, and she is actively participating in the research in the Kerala Agriculture University. And as the co-chair, we have Dr. Anu Agustin, Associate Professor of Department of Biotechnology and Microbiology, Kannur University. She is working on the salinity tolerance mechanisms in mangroves, and she is having in her name around more than 30 international publications. And she was a Boy Scouts Fellow and worked as a Fellow in Scotland for more than 2 years. And the rapporteur is Dr. Sinisha Eke. She was a student of Botany Department, University of Calicut. And presently she is an ad hoc faculty in the Sri Kerala Varma College, Thrissur. And welcome the chair, co-chair, and rapporteur to this section. And I have a great privilege to introduce the resource person today, Dr. Babu. Babu is delivering the P.S. Krishna Memorial Lecture in this session. And Dr. Babu is he's an alumni of this university, and he had done his post-graduation and PhD from this university, and later on he had moved to Center for Plant Biotechnology as a postdoctoral fellow, ICAR New Delhi, ICAR Pusa New Delhi, and then from there he had moved to US, and a very brief biodata of Dr. Babu is with me. I will read out it for you. Dr. Babu Valliodan, now he is as a professor of molecular genetics and genomics at Department of Agriculture and Environmental Sciences, Lincoln University, Missouri, USA, and he is the chair, LUHI, and director of Center for Climate Smart Commodities in Lincoln University. In addition, he is an adjunct faculty at the Division of Plant Sciences, University of Missouri, Columbia, MOUSA. Prior to his current position, he was a senior research scientist in genetics and genomics at National Center for Soybean Biotechnology and Division of Plant Sciences, University of Missouri. He completed, that is, I have told you before that he is an alumni of our university, and his research focuses are on the development and deployment of genetic and genomic tools for crop resilience, then climate smart and sustainable agriculture. He had coordinated large-scale genome sequencing projects in soybean and providing legume genome sequences, information, and halotype maps to the public breeding programs worldwide. Current research focuses of Dr. Babu's laboratory are applications of genetics and genomic techniques to study genome variations and complex trait improvements in legumes and major crops. This includes dissecting the molecular and biochemical mechanisms of stress-tolerant traits, carbon sequestration traits, studying the role of plant microbe interactions, and genome editing of water and nutrient use efficiency towards crop improvement. Dr. Babu is collaborating with various research institutions, including University of Western Australia, Nanjing Agriculture University, and our own university with our Inter-University Center for Plant Biotechnology, International Crop Research Institute of Semi-Arid Tropics, and Indian Council of Agriculture Research on Biotechnology and Genomic-Assisted Crop Improvement. He is serving as an editorial board member of several scientific journals, including Crop Sciences, Plants, Plant Physiology, and Frontiers in Genetics. He has published more than 80 peer-reviewed research publications, including journals in Nature, Nature Communications, Nature Scientific Data, and 12 book chapters to his credit. He has 2 patents in US patent applications pending, and he has achieved more than 12,000 citations, H-index of 41 and I-10 index of 70. With these credibilities, I invite Dr. Babu to deliver his lecture, or we can say that it's the P.S. Krishna Memorial Lecture. Welcome, Babu.
I don't know whether I keep my time. Let me pretend that I can keep some time. Good afternoon. Can I have that, what do you call, the technology to move the slides? What is it?
It was supposed to be here.
It's supposed to be there. It's okay.
It's not here.
If not, I have some in my...
Somebody must have taken off the last talk.
Maybe somebody's very much excited to take that with them, I think, yeah. Well, I have a pointer with me, but somebody should move the slides, okay? I have a pointer too.
The pointer is not going to work out on that.
Thanks, Om, for taking care of the technological glitch. Good afternoon, my friends. First of all, let me thank the organizers, especially Dr. Jose Putur, for inviting me to deliver this speech today. Above all, I'm very happy today standing in front of you because it's my homecoming. So I'm coming back to my home after about 20 plus years. And I'm very proud being a plant physiology trained plant biologist. And above all, again, I'm very much proud that my old lab is organizing this meeting. And hats off to Dr. Jose Putur and his team and all plant physiology and biochemistry faculties and students, and all the botany department faculties and students. Then my job here is to give Professor P.S. Krishnan Memorial Lecture. I'm very much honored, and it is a privilege to talk about my mentor, or the mentor of our plant physiology and biochemistry team of Calicut University. Once again, thanks to Dr. Jose Putur. And even before inviting me, we used to discuss about the organization, how we can make this program very successful. But he did a great job. And thank you very much, and congratulations. With this words, let me go to my job here. The title of my talk is Genomic Assisted Improvement. So, as Yusuf mentioned about my introduction, my lab is mainly focusing on developing genomic resources in legumes, mainly soybeans. Then I'm using those genomic resources to interrogate the abiotic stress and find out some resilience formulations through different identifying the molecular mechanisms and application of that tools to find out the solutions from the stress tolerance mechanisms. So before going to my talk, as I told you that I'm here only because of Dr. Krishnan and his influence on my research. My PhD was on winged bean. So when I came here after my master's, when I came to the biochemistry lab, so then they asked me, then what do you want to work on? We have two options here. One is winged bean, another one is banana. I thought, okay, maybe I never seen winged bean in my life that time. So then Dr. Nandagumar and Dr. Pillai Madhu, he told me that this is Dr. Krishnan sir's instruction that the first student who comes here should work on winged bean. That's how I started physiology, genetic stabilization of winged bean. But that was a great start. Later I found out that was a great start. I could say my peers over there in the United States that yes, I have trained in legumes before coming over there. So Professor P.S. Krishnan, I got these pictures and everything from our alumni, plant physiology alumni. Dr. Khalil made all this. I copied from his essays about Dr. Krishnan. Professor Krishnan, full name is Dr. Padmanabhan Subramanian Krishnan, born in 1940. And from all of us, we can proudly say that he is an effective teacher and mentor, dedicated and successful researcher, and he published several of high quality papers in the biochemistry journal, mainly biochemistry journal, and a couple of the plant physiology related journals. And above all, he's a compassionate human being. I still remember that. And during the end of my PhD time, I used to go to Trivandrum. He was living in a geriatric and aftercare home over there. That's his family over there. So I used to go with the bundle of paper to him to correct. And he always liked some specific banana. So I used to buy that banana and go to his place. Now I know that why he asked the other student to do the PhD in banana. But he sit in front of me with that age, and we go by each and every line of my thesis, and he gave valuable suggestions, and somehow I'm rescued by him. So I'll give you a brief bio. He got a BA in physics in 1934 from Kerala, and did his two masters from Madras Presidency, then worked as a research assistant, and then British Indian government provided some scholarship to go to Cornell, and he did his graduation PhD with James Sumner. J.B. Sumner was the first Nobel laureate in enzyme biochemistry. So you can see J.B. Sumner, and he's working there. He got a PhD in 1947, and his first publication in Archives in Biochemistry in 1948. He worked on eparase enzymes. After his PhD, he came back to National Chemical Lab. Then he became a scientist over there. Then moved to Lucknow as an assistant professor in career. Then he retired from there as the dean of that biochemistry department. But even after retirement, he continued to guide and help students and researchers, including people like me. He came here as a professor of emeritus in biochemistry because Dr. Nandagumar, he is our biochemistry professor at Calicut University. He passed away in 1997. He was such a great human being. I've seen him always in white and white. I mean, a normal white and white cotton dress, and he eat mostly fruits. That's what he do, and he always spend a lot of time at the botanical garden doing a lot of things. Sometimes he'll show some phenomenon and say that why don't we find out some phosphatase enzyme or phosphorus enzyme. And that's such a motivation we got from him. So I got this photo also from our plant physiology alumni. You can see the eminent people who came out from my lab. I'm very proudly I can say that, my lab. And thanks to Jose to making it as internationally known again after that time. So you can see, I don't know all of them, but I know most of them in this group. So let me come to my topic. So I normally show this slide first when I start my talk. So what we need, we all know that we need more food. A lot of people are they're hungry around the world. So as a scientist, as a plant biologist, you're a plant physiologist or a technologist or whoever it may be, it's your responsibility to provide food to them. So that's sort of how I see that. So what are the major challenges? Major challenges are, first of all, is this population burst. A lot of statistics say that we get double or triple the population by the end of the century, and we don't have enough food for them, and also the nutritive value of the food is going to go down. And another problem is the climate change. So one side, the population increase, the other side, the climate change. This cartoon, this study shows that by 2080 or 2050 or 2090, you'll lose more than 57% of the crop yield. So, but Dr. Om did a nice presentation at Kannur University, giving more elaborated view of how much and when and where all this is going to lose. But I want to show this thing, why we should be very serious on looking at the specific problem to contribute towards the hunger or the food shortage in the world. So one is the population increase, another one is climate change. So all this says that, this is an old slide, but still it's very valid. And this projection says that the farmers should produce double. So we have to ask the farmers that you guys go and make more food. But how they can do that? The study says that we need more than 2.5% increase of yield for most of the major crops. But yeah, farmers are ready to do that. They need better varieties, better plants. So again, we are the one to give those better varieties. The major challenge, how we can efficiently identify and utilize the desired genes, alleles and markers for crop improvement program, especially identifying mechanism of traits or functions. As Sergey and Dr. Shetty mentioned yesterday, that yes, we have to look at the physiological basis of and each phenomenon and use this new biology techniques or biology tools to get that one interrogated. Then translate that untapped information and give that to the farmers, yes, produce this food. For that, this is not a kind of a rocket science kind of the people started thinking about from early. If you look at the Charles Darwin, it says that where you have to look at the variation, natural population, or you have to look at the environment, how we can look at the environment and help on the growth. The same with if you look at the DeVries also, he said that we need to look at the mutation, or we need improved races or greater value, and in the beauty, I would say that productivity. And we need, this is what always, in 1909, he told that we need improved species. So it is already there. So as a scientist, we have to dig into that and help the world. Since I am in the Midwest, and this is our cash crop over in soybean, and above all, it is high, you all know that the nutrition value is high, I mean, the farmers love it. They get a lot of money. Now the price is almost $11 or $12 per bushel in the United States. When I started working on soybean, it was $3 or something like that. Now it's $11 a bushel. The reason there, because we got very improved varieties, and that farmers love them. So you can see that one of the uses of the soy proteins, like how much it's needed for as a feed. You know, you all know that soybean can be used for as a fiber, feed, or fuel. So I'm giving one of the cartoon over there how much soybean is going to our diet through these animals or as a vegetable protein. So soybean is a model crop to study in the case of legume. But the problem with soybean is that narrow genetic basis. So in our USDA soybean germplasm collections, we got about 19,000 soybean. But these are all, you can trace back its pedigree. You can see only 15 as a founder lines. So what it means that you got only that genetic space to improve this crop. This narrow genetic basis is a major bottleneck, and we have to find the novel genetic combination that increase yield potential. So when we look at the crop yield, what is the crop yield? How you say the crop yield? Crop yield is water use efficiency, and harvest index. So, I mean, I was thinking about putting these two together, water capture and water use efficiency together as a one box. I mean, but I thought it would be good for the student to see kind of a separate. So my lab is mainly focusing on the hidden half of the plant, that roots, the deep roots, root angle, root elongation, root length density, nitrogen fixation. All those help those traits for water use efficiency or water capture. And as a push-pull mechanism, we look at the stomatal control, slow canopy wilting, and its role on the root system architecture and water use efficiency. Then harvest index traits, you can see reduced flower abortion. Yesterday, the photosynthesis folks were talking about all this traits, especially Dr. Gomadi was talking about the heat-induced pollen abortion and the yield. The carbon partitioning, photosynthesis, and seed number, seed size, all those the traits, we had to look at it. So this review is one of the historical review in advanced agronomy. It says that drought resistance is yield in production agriculture. So that's what your drought resistance, that we should be looking to that. So when you go to the root system architecture, yes, it exists in soybean. You can see the natural variation of root system exists in soybean. You can see there's a different accessions. We just grow at the seedling stage and look at the root structure. So we focused on water uptake, water use efficiency, nutrient uptake, storage, nitrogen fixation, performance, interaction with the rhizosphere. This all contribute through the root. And these are some cartoons kind of depicting some of the root. Again, here, people started talking about root early in 1800. I mean, like, so the earlier paper, they say that, see, where the radicle grows, it grows towards the earth, the gravitropism, and also, what are the different types of root, fibrous roots, or what are the difference between the fibrous root and main root? I mean, people started looking at the root long time back, if we go back, and this paper is from 1892 from Torrey Botanical Club. And so this all gives that, yes, we have to focus on root to get more information towards the stress mitigation and more yield. But still, our breeders are kind of, they don't know much about the root system. They just stand in front of the plant, what to do with it. We get some wilting and we lose less yield, and some plants don't grow well. So they need more information about the roots. So as a root lab, I mean, part of the root lab, it's our job to provide this information to them. So we are focusing some of the major traits in soybean roots. So we look at the cellular level, like its cell shape, and also anatomical level. Yesterday, Sergey and others were talking, Dr. Parthasarathi also talking about we have to go into the cellular level physiological mechanisms to identify the exact reasons and exact solutions. So these are the traits what we are looking at in the lab. And for that you need a root phenotyping platform. So it's not very easy. You know that, I mean, the shoot can have some combat easy. You can go walk towards it and you can put your Lycor and you can get your reading. Root, it's a little bit difficult. So, but we developed a cost-effective programs. So we do seedling stage root evaluation. We grow different varieties, then find out how it grows and what is the difference. Is there a better one, we can take it. Then we use WinRISO, a program. That's a common program. It's a Canadian company who make that, and we scan it, and we get those. We can get about 70 traits out of that scanning, like root thickness, root angle, root length, and all kind of stuff. Then we do kind of a vegetative stage root screening. We use about 2 to 3 week old seedling using the cones we grow in the greenhouse in controlled condition. Then we take that out. You can see that how we take it out. And we use, again, I mean, some kind of an old school method. It's like taking length and kind of stuff, but still we do the WinRISOs with them. Then reproductive stage, we do a little bit more next level because we want to see in every stage how it is there, you know. So then we do a kind of a 1 meter old tube. That's the same method what the rice group in Japanese rice group did about 50 years, 40 years back. We were using the same one. We put take the tube, put it kind of a little bit slanting, and we grow the atom. Then we collect the root. You can see the variation on the root system. We tried this thing. When we have to go a little bit more modernize our phenotyping system, but we failed. Even though we published the paper, though, but it is not high throughput at all. But we can share that, yes, we worked with the engineering department, and we told them that this is root. We want to get this picture. They told, yeah, why don't we grow it in the media and we can get a good picture out of it. And that's what they did. But it was, but it wasn't just an academic exercise. We got a paper in Plant Phenome Journal. But this is not high throughput at all. This is the best one. Shovelomics. Take the shovel, go to the field, dig it, take it out. I mean, it takes some time. And some of my students, my root team students, are they like it. That's a very good workout for them. They can make 6 packs by doing this thing in the field season. So this is how we screen our root system. We dig it and take it. So we dig it and we kind of score like this, giving some numbers based on the visual scoring. And at the same time, we do a little bit correlation studies with other published works too. So the same way we do the root angle. The root angle, what we did is, and we bought a big protractor, like what we used in a math class a long time back, the box you remember. So we used a big one, then we used that one for a root angle measurement. So this is one of our key traits we are looking at in our lab, the root angle. So we try to work with Chris Top and other experts in roots. So they have some program called the DIRT. It's a publicly available program. So they just photograph you put your root, hang the root, and take the 360-degree photographs and put all the photographs together and get your information. But still, high throughput phenotyping is missing in the root system studies. So we have to look at the field level. Sometimes we have the stress levels and all kind of a high throughput method is still missing in the case, kind of a mature plant. So we have to work on that one. So normally what we do is that we do at the different locations to find out the genotype environmental interaction and different soil type. And also these are the major soybean growing region of the state of Missouri. So we have the farmers field there and also university facilities we go there. So that's about the phenotyping side. So we have to have, when you do study, you have a phenotyping. So how you study the phenotype, how you query with your genetics. So we need some genetic tools or that time when I started about 15 years back on this, so we had only some unfinished maps, linkage map or some other small bits and pieces of the ESTs. And kind of the genome information was very, very scarce at that time. Then 2010, we published the first legume genome, and that helped a lot of our legume scientists to go a little bit deeper because of a complete more than 90% genome, so that we can use that for our genetic studies. So after the legume genome, single genome, we thought that why don't we sequence the lines what we use for our crossing. When we're doing our QTL mapping, because without QTL mapping, you cannot continue your trait discovery. So we used those parents and some of the wild soybean, and we sequenced them like about 15x genome coverage. No, we didn't do a kind of 100x, we do 15x genome coverage, and we found about 8 million SNPs in the wild soybeans, and about 7 million SNPs in land races, and 7 million on the elite. So you all know that SNPs are single nucleotide polymorphism. When you compare all these 50 sequences together, and what is the difference between one sequence to another sequence? If it is A in position 225, that will be a B or a G or T or C in position number 25 in other people, other organism or other germplasm. So that's we call the single nucleotide polymorphism. That's a very valid mutation matrix, what we need to look at the genotype and phenotype association. So at the same time, we use this genome information in our QTL studies. So here we cross a cultivated species, that is V71, with a soja. I mean, soja is a wild soybean, to get more alleles from soja to our cultivated. Yesterday, Sergey, Dr. Parthasarathi, and all are mentioning about that. During our domestication, we lost a lot of our beneficial genes. What happened is that we were selecting only for yield. You grow this year, select the best one for the yield. Next year, best one for the yield. What we are losing? You're losing your disease-resistant genes, excuse me, or stress-tolerant genes. So we have to get those genes back, and we have to improve the diversity and so therefore that the soja material or wild soybean is the best untapped reservoir for all those SNPs. So we identified some QTLs using the genetic sequence and the associated the QTLs. We found QTL 6 and chromosome 6 and 7 for surface area and root length gene. That means we got some allele from soja to the cultivated type. So when we interrogated a little bit more, we found a specific gene, like for example, NED activating complex, that gene having very potential role in the root system phenotype. That can be a best candidate for a marker in the future breeding program. So we did the same way with another population. We developed another population with the sequenced genome, William 82, another sequenced soja genome, that's PI. PI means plant introduction, 483, 460B. That's most diverse soja or wild soybean available in our germplasm collection, and we made the RILs. But here, we did a little bit different technique. What we did is that all the RILs, we sequenced them in a kind of a 0.2x genome equivalent. That's we call kind of a skim sequencing. So either full genome sequence, then you have 15x to 20x kind of a partial genome sequence, and we get essential elements using a skim sequencing. We use that skim sequence technology to identify the QTL, and we identified several genomic regions associated with the root system architecture. So if we summarize that, by 2017 we published this one, we identified all these genomic regions with some indications to root system architecture targets for marker development in the breeding program. At the same time, we want to look at a little bit more inside the tissue level. So it's a combination. We cannot go only with a QTL mapping or sequencing. Look at it with QTL. So Bob Sharp is an eminent physiologist in our department he developed a root seedling system for maize. So we adapted that system, and you can see the hypothesis here it is that the red color is the stress treated or drought stressed, and this is the well watered root tip. So you grow the root in a controlled environment with a specific water potential, and you can see that region 1 is kind of it's elongating region, it's maintaining its elongation. Region 2, it is here it is a kind of a reduction in elongation, and region 3, there is no such kind of a elongation there. So we divided this into three major regions, and we looked at the growth rate of the root system, and we found that 0 to 4 millimeter of the root tip is kind of elongation region, 4 to 8 millimeter for the deceleration region, and 8 to 15 as a region 3. So, yesterday, Sergey was talking about cutting the root tip. So we were doing this kind of based on the root growth rate. We identified these regions. Then we used this region to study the protein differences. We used a proteomics approach, and we used a transcriptomic approach to find out what are the genes affected with or affecting or controlling the water stress. So this is kind of an earlier data we did in 2010 when the genome came. So kind of all those kind of work, it gives us that we have about 4 proteins upregulated in region 1 and about 9 in the region 2 associated with the water stress tolerance. And we did some, there was some kind of isoflavonoid synthesis pathway genes and lignin biosynthesis genes. We did some functional studies, and after that, we found some cell wall loosening genes might have some help in this water mitigation, and we tried that as a transgenic. But yes, we got a good transgenic first year, and next year, no, we couldn't find a stable transgene on that one, even though we got a publication out of that one, though. That's we can say that. So the same way the protein, we looked at the gene expression profile and see how this gene atlas of these specific regions are regulating this. And we found a lot of ABA-related and GA-related genes having specific role, and we tested them. We go a little bit more deeper, and we found what are the region-specific and stress-specific genes. Then we isolated the genes, and we looked at how these genes are interacting with other genes. Is there any specific promoter or a specific a common hub gene? So instead of taking for 3 or 4 genes, why don't you focus on that hub gene and find out if that gene is the one responsible for elongation even during the water stress? So we found a couple of targets, and we did a couple of them expressed in Arabidopsis. Me and my former lab, we are very shy on Arabidopsis, but we used Arabidopsis in this case to have a quick data out. So we took a couple of the GA biosynthesis pathway genes, and we found a significant phenotype. Or when we overexpress of them, we can find more primary root length, more total root length, and root tip number. This is with the Arabidopsis. So we want to see this in the soybean by itself, and we are trying to use the CRISPR-Cas9 and see is there any way we can do functionally analyze this phenotypes. Then we go a little bit deeper into that. What we did that there is a population called a NAM population, nested association mapping population, for developing QTL for yield and QTL for other characteristics. So we thought about looking at a little bit more in the anatomical side. How is there any difference in the cell structure or cell shape? And we did the microscopic sections, and we used the genome sequence information to associate this phenotype with the genome matrix to identify the genes associated with the cell structure differences, giving some guidance towards the water mitigation. So we found that root xylem plasticity can improve water use and yield in water-stressed soybean. Yield, still I use the word yield as a loose because we didn't test that yield yet, but that's our goal to look at into that. Yes, the root plasticity, one of the candidate is showing a little bit kind of improvement on this trait. So the kind of almost same with different patterns what we use for mapping. So when we did that experiment, we got limited information on the genome. By that time, we were sequencing more genomes, like more 418. Before that, we got only about 106 genomes to query with. Now, then this time we are doing the 480 most diverse genome. That means out of the 1900 soybean genotype available at the United States Soybean Germplasm Center, we selected 1400 lines as most diverse using diversity analysis. And then from that 1400, we took the first most diverse 480, sequenced, and we used that for our studies. And we used the same anatomical data, what we had from the 256 lines, and we looked for the SNP variant in those lines. Then definitely, I mean, it can give the more precise QTL information. We can go very close to that specific genes or genomic regions. So one of the genes is kind of, it's a cell wall related gene that we seen in earlier case too. So we did a little bit more deeper, and we looked at that. Okay, cell wall loosening genes having any role on that PIs or accession, and we found this allelic difference in those specific accessions. But still, it's still a black box for us. Even though we are studying, we are trying to investigate why these 15 accessions have allele to serine or allele to serine A to C in 11 accessions different, and why the lateral root number or lateral root, primary root length is different, and is there any ABA and auxin-related genes are influencing all those things? It's still questions for the future to work on. So we did some kind of a quick and dirty studies, like we tried the transgenic hairy root assay. That is a most easy one. Like you take a soybean plant, cut its root and throw it away, and use that stem, dip it in some agrobacterium that can make more roots. But that agrobacterium having your gene construct, so one is infected, you get some transgenic root, and use that transgenic root for your assays. So that's what we studied. When we looked at those specific candidate, we found more positive results on the root system development. So that's our goal is to, okay, this GLR gene having a role on the improved root system that can be used for enhanced water use efficiency, or can be used as a marker, or you can use as kind of molecular breeding programs. So this is the gene I was talking about. So that's a cell wall loosening gene we're trying to overexpress. By that time, our earlier microarray studies found this XTH31, that's also cell wall loosening gene. That's what the story we're talking about. We expressed that in soybean, did some field trial. First year, wonderful. Second year also, okay, we got some results. But no, that's not, we found later that it was not genetically stable. But that's a good indication that, yes, the cell wall loosening genes have some role in the water use efficiency traits. So then we get more sequencing done. So recently we did one of the Chinese line, that is W05, that's most diverse Chinese line. So from US we sequenced the most diverse US line, and the Chinese line we sequenced. So we got more information about the untapped genetic information. So wild soybeans or wild crop relatives are the best measurement for you to get more information to help plant mitigation to stress. Again, what we did sequenced more sophisticated way to get more genetic information to put this in the pool, so we can use that for precise identification of genomic studies. So this is what I was mentioning about. We used that for our root studies earlier. So we used this, the 4 million SNP from cultivated soybean, or total about 8 million SNP to query that genomic region to find out the specific mutations associated with that lateral root number or the root length. But at the same time, we collaborated with the Canadians. The Canadians came up with a haplotype map with all the Canadian germplasm. You know that soybean have 10 or 12 maturity groups, because if you did the 0, 0, 0, 0, 0, 3, 1, and 2, the earlier maturity groups grows in Canada, Minnesota, that area. And in my place, we grow maturity group 4, 3, and 4. And if you go to Brazil, grow 5, 7, 8. In India, I think kind of a late maturity group we grow here. And as you all know, the Brazil is the #1 producer of soybean. So US is #2, and India is #5. And there's a lot of potential for India to improve soybean. So we use this haplotype map also to look at more precise information. We want to go to that specific allele. We can tell the breeder that use this allele for your breeding program. So the new tools are coming in. So this is kind of a train, as I was telling in there in Kannur University, that if we miss the train, we'll be far behind in that area of investigations. So what we did is that, why don't we put all these 1100 genome together and find out only the key dispensable genome. That means that's the key mutation. We put all the 1100 together and mix it together and find out the difference between 1 to 1 to 1 to, like between each of them with all the 1100. That's called a pangenome. Develop a pangenome. Use this pangenome metric. Look, in my case, root system architecture traits. That is still going on. All these 1100s are coming from the different parts of the world. So that's very important information needed to look at the root system. We found that some of the region-specific root system architecture differences. So in this pangenome, what we found, as you mentioned, that we found about 13% of genes are useful to study this variations, because that's called variable genes. Excuse me. And at the same time, we found that 583 genes show presence or absence variation in wild accessions but are fixed in domesticated lines. That's exactly yesterday Dr. Parthasarathi and Sergey was mentioning about that, how were these genes gone and how its genes gone. So we look into that so we can get some answers to in the case of soybean. So these are kind of a lot of studies looking at how the reduction of diversity can be identified using this SNP matrix from the pangenome. So again, we have that lines. The students and we all dig these lines from the ground, and you can see the natural genetic variation of those lines. These are the different accessions. We gave the code. That's the code for the sequencing company to sequence those genome. So then the number is based on the funding, where we get the money to sequence those ones based on that. So this is some of the lines in our maturity group 1 to 8, what we selected and did the studies. Then we used that pangenome matrix for the root system architecture. What are the data what we collected from all this major 4 or 5 traits? We looked at it. I'm giving one example here. That is root angle, and we found this key gene. Look at its probability. Its probability is very high, so this will be a candidate. This is the candidate we are working on now to look at whether it's having any role on the root angle. So we hope, because we used that much sophisticated data, and from the looking at the cellular level phenotype, then we are not overconfident. I'm thinking that this will be a good lead towards the root system architecture or root angle gene, the same way with the primary root length, the genes over here. So the functional analysis of this gene is kind of ongoing in the lab. So I think that's the root story, what we have it now. So we are still going on. So you can see that evolution of the technologies used to investigate a specific trait. So starting from some EST sequence based or RFLP marker to some half genome marker. Then we have kind of a full genome came to see the marker. Then we have more resequencing data, then to pan-genome. But we thought that that's enough. No, that's not enough at all. I mean, for the root system studies, we need more data. So everybody should read this, this review. This is one of the best reviews I read recently by Su's group from China, that says that we have to put all this tool together. I mean, we are querying a specific physiological mechanism, collect maximum data. Whatever you can get, maybe you count the soil there. I mean, look at the color of the soil data. So you can see that, maybe in my next slide I can see that. So you collect all the data, the soil parameters and the climate, even the wind and the shade, whatever the on the plant. We have to collect all the data, use artificial intelligence and machine learning technologies to put those things, because now this is very handy now, because you know that in the United States, most of the tractors are kind of ultra sophisticated artificial intelligence tractors. They have all the sensors and every kind of stuff. So use those data for querying this, and look more on the genotype, phenotype, and environment. Then make models. They look at the interaction between genotype, GPE model. Then look for the breeding pipeline. Then add the new breeding technology. So you can see that from this, all this, how we put it there, level 1, level 2, 3, and 4. So you can see that level 1 and 2, you can have the data, make models. Level 3 and 4, you have the breeding pipeline, and we can make the breeding decisions, that whether you have to use a mass or use CRISPR-Cas9 based breeding, or you need a speed breeding, or any kind of a breeding technologies, how we can assemble. We can make some breeding design for that with a lot of data. That's the key, is a lot of data. So for example, data. So data from climate, different types of data. But this is a wonderful review I read, had that. So then soil, crop canopy, crop management, and even your rhizobiome. Like how your bacterial or fungal species around this root help to grow this plant, or help to mitigate the stress, both biotic or abiotic stress at the cellular level, and how it's affecting our physiological status of the plant. So you have to collect the environment data, genotype data, phenotype data, and use them and make the models and develop the breeding decisions. So that's the way to go. So what's the path ahead? So we need genetically enhanced and genetically diverse germplasm, because in the case of soybean, I told about the narrow genetic diversity, so we have to put more diversity in. Then the genotyping is easy. I can just sequence my genome with about $500 now. So it's not a big deal. That's what I did. Or that's what he did. Dr. Ormus did. That's easy. But phenotyping, that's the toughest part. It's expensive and laborious, and how we can have a high throughput phenotyping strategies. And we had to develop a smart breeding pipeline with the big data and the artificial intelligence and all the tools, what we are talking about, available tools, engineering tools or biological tools. What are the tools available? We have to investigate thoroughly to get the root of that problem in the case of root system architecture. Then we have integrated breeding technologies, all speed breeding or genome editing or smart breeding program. So that's it. And at present, my lab, the Lincoln University lab, is over there. And the first 4 are the graduate students. The next 4 are the undergrad students. And Chris is my field superintendent. He is kind of doing his 25% job on helping the root phenotyping and the drought phenotyping in the field. And I got a lot of collaborators. My former lab, I would work with the Wen lab. And Dong Shu is a very good protein and bioinformaticians. And we work very heavily with Dave Edwards at the University of Western Australia and Philip Bayer. They are the pan-genome gurus. They made the pan-genome for us. So, and we work with them to get query the traits to get those data out. Hongming Lam and the Tingfu Chan, they are also they're working on that. Hongming working heavily on soybean. Tingfu is a very good genome assembler there. And Dr. Juni Gai is a guru of soybean research in China. So he gave a lot of leads on the wild soybean. And the Canadian group, Franzo and Dawu on the haplotype. Ricardo Albanor is in the South America, so we can get some South American jam blossom. We published a paper about 30 Brazilian genotype sequenced to find out the genome variability. Jeremy helped us to sequence some of the genome. And we work very closely with the USDA ARS, like our ICAR scientists. And we have a legume federation team, and we keep all the data at the soy base. So if you go to soy base, you can get all the data what I mentioned here, and even the root data. We are going to put all the root data there, and all the root system data, even the anatomic data we are putting there. So we got money from USDA and the United Soybean Board, and some of the corporates also gave us money because they also need the sequences. So Corteva, Bayer, and they gave a sequence. Pacific Bioscience, they do the long read sequencing, and they helped us to sequence a lot of them with a reduced price. And that's it. Yeah, like yesterday, I'm talking about the diversity in the lab. Yes, my lab, my lab is a very young lab and recently started. It's only about close to 2.5 years now. So I got the people from Nigeria, China, Nepal, United States, Caribbean islands, and myself from India. And I'm getting one student from Punjab Agriculture National this year, and another student from Nepal also joining by the next fall. That's my team. Thank you very much for listening it. If you have any questions, please.
So thank you, Dr. Babu. That was very informative and very elaborate talk. And in the context of climate change and the quest for more food, the powerful tools of phenomics and genomics assisted breeding will be definitely a leap in crop improvement program. And exposure to recent tools and programs for phenotyping and genotyping platforms where expose identification of markers or landmarks to locate genes for water mining traits using most emerging technologies for smart breeding pipelines was also very informative. And with these few words, I invite a few questions from the audience as we are running short of time.
I'm sorry that I took, I'm 15 minutes more, I think, right? So more than, I'm sorry about that, but I took that. Students, I need some questions from students. Can I? Okay. Please, go ahead.
Babu, that was wonderful talk. Really, a lot of data, a lot of ideas. So my question is always translating into yield and output. So with the root architects, this root density and angles. So you grow these crops, these soybeans in the field. So did you see the difference of the root density and root angles is translated into, I know it's related to drought, water use efficiency, but did that, you get the data for yield?
So we collected the data, the 100 seed weight data there, but a lot of variations over there. So still we are looking a little bit more deeper because we need several years and several locations. So it's only we did about 2 years, the second year this year. So hopefully we can get more data on that one. Yeah, that would be a good indication to look into that, okay, this is the one having direct relationship with the yield. So whatever the molecular changes you want to make it, you can get some results. Yeah. Thank you very much for that.
Thank you.
Sir, very good presentation on this phenotyping and how we can relate the phenotype and genotype. My little query regarding that, just a question on adapted seedling system, whether can you elaborate, because whether that we are doing only for drought or we are trying for other stresses also.
You know, I'm doing that for mainly, that's for drought, because I can easily measure the water potential, and I can kind of have a good idea about a uniform water potential. I mean, you know, the root is a very difficult thing, because if you go kind of a 10 centimeter, I mean, if you put in a 20 centimeter tube, the water potential will be different. So you're not getting the same stress level if you're using drought studies over there. So the system what we developed and using longer probes, we can have different levels of stress. That we are using that system mainly for drought, but I know some of my colleagues is using that for salinity studies too. But
sir, one more, the reproductive stage tolerance for that one. Whether we are doing the, means you may be doing in the phenotyping and lysimetric kind of means structures are there, or simply?
No, we do the classical plant physiology traits. We use our Lycor machine, we got the field, we get all the maximum data, and also we get, now we are using a drone. So we have the infrared cameras on the drone, and then fly them and get maximum data. That's in canopy structure and IR reflectance, and all the data we are collecting with.
So very, very congratulations for your nice talk, excellent presentation. Thank you, sir.
Thank you. I don't know why students doesn't have any questions though.
So I just have one doubt. You mentioned at one place that during the domestication we are losing some important genes, and the breeders are focusing on the yield. So as a common man, like I am not from the plant guy, but as agriculture, like the main focus is the yield. If yield, you are getting the very good yield, we don't care about anything others. And overall, if everything is good, then you will have the yield, good yield. That my belief. So how that thing matters, and if you have lose this, is there any way to get back those genes?
So that's what I mean, like these investigations help us to find out that where it is lost, when it is lost, how it is lost. So I mean, soybean is kind of, it's a whiny plant, you know. Now soybean is again upright. How did you add that? You did the breeding selection for standing up. That's it, right? And also at the same time you do some yield. But you do continuous selection for this thing, we lose a lot of acquired, essential traits that we go along with it, like water use or water relation traits, disease resistant, a lot of disease, pest resistant, nematode. Why the nematode infections more higher in soybean? We lost that thing. If you look at the 60 years of breeding history in soybean in the United States, then can we put it back? Yes, that's why we use this wild, untapped genetic material, the wild soybean. Take them, use that for in your breeding, or breeding designs, the alleles from the wild type.
It's a very good presentation, Dr. Babu. Yeah, my query is about the QTLs. You said QTLs, right? Identified QTLs for different root traits. So what short of QTLs that you identified, any particular root trait specific for root angle or root?
Yeah. So whatever we did with, we use the same material later we got. The major focus on, we focus mainly on the root angle, root length, root diameter, lateral root number.
Any major QTL like in chickpea?
Yes, chickpea got a wonderful result on the root system, and we got that chromosome number 6 and 8, whatever I've been shown that, but we are kind of walking down inside using this data set and find out, hopefully, you see that my GWAS, I didn't see any gene on 6. Yeah. You see, I got the chromosome number 2 there, another one is somewhere on the chromosome 18 or 16. So we have to sit with more data and see what is that specific allele sit there, you know. I'm not telling that whatever the QTL we studied earlier is obsolete. No, there are some obsolete, because when we sequence more genome, we can find, for example, soybean genome. We look at soybean genome version 1 annotation. Look at the version 2 annotation. You can see a lot of genes not present in version 2. And the one what we published in 2019, we lost, we didn't see a lot of genes, because that's kind of the problem with the sequencing and assembly. So what happens is that we try to relate our genome sequence with this QTL and say that that's the right one, you know. So what I found out that my new genome sequence, I couldn't find a lot of the QTLs identified earlier on the oil protein, especially oil protein QTL, except the QTL20, what for the high protein.
Yeah, one more query.
Yeah, you can shout at me.
Yeah, you have studied about the structural traits of the roots. What about the functional traits like transpiration efficiency or water use efficiency? Or
no, I didn't.
No idea.
We didn't look at. I have no idea about that one. So we have expert here. Yes, sir.
In fact, it is a very wonderful talk. After listening to you, one question came to my mind that a lot of people work also on above ground as far as drought tolerance is concerned. Now, if I ask you, how much would be the contribution from below ground and how much is from the above ground? Or if I ask indirectly, if there is a brain in the plant, where it would be?
That's a good question. Now I got another problem to solve now. So that's what. Truly speaking, I don't have much answer. But being a root scientist, I would say that all the signals from the root. That's what I would say. So the hidden half is the brain. I mean, it sends water, it sends nutrients, and it sends who's next to that root. Then it gives that, okay, you grow like this, and this, if you want more yield, I can give this much water. If I shut water, you cannot give that yield. Yes, I think roots are the brain. Yes, I'm from.
Root wilt.
Yes.
They call it as root wilt, but it is happening on the top. Because of the top, root is getting wilt. So.
Yes, but maybe I'll pass that. Maybe I won't take that comment back though. What I made. I don't know, but maybe some kind of a pathogen or a planned biotic interaction, something. I don't know. Yeah, but for the sake of answer, when I'm thinking it, yes, plant root decide that how it go. I mean, what to take or not. And by example, what yesterday's talk, Dr. Ohms, I'm like, root absorb that, right? The arsenic or thing. Root decide that, yes, we do that thing more. Signals also come from up and down to you there.
Sir?
Yes.
Hello. Yeah. About the nodal formation, so legumes have most interactions with the biotic factors as well. So won't that affect the root architecture as well?
No, that's what we are looking at now. What we are doing that's a very new, because I'm kind of a what do you call stress? Abiotic. Stress guy, and this new rhizobiome studies are very new. And fortunately, one student have some microbiology background, kind of. So we have recently started looking at those rhizobiome. We don't have any conclusive answer there.
Because growing them in the field and the growing
should be
them
there,
in the lab is quite different.
No, we are doing that in the field.
Okay.
All the rhizobiome studies we are doing it. We grow the field, and we collect the root from there, and we collect the soil sample from the root tip, root on surface from the field only. No, we are not doing any rhizobiome studies in the greenhouse or lab. No.
Okay. That's it.
Thank you, sir. Thank you very much for a very informative session, for delivering a very informative and elaborate talk. And with this, we'll move on to the short lectures series.
Thank you very much. Thank you very much for that.
Thank you, sir.
Thank you, sir, for an engaging lecture. I would like to call upon Dr. Abida PS to present the memento as an expression of our appreciation to Dr. Babu Valiyodan. Ma'am, please. Thank you, sir. Thank you, ma'am. There is an announcement. The oral presentations will be held at 4:15 p.m. as parallel sessions at following venues. Theme 1, Impact on Photosynthesis, Junior Category at Botany Seminar Hall. Theme 2, Strength of Redox Systems, Senior Category at Zoology Seminar Hall. Junior category at Biotechnology Seminar Hall. Theme 3, Power of Phenomics, Junior category at 2nd MSc Classroom Botany Department. Theme 4, Water Relation and Mineral Nutrition, Senior category EMS Seminar Complex, and Junior category at Aryabhatta Seminar Hall in Science Block. The participants for oral presentation are requested to reach the Department of Botany for further instructions. Kindly report at your respective venues sharp by 4:15 p.m. Thank you.
I'm sorry. So we shall start the short lecture series. We have 7 presentations, and the first presentation is by Dr. Arun Kumar, Senior Scientist, ICR, Sugarcane Breeding Institute, Coimbatore. Please, sir.
So shall I start? Good afternoon to everyone, chairman, co-chairman, reporters, senior colleagues. Now I am going to present the short presentation on negative impact of restricted irrigation on sugarcane physiology and photosynthetic efficiency. Sugarcane is often blamed for as a water guzzlers, and water is a very important thing for crops as well as for human being also. And in this I am going to present the main importance of the sugarcane and how the physiology is translating the form of yield in the form of energy utilization. So in this map we can see that different places, almost in everywhere we can see that drought is the most prominent one in all the parts of the sugarcane growing areas. When we consider the light, out of 100% of the light incident rays, only 5% is translated in the form of carbohydrate, and only we can get at a maximum of 198 tons of dry matter. And I have calculated the most of the major important places where the sugarcane are being grown is in India. Overall we can get like 527 tons, but however we can get only maximum of 426 only as achievable yield, and under Coimbatore condition also it is less, and in other parts of the world also it is almost a similar one. And even though the radiation is common to all the crops or clones, some of the clones like they are having the erect leaves and planophiles and erectophiles, and that determines the intercepted radiation variations and which results in the yield. And this is the table showing with the various crops and the radiation use efficiency also. And the methodology what we follow is that power sensor and other how they are intercepting the solar radiation. And in this slide we can see that crop duration, incident radiation and intercepted radiation, all these things together along with the leaf angle, leaf orientation, canopy size, the form of leaf area index, that determines the crop yield. And among these things, overall the leaf extension coefficient also determines the yield. You can see that 2 different crops varieties are there, and different types of leaves also there, and these are all the variations which results in the yield variations. And this is one of the energy budget which we have made, that during the early phase of the sugarcane we can get a enormous light intensity, and only 79% maximum it is utilized. So again, the variations in the radiation use efficiency in the different crops, and in our present experiment, what we have reported in this presentation is the weather which we have got is that rainfall is almost okay, almost 690 mm rainfall received during the cropping period. The long time average is 670-74. And the solar radiation during the 24-hour cycle during the March, April, May, during the early pheno phase, we are getting a plenty of good radiation, and above 70 calories per centimeter square per day, we can get that photosynthetic rate will be less compared to the other initial phases. And also the global solar radiation during the May month is very high in the sugarcane grown at Coimbatore condition, but however during December it is very less. But during December only it is essential for the sucrose formation. And these are all the few things, and this is one of our experiment layout, how we have imposed stress with a different and irrigation layouts, and through this thing we have restricted the irrigation in some plots, and finally we found the impact on the yield and the physiology also. And this is the overall our aerial view, and these 6 plots with the different treatments are there, and with various different clones. These clones are not available in many parts of the world, and these are all we are having the exclusively with the world collections of germplasm. We have utilized this one, and can see that the border one we are having the mostly highly irrigated, and subsequently the middle center one are like stress condition. And these are all the different clones which are used. And the main observation what we found is under restricted irrigation condition, canopy temperature depression plays a major role in determining the yield. You can see that weed is also having a competition. She is having a cooler canopy, and that is that left side is a crop which is having little bit warmer than weed, and the ambient temperature and the soil. Among these things, the crop has to sustain, and we have to see that how much it is translating in the form of yield. So this is the sugarcane phenophases. 4 phenophases are there. First 2 months, and next subsequently 3 months, and again finally 4 months. You can see that solar radiation, amount of solar radiation we received, and how much it is useful for the crop, and the irrigation schedule also I have depicted in the form of graph. We can see that the brown one is the normal irrigation, and the blue one is a restricted one. Almost 50% of the irrigation is restricted, and another one is only 100% based on the pan evaporation. So based on this stress treatment, we found that under stress condition, the canopy temperature depression is having the high correlation with the biomass, whereas in control plot it is not having that much correlation. And the yield also, we got that variations in the yield, and a few of the clones are excellently performing under stress condition also, and those clones are having the better canopy temperature depression, and the energy utilization efficiency also, which is otherwise called as photosynthetic efficiency. People used to say in the photosynthetic efficiency form are different, Fe by Fm also they will tell. So this is one form that how much energy is translated. You can see that the commercial cocaines, CO 15021 and other many clones are there, they are having very good yield. And in these species clones, as a special collection, in these also this Fiji 55 are having the highest yield, or also with the energy utilization efficiency of 2%. The maximum percentage of energy utilization efficiency where we can get is maximum is 6% only, and this Fiji, which is a wild type of Erianthes type, which is having the better energy utilization efficiency. And this is the biomass variations, the form of the leaf, stem, sheath, and total biomass in the different colors I have depicted. So due to the stress, the biomass also there is a variation is there, and overall finally I can conclude that several factors are reduced. The form of number of leaves, leaf area index, everything was reduced, and among these things the canopy temperature is helpful to identify the stress tolerant clone, and among that thing the CO 100 12009 and 14002 and few other clones are having the better photosynthetic efficiency in the form of cane production. So among many parameters, the intercepted radiation was having the good correlation with the yield. So these are all the few other classical examples which we have compared in other places where they are having the higher photosynthetic efficiency, like sugarcane highest record was obtained in one place in UP, Lakhimpur Kheri, 329 tons, and the photosynthetic efficiently utilized was 65%. And these are all the maximum or highest yield based on the best management practices. So these are all our final conclusions, madam. Thank you.
James? If there are no questions, shall we proceed? Thank you, sir. Thank you so much. May I invite Dr. Lakshman RH, Principal Scientist, ICR, IAHR, Bangalore, for the presentation.
Respected Chair, Co-Chair, Rapporteur, and distinguished delegates from different institutes. And at the outset, I thank the organizers and especially Dr. Josh for inviting me to present our work here. Yeah, so yesterday Dr. Vanaja has already explained about the climate change issues, and I just want to bring in the attention of the House that under 6th assessment report we have seen that there are the agricultural ecological droughts are increasing, and in India also there are reports which we clearly show that the more dry spells are occurring, and of course the heavy precipitation events are also happening, but we are also observing the dry spells during the monsoon. So these frequencies are going to be increasing under climate change situations. So what we are going to observe is the rise in temperature, of course, because of the global warming, and associated changes like the precipitation patterns, what I told. So the water stress situations the crops are going to experience, and because of the high temperature which is prevailing under climate change conditions, we are also going to have the additional evaporative demand on the crops. So in addition to the water limiting situations, under high temperature conditions, the additional evaporative demand also is going to increase. So these stresses, both high temperature and water stress, may occur individually or in combination. So they may coincide with very critical stages of the crop growth. And of course, this has been already justified and also impressed upon that with the increase in population and the increasing demand for food, with the reduction in the available water, with the competition from domestic and other industrial water requirements, how we are going to produce more food with the limited water availability. To address these issues, the water limiting situations, the phenotyping efforts have been made across the world. And of course, I was a little worried. Since yesterday, we have been listening to all molecular studies, and Dr. Babu, he broke that chain. So he integrated both genotyping as well as phenotyping issues. So the phenotyping of the available germplasm is very important, and breeders and physiologists have been struggling to phenotype the available genotypes, especially for the desirable traits. So we have been having the approach of both destructive and non-destructive assessments. So we have been seeing since yesterday
both simple selections to most complex molecular techniques are employed to identify the desirable traits. As Dr. Babu told, that we have made considerable progress in genotyping. In terms of phenotyping, there are no commensurate work. So there is a genotype and phenotype gap. So this was very characteristically called as phenotyping bottleneck by Forbank and Tester. So these, to address the phenotyping issues, the conventional, to overcome the conventional limitations, the non-invasive imaging techniques, which include the imaging, then image analysis, and then robotics, and with high performance computing, because when the high image data is generated, so the computing is very important. Of course, these phenotyping platforms using the imaging techniques both use the greenhouse as well as field phenotyping. So the basic advantage is that they eliminate the destructive measurement of the plants, and it helps us to evaluate the same plant. So the time series measurement of the same plant is advantageous here. So this, of course, I don't want to go through because of the time limit. So ultimately, the phenotyping, it aids in the dynamic assessment of the plant growth. And there are phenotyping platforms. This is the phenotyping platform established at IHR by Lemnatech. And the advantages and disadvantages are there with both controlled condition phenomics facilities and also the field phenomics facilities. So here it is integrated with Scanalizer, then conveyor system, weighing and watering system, and then RFID enabled car system, which move in the phenomics facility so that they are imaged on a daily basis. So what we have done in phenomics facility, we have imaged the tomato crop, and this image is taken at two angles, one at 90 degree, the other one is again rotation at 90 degree, and one from the top. So using this data, the pixel of the plant is quantified, and then using this pixel, we can quantify the projected shoot area or the digital biomass. So there are different formulas which can help us in quantifying the digital biomass, and ultimately with the imaging system, we can use these pixels for quantifying the digital biomass. And we established the relationship between the digital biomass and then the plant fresh weight. So we got a very good relationship between the plant digital biomass and the plant fresh weight, and also the projected shoot area. And we used different digital biomass quantification formulas also. And what we have seen, so there are some limitations, as I told. So the imaging system, we took them at different durations, like 30 days, 45 days, 60 days. And there is very good relationship up to 60 days. Beyond 60 days, when the plant canopy grows, so there is mutual shading of the leaves. So that will not help in accessing the whole plant pixels. So that is the means the limitation. So whatever we want to phenotype in terms of the growth under different stress conditions, so we can assess them before the 60 days. That coincides with tomato, it coincides with the peak flowering stage. So we can use that stage for assessing the genotypic responses. So as I told you that beyond 60 days, there is a shading. So because of that, the whole plant canopy pixel cannot be accessed through the imaging system. And subsequently, after establishing these relationships, we evaluated the available germplasm, which were already identified in the field conditions. So we could see the differences in the percentage reduction under water stress conditions for 4 days. So there are some genotypic differences like IHR 2195 and then 2190. So they showed a lower reduction in the digital biomass, but in the recovery they had a different response. So though they are showing the less reduction in the digital biomass, but the recovery is different. So the genotypic differences are existing with respect to their response in terms of water resistance, water stress tolerance. So we could also see, Dr. Ratnakumar was asking Dr. Babu that the functionality. So we are just imaging, and then that whole image parameters, the digital parameters need to be associated with the physiological parameters. Then only we can establish the tolerance. So we could see the very good relationship between the relative water content and digital biomass. So it ultimately shows that when plant is maintaining its digital biomass under water stress condition, so it is maintaining its overall water availability in the system. And subsequently, because it is not possible for everyone to have this high throughput phenomics facility because it costed around 10 crores. So we thought of having our own indigenous system. So we developed this platform where we can place the camera on the tripod, and with the laptop we can operate it. So we use the simple digital imaging system, that is Nikon, and then this can be connected and operated through laptop. And we also use the thermal imaging system, VarioCam, for acquiring the temperature of the canopy. So this again, one of the research associate. So she has developed this software. She has developed the algorithm for assessing the images, and these images were analyzed using the MATLAB. And again, the background has to be removed for obtaining the pixels of the plants. And we used two parameters. One is the digital biomass, the other one is convex hull area. Again, the digital biomass was calculated as per the formula given. And then this convex hull area is the smallest convex set that contains all points in an image. So you can see here, this is the convex hull area which connects all the points. So when a plant is experiencing stress, so that time the leaves tend to droop. So there is reduction in the convex hull area. So that can be captured. And of course, we conducted experiments and then saw that during stress and recovery, there is a reduction in the convex hull area during stress period, and subsequently it improves. And these kind of differences, what we could see, we see some wild types. So they have different response. It's not that all the capsicum species or these solanum species or even the tomato genotypes, they respond in the same way. And we could see the differences in the temperature also. And we evaluated some 26 genotypes using this imaging technique. So along with the imaging technique, we also took the conventional parameters like the total leaf area. So we used breeding lines, then rills, then hybrids and wild type. So what we could see, but of course we need to extend this experiment and then conduct a very specific experiment. We could not see the differences among the lines, but when we are comparing the wild type with hybrids and the breeding lines, so there is a lot of difference. So we need to conduct specific experiments for those groups. So we also quantified the total dry matter accumulation and water use efficiency of these genotypes, and also the water transpired from transplanting till harvesting, that is around 70 days of growth. And we did not see, because this photosynthesis rate, what we observe is only at a particular point of time, and then that is also the unit leaf. So we did not see the differences with respect to the percent reduction in photosynthesis rate with percent reduction in biomass. See, this ultimately what we see, we wanted to see with respect to the percent reduction in digital biomass. In the rails, we could observe some lines which have got very low reduction in digital biomass, and here also in the wild species also we could observe the reduction in less reduction in digital biomass. And the canopy temperature also, these wild species, they have shown lower increase in the temperature under water stress conditions, and then some of the breeding lines also as compared to the hybrids. So based on the we want to further conduct the experiments by taking these groups separately and then see the relationship between these digital parameters and then the physiological parameters, and then work out the functionality, whether under stress condition, when the digital biomass is reduced, whether the plant is able to maintain its water potential and relative water content, like that. These are the, we are very grateful to ICR for providing the support through NICRA project, and these are the workers. Hema Malini, who is a research associate in computer science, and then Kannan and Rashmi. And thank you very much for your patient hearing.
Thank you, sir, for the nice presentation. Any questions?
Thank you, Dr. Lakshman, for your nice presentation. Actually, sir, you have the two systems. One is the sophisticated one and the one you have made. So how different is this system in their performance?
Lemnatech Phenomics, yeah, we can.
So how is it effective to have that simpler system to work with, and then can we get the solid data so that it can improve our research?
Yeah, see, the difference, major difference between the Lemnatech Phenomics facility and what the simple platform what we have developed is, the Lemnatech Phenomics facility works with the automation. The greenhouses, in the greenhouses the cars are sitting, and then the cars will be called when we give the imaging programming. So the watering and weighing also is done in the Phenomics facility. So, but here what we have done is we have used the rainout shelter, and we have used the containers, the around 25 kg containers, where we can grow the plant, and then using this platform we can take the images, one from the top and one from the side. So based on these two images also, we can quantify the digital biomass. So the advantages of field phenomics is that we are really testing the plant's ability, because it is also exposed to so many other stresses. But under controlled conditions, we are only whatever the treatment we are interested in, we'll be controlling it. And the other issue is the cost. So when we are looking into the cost, so we have to somehow compromise with the available facilities, and then see that the image analysis is done properly. So imaging and image analysis.
I wanted to particularly ask that software which you will be using for your own system, is it comparable to the Lemnatech, or you use that Lemnatech software also?
Lemnatech has its own software.
Okay,
so these algorithms were developed in the MATLAB, using MATLAB.
Okay, you have that expertise in that. Okay.
So that you can ask any computer people, they will do it.
So there is a ImageJ software system is there.
Yeah.
Can it be applied here also?
Yes, yes, that can be used for.
Okay.
no customization. See, this only requirement is some structure to hold the cameras, and then there should be some platform for keeping the laptops so that we can acquire the images of particular plant. So, but further improvement have been done. In NIASM, for example, Dr. Rane has developed. So he has integrated even the barcoder in the system. So it automatically moves, and through barcoding, the plant is known. So which plant the imaging is done. So ultimately both the data are put and they can be used for analysis.
Thank you, sir.
Sir, any trait relation here, sir? Any trait relation to yield and all?
No, that's what now we had taken 26 diverse germplasm. But in the earlier studies, what we have done, the digital biomass, when under stress condition, when it is higher, so subsequently yield also is higher. See, one particular study we have seen. So it is integration of both imaging and then the field study. So ultimately the field study has to be done. So this will give us the initial information about the germplasm.
Any more questions?
Yeah, thank you very much.
Thank you, sir. Thank you. So now next presentation is by Dr. Yogesh. May I invite Dr. Yogesh Mishra, Assistant Professor, Benares Hindu University.
The laser is there, but it won't work because of the screen. Because of the smart screen, the laser won't work. You can move it up and down.
Hello. A very good afternoon to all of you, and I'm very thankful to Professor Jose Puthur sir for giving me this opportunity to present my work, what I'm doing in my group. From very morning we are working with the yield and all these parameters. I want to bring you back to some basics that how the plant perceive the stress. Because until and unless you don't understand that how plant is going to perceive the stress, it is very hard to design the traits. So I'm trying to bring you some basics that how plants are perceiving the stress and what strategies they opt to combat with that. So here I'm going to present my work, gene encoding legume-like protein in Rhabdosis thaliana appears to be crucial in abiotic stress mitigation and related signaling pathway. So if you look at the signal transduction in Arabidopsis, around 4% of entire genome of Arabidopsis is represented by the kinases only. And the entire kinases family is divided into the different names and different class. And I am focusing on a special class of kinases that is called the receptor-like kinases, which is called as RLKs. And these RLKs is around more than 600 members are present in Rhabdosis thaliana, and they are called as receptor-like kinases. And they have a very typical structure that they have 3 domains, that is extracellular domain, transmembrane domain, and intracellular kinase domain, as you can see in the cartoon. These RLKs are further divided into the 16 families, and their names are given here. I am not going to describe each and one. even though there are a huge amount of RLKs is present in Arabidopsis, only few have been functionally characterized, and they are related to the development, innate immunity, self-incompatibility, and stress response. Then my question is that I always, people are think the negative impact of the ROS, and I always feel that there some have the positive impacts. So we looked into that, how this ROS can be important for the signal transduction. And among these RLK families, there is a class of receptor-like kinase which is called cysteine-rich repeats, which is also known as DUF26. The important part with these kinase that in their extracellular domain, they are conserved cysteine residue. And these conserved cysteine residue can be a very important domain for perception of ROS, and they are the redox. They are giving the clue that they can be the redox regulated. In addition to DUF26, there is cysteine-rich, conserved cysteine-rich extradomain is found in another class of the RLKs, that is called S domain, thaumatin and LRKs. One of my students, Subhankar, presented our recent work in this meeting that how the SD-RLKs involved into abiotic stress tolerance and signal. Here I am going to talk something different because in this RLK family there is a class of, because in any abiotic stress signaling there must be connection between cell wall and plasma membrane. So among these there is one class of protein which is called as LEC RLKs. They are kind of a cell wall and the plasma membrane linker. The important feature of this particular protein, they are having the signal peptide, L-type lectin domain, transmembrane domain, and intracellular kinase domain. And they are well known to play an important role in the plant development and especially in the biotic stress response. And they have also linked with the seed germination, lateral growth, pollen development, hormone signaling, defense against plants and in insects. In Arabidopsis genome, there is a very small class of protein which is called as legume lectin protein. There are only the 7 members present in Arabidopsis, but the important thing is that this protein do not having the transmembrane and kinase domain. They have only the ectodomain, and that is showing resemblance with the LEC RLKs. So we put the hypothesis, if the ectodomain of this legume-like protein in Arabidopsis showing similarity with the LEC RLKs, they may be an important candidate which can transfer the signal from cell wall to plasma membrane, and they should have some role in abiotic stress mitigation and tolerance. So our hypothesis was they should act as a labile sensor or carrier for the cell wall, for the signal transduction and abiotic stress mitigation. So the idea behind is that any abiotic stresses affect the cell wall rearrangement, pH and redox status of the cell and osmotic balance. Therefore, there is a huge possibility that these AT-LLP, AT I put for the Arabidopsis, must participate in abiotic stress signal transduction related pathway. In order to prove my hypothesis, we started using the in silico approach first. So we try to look at where these genes are located. So these genes are located on the 4 chromosome, 4 on the chromosome number 1, 2 genes on chromosome number 3, and 1 on chromosome number 5. And when we look at the domain analysis, there is a 7 motif were highly conserved, 8 conserved motif, and the sequence alignment of the peptide sequence of AT-LP showed that the lectin domain are subsequently conserved and signal peptide are less conserved. And after that, we looked the phylogenetic position of these AT-LLP in other 8 model plants, and we find that these proteins are highly conserved in lower non-seed plant, and they form a separate group number 1, and they are also separated in monocards and dicards. It means these proteins are very important because the non-seed plant, especially the bryophytes and all these things, were the early invaders on the land. So if some protein are highly conserved there, it shows that they should have some role in abiotic stress tolerance. So we also looked into the evolutionary divergence of entire family using Arabidopsis ATLP and looking into the available other model plants. And what we found after comparing the evolutionary divergence time that was less than 1 suggested that nature did not wanted to change the protein. It again giving a clue that this protein is highly conserved and have potential to have a role in abiotic stress mitigation and tolerance. Then we started our data again using the in silico approach. So we took the expression data, microarray data set from the bar of Arabidopsis, and we looked into the 9 abiotic stresses available. And this study shows that most of the tested ATLP expressed show the expression into high or intermediate level in both root and shoot tissue under studies tested abiotic stresses, and it again giving a clue that they should have some role in abiotic stress mitigation. After that, we did the promoter analysis and we tried to look at the CRE element present in the promoter region of this ATLP using the 500 base region in the promoter. And the important finding is that since in the promoter, the CRE represent to the oxygen responsive chloroplast development cytokinin, but for our interest is that there's a 27% of module showing that the multi-stress tolerance CRE present in the ATLP. So after that, because these CRE elements are very important with their cognate transcription factors. So we did the cognate transcription factor with the promoter region of this ATLP, and we found that how many transcription factor can bind to the CRE of this particular ATLP. An important finding is that there is a very nice correlation between ATLP and the CRE with their cognate transcription factor, supported the idea that ATLP should have role in abiotic stress tolerance. So after having the clear picture of the in silico data, we started moving toward the wet lab experiment, and for that we have chosen the 6 abiotic stresses: ozone, wounding, paraquat, heat, cold and light, and these doses were selected on the basis of available literature on the Arabidopsis, and we took a 10 days old Arabidopsis seedling exposed to these abiotic stresses and did the expression analysis after 0 hour, 2 hour, 6 hour and 12 hour. Before going to the next level, we tested the integrity of RNA both using the spectrophotometric and running the gel so that RNA isolation should be good. And after that, we did the real-time PCR analysis for all these 7 ATLLP, and this ATLLP showing very nice differential expression pattern in the all tested 6 abiotic stresses at the 0 to 12 hour. The important finding is that out of these 7 ATLP, 81G5370 and 85G03350 appear to be important key player in abiotic stress tolerance. Then the question arises, does this protein have a role really or not? For the functional characterization, we took one gene, that is 85G00350. In the literature, it is named as LLP1. So for any functional characterization, you need to have the overexpression line, mutant line, GFP line, GUS line, and all these things. So we initially developed the overexpression line for this particular. These are the steps how we have developed. This is more for the students. And after making the overexpression line, we did the PCR-based genotyping, and we prepared the overexpression line, and we finally get the homozygous line for this particular gene. So we get like 5 lines. And after that, since this protein is very important to show the localization, so we created the GFP line, and here you can see that these proteins are located in the cell wall with the confocal analysis. And after that, we procured the mutant plant from NAS, and after that, after getting this Salk line, we tested that there is the real mutant or not. Yes, they are the real mutant. And after having all these lines, we also created the complementation line, and we tested the expression of this gene of interest in these line. And here you can see that this particular gene is very stable in wild type and complementary line. and in mutant almost no negligible expression, and in the first overexpression line the overexpression was like more than 5-fold, and in the second overexpression line the expression was more than 12-fold. After having that, since our idea was that they should protect the plant from the ROS, because all the abiotic stresses indirectly or directly create the ROS enhancement into the system. So we used the ROS accumulation pattern by using the testing the accumulation of the two markers by superoxide radical and hydroperoxide using the two visualization method, like nitro blue, NBT and DAB based method. And if you look at this picture, it is clearly showing that under cold, high light, UVB and wounding, the overexpression line is showing lesser ROS as compared to the wild type. However, the mutant line is showing the more ROS accumulation, attesting the initial thing that, okay, this particular protein have a capability to offer the protection against the abiotic stresses. But we tested further to the next level, that since we got all the initial data, then we exposed these lines to the UVB. So we exposed to the 7-old seedling for the UV stress like 0.9 watt per meter square, and in the presence of PR. And as you can see, the mutant plant is more susceptible, and overexpression plants are showing tolerance towards the UVB. Similar pattern we have also observed under the cadmium stress, that is heavy metal, that here the mutant plant showing the tolerance, showing the susceptibility, however the overexpression plant showing the tolerance. And when we move to the other two abiotic stresses like salt stress, after the exposure to the 150 millimolar, I am showing the 150 millimolar data here because we exposed the seedling from 25, 50, 75. I am not showing all the data. So significant changes we have observed at 155 millimolar. So here 7 days old seedling were subjected to the salt stress. Mutant is showing more susceptibility, and after the 14 days observation, this significance enhanced. Here you can say the mutant line is more susceptible, and overexpression line showing the tolerance towards the susceptibility, again attesting the role of this ATLLP. And I am very happy that part of this study published recently in the BMC Genomics. And in nutshell, I can say that our findings supporting that these ATLLP have response against the abiotic stresses. Out of 7, 2 the ATLLP are the key candidate, and this LLP1 offer protection against UVB, cadmium and salt. But how and why? This is the next question what we are trying to answer using the different protein-protein interaction technology. And I believe in near future, I will able to answer this query. Before, for the details, I would request that please read this paper because entire story is published here. I would like to acknowledge a few people who has been supported this work. I am very happy SERB for offering me the Early Career Research Award. And I am very happy Institute of Eminence from Banaras Hindu University for giving me some fund. I am very happy to share the help of Professor Sunil Kumar Singh from MS Baroda, especially sharing the gateway technology with me. And I am happy with share the contribution of Raju Mandal. He was my former PhD student working in a Silk Institute as a scientist. And before saying thank you, I would like to say show the current activity, because I started my group at Banaras Hindu University just 7 years before, after returning from Max Planck, Germany. So I am, apart from the Arabidopsis, I am working on the Enabina especially, and I am working with the two other model system, that is Dumor Sierra hirsuta, a liverwort which is found in India, and it is very less explored plant at the molecular scale. So Subhankar is here, and he is an Arabidopsis guy. And in the Lexay part, in Enabina, we are doing something completely different, because Lexay is known to do the role in the DNA damage and repair in the case of bacteria. And for the first time, we try to look at this, does the Lexay have any role in cadmium and gamma radiation?
and we proved that the LEX-A has a role in the regulation of photosynthetic gene. Apart from that, in the case of liverworts, I believe that these tiny plants were the pioneer who really faced the nature-induced abiotic stresses. So we are looking from the last 5 years that we try to look at that how these liverworts adapt against the seasonally induced abiotic stresses. So we took Dumortira hirsuta, and because they have the 4 growing season, pre-monsoon, monsoon, post-monsoon, and fruiting time, here we applied the 2 omics approach, transcriptome and proteome, to find out the candidate gene, what are the important role in doing the seasonal changes. In parallel, we have also developed the tissue culture of this bryophyte, so that by isolating the protoplast and making the knockout, we can characterize the function of this gene. And thank you very much for your kind attention. Thank you.
Any questions? We are running very short of time. So please, if any questions are there.
Sir, please. Hello.
Hello. Dr. Yogesh, really very good presentation, very impressive. So my question is, you have identified 7 genes, right? ATLLP, right? 7. One of them you have overexpressed. Only one.
One,
So
yes.
why so? I mean, if you maybe individually if you do it, or combinationally you can do it.
Yes, you are right, sir. Actually, out of 7, the 2 candidates maintain their expression from 2 hours to 12 hours. So I use this approach. The colleague sitting next to you that who maintain the gene expression level high, that might be a good candidate. I am not nullifying that the opportunity the other gene are not knowing. Sir, with the limited resources, I started with 1, and I believe in the near future I would like to characterize the others also, sir.
No, as a young colleague, I suggest you to concentrate on the mechanisms. This will be a nature communication.
Thank you very much, sir, for your kind words and appreciation, sir.
Thank you.
Thank you.
Yes.
What was the promoter used?
A native promoter. Native promoter, sir.
Any more questions? Thank you so much.
Thank you, ma'am.
Yogesh.
Thank you.
So, yeah. Shall we continue? 3 more are there. 3 more are there. Okay. Okay. So we'll have a break for the tea. Huh? 10 minutes. Okay. Okay. So we'll have a tea break for 10 minutes. So please take exactly 10 minutes and come back. start? Yes. Yeah, may I invite Dr. Ratna Kumar Pasala, Principal Scientist, ICR, Indian Institute of Oil Science, Oilseeds Research. Please, sir.
It's not there. Yeah, very good evening. So I'm just adjusting with whatever the audience here. Thanks to the organizers for inviting me for this presentation. Myself, Dr. Ratnakumar Pasala, and here is my team, including plant biotechnologists and breeders. All together, we work on sesame crop. I'm from Indian Institute of Oilseed Research. It's located at Hyderabad. So we are working on different oilseed crops, not on the top 3, remaining 6 from sesame onwards to Niger. So these are the crops, area, production, productivity. So my topic is the phenotypic and genotypic diversity of sesame. Because I'm working for Oilseed Institute, we have to work on oil crops. So sesame is one of the important oilseed crop, which is used for conventional purpose, medicinal purpose. Most of you Keralites, you know that sesame used for medicinal purpose also. Sesame is rich, Kerala has rich wild populations of sesame. So sesame under terminal drought stress, how the phenotypic and genotypic diversity impact on sesame production, as well as what are the traits that we consider for the seed yield, and what are the lines we identified that registered and released last year. So that all the topics that I'm going to cover in my talk. Sesame is area, these are recent projections, 1.5 million hectares of area occupied by sesame. Cultivation is going on, and the production is 0.76 tons per hectare, and yield is 500 kg per hectare. Yeah, we have almost 3,000 plus germplasm is with us in sesame. In India, almost 6,500 sesame germplasms are available with NBPGR. Right now, this is a crop newly added to our institute. So we have 3,000 germplasm, including varieties, available at Indian Institute of Oilseed Research. So you can see here the variation of the cultivars. These are not cultivars, these are IC indigenous collections. So you can see the variation for leaf area and branching type. Some are the lines you can see here in red mark. There's a monoculm in the sense single stem lines with multi-capsules at each axil of the leaf. So a lot of diversity is there available in sesame. So the need of the hour is to identify the traits, how best it fit into our abiotic stress, especially for drought. And one more thing I wanted to tell you that sesame, most of the oil seeds that grow under rain-fed conditions in poor soils. So obviously they exposed to the different types of abiotic stress, especially intermittent drought or terminal drought. So we are working on the terminal drought and intermittent drought, how best we can identify the traits. The traits are contributing the yield as well as the genotypic diversity and genetic diversity of the traits, and how best we can bring into the variety. That's what we are doing there. So this is the one slide I just want to bring your attention. So these are the field screenings. We are doing part also, but most of in ICR institutes, part studies are not accepted by our competent authorities. So we do work in the field. So field conditions are different.
So we are working whatever the available collection of germplasm with us, and we are imposing the stress at different levels. Here I'm presenting terminal drought. So you can see here the second photograph, the sensors, which are real-time sensors that are monitoring the water moisture content from the soil and temperature of the soil. So that helps us, what is the soil moisture percentage at particular stage of the crop, where we can impose the stress, how best we can monitor the stress levels. So what are the desired levels of the stress we require? That all we can derive out of these sensors. These are sensors developed by IIT Mumbai. They are cost effective, like 25,000 rupees each sensor. Some of the traits that I'm working on, root traits, functional and structural root traits. In coming slides, I'm presenting that. So some of the lines, promising lines which are identified. So I just want to bring your attention here. The data which we derived here from the sensors, the top one is that holes without filling is continuously that different seasons with irrigation, and the black line below the top line. So you can see the drought, how much percentage of moisture that available in the soil. Based on that percentage of moisture, we try to derive this metric potential or bars of the soil moisture, because if you are releasing any variety or you are recommending any line, so you need to work on multiple seasons and multiple years. We are not sure that every year we expect the same drought. Until unless you have sure, so simply stopping the irrigation is not the matter. Okay, you should quantify how much stress levels you are imposing. That's what we have done using using these soil moisture sensors. These are the real-time sensors. So based on that, this is that the well water is at irrigated plants. There, you can see the soil metric potential where it is below -1, where this water stress is almost 4.5 and above. This last graph. So these are the data published in recently in Frontiers and Plant Sciences. I'm not going in detail of that. So we are worked on the structural roots. Dr. Babu made things too easy for me. So because yield is defined by water use efficiency, and harvest index. So based on that, water use and water use traits we are quantifying here. So here we have quantified the structural traits. So you can see here that irrigated and water stress variations of the traits. So these are the plants grown in polybags. Polybags is almost 1.5 meter length and 1 meter width. So those we are not restricting any root angle or root penetration. So we are extracting those roots at the maximum root potential levels, like 45, 50 days of the particular stress or the particular stage of the crop. Okay, so after that, as I shown in my earlier slide, so we have used root scanners and WindRiso software. Dr. Babu explained that. So we have quantified the different structural root rates. So you can see here, this is the root rates of 0 to 30 centimeter. So you can get a root almost 1.2 or 1.5 meter length. So we segmented those roots into 30 centimeter segments, and we scanned them, we analyzed them, and we looked into different traits like root angle, root seminal roots, and main roots content, and root length, root volume, root length density particularly, the length of the root against the proportion of the soil diameter. Okay, you can see here the variation in the irrigated and stress levels. This was published in Acta Physiologica Plantarum recently in 2021. So you can go through that paper. So, but here, with root structural traits, we could not got very good correlation to the yield with structural traits. Then we realized what other things that can be contributing the yield. So not only structural traits are contributing certain level, only 40% to 45%. What next? The traits are contributing to the yield. Then we looked into the functional traits called transpiration efficiency. Until unless you have transpiration pull, the roots cannot function. So roots, once it have pull from the leaves, then roots can absorb the more water. Though they have, in most of the legumes, they tend to produce more root length or root volume than what they require. So until unless we have the transpiration full or transpiration efficiency or water use efficiency, it won't be fulfill whole story. So we looked into that transpiration efficiency. This is a gravimetric method. So we can't weigh the plants in the field as such. So we have identified some of the lines. We brought into them based on the structural root variations, and we brought into the field, pot structure, pot culture. So there, where we restricted to apply the low density polythene beads, these white beads which are appearing in the second photo, they are low density polythene beads to restrict the evaporation from the soil. Only plant can transpire. So based on that, we have estimated the transpiration efficiency. And you can see here the relation between the transpiration efficiency and seed yield. It's a very good relation, about 6.5 under irrigated and stress conditions you can see here. And transpiration efficiency with harvest index also you can see here. So the main aim of this work is any particular variety, after reaching a threshold of drought or any stress, it drops. It drops its transpiration based on the soil water available in the particular environment. So you can see here normalized transpiration, how it fit against this transpiration of soil water. Another thing, so leaf potassium. Even yesterday somebody has talked about leaf potassium plays important role in other stresses, like Dr. In first speaker, he talked about the sodium-potassium relation under salt stress, how it works. So here we have used this trait to identify the lines which are better having more leaf potassium under stress conditions. So this also work published recently in Journal of Plant Nutrition. You can see here the seed weight and the potassium content under irrigated conditions they are correlating. Though under irrigated condition potassium is available in the leaf for stomatal, you all know that potassium plays important role in stomatal conductance and water uptake and water use. Okay, so if you look at the stress conditions where potassium is highly correlated with the seed yield. So based on that also, we have identified different germplasm which are good under drought conditions based on the leaf potassium content. So you can see the diversity of these traits on the lines, which are the traits are in the red color, and that other indigenous collections what we have selected for that is all published in Frontiers and Plant Sciences recently. About the genetic diversity, so we have used neighbor joining tree-based dendrogram to dissipating the genetic diversity among the lines which we have selected. So, and sesame is first of all is a self-pollinated crop. So we could not find much polymorphism, and we have used almost 300 SSR markers. Out of that, we got very good, very less polymorphism. You can see the diversity of the lines which we have selected for the evaluation. This is a work of past 4 years I'm presenting. It's a pool data. So you can see we have done phenotypic diversity using multivariate analysis and using SSR markers, the genetic diversity also. So you can see 4 groups of genetic diversity that we could able to see in 76 contrast lines which are based on the 4 years of our data. These are the lines which we have identified. So in one state, they used to have this sesame in different colors. For example, in Gujarat, they want black sesame, black color sesame. For Andhra, they want brown color. For Telangana, they want white color. So based on that, farmers' interest also, we have identified lines which are better under drought conditions. You can see here, these are the lines which are better. This is the GT10 And Sweta Til, they are the local and national checks. They are compared to 10 to 15% higher seed yield with good physiological traits that are contributing seed yield. So this is the one line which we got last year, CVRC, Central Variety released this one for cultivation in for summer cultivation in Telangana. This is a white seeded sesame cultivation, sesame variety recommended for summer cultivation. Sesame can grow in 3 seasons, one for Kharif, one for Rabi, and summer also, wherever the water available. So this is white color and about oil content is 49%. In sesame, almost 52% is the maximum water oil content, whatever the germplasm available in India. If you go to China, so 60% also available there. This is one more line, this is one more variety, which is released this year for under state release board of Telangana. This is again white seeded one. This is recommended for high density plant population. So this is normally farmers, they broadcast this sesame. They never go into the line to line, row to row planting. So this is recommended for that. Those farmers interested in broadcasting, maintain the high population. And this is a single stem. You can see the capsules which are bearing with seed. So at every axis, you can see 5 to 7 capsules. This is these are 2 important things from our groups recently we could achieve based on the physiology and breeding group together. These are the lines which are in pipeline. This is DT in the sense drought tolerance 112 and drought tolerance 97. And other lines which we have identified, these are, we are about to register this line with NVPGR. So my findings and way forward is, we have identified the lines which are better for farming, better suitable for summer cultivation and high plant density under limited water conditions or drought conditions. And the other one, we have identified lines based on the phenotypic character and genetic characters. And we are looking at the molecular level, how these plasma membrane incentric proteins are playing a role in water uptake, particularly under stress conditions in sesame and safflower as well. And still we have to confirm this validation of these other lines which we have identified, which are going to be registered with using high throughput technologies like phenomics and all. So we are in touch with the IRI. Hopefully we may get next year the data. And best management practices. So we are in process to develop the best management also involved in playing of variety and the field conditions. So best management practice also, our agronomists are doing to enhance the yields under rain-fed conditions. Thank you very much for your attention.
Thank you, Dr. Ratnakumar, for a very, very nice presentation. Any questions?
Dr. Ratnakumar, it was very nice. My question is how reliable the soil moisture sensor is, because when we try to use soil moisture sensor, it is not giving the real moisture status. What kind of moisture sensor?
So you have used same sensors?
I know this moisture profile meter.
Okay, then you do one thing. You do geometrical and this profile, which are, if they are almost similar, then you go with this confirmation.
Okay.
And this one is a real-time one. We have to install that at particular place for a plot 1 or like that. So based on different depths, it will give you every 1 hour data. So you can monitor through your mobile also.
It's about time domain refractometry, that kind of TDR, no, they say.
No, it's not time TDR based. Okay. It's a completely soil moisture sensors with solar power, enriched one.
Okay. Thank you.
Any more? You have used SSR markers for analysis, no?
Yes, ma'am. Yes, ma'am.
Is it related to SSR for drought-related QTLs?
We are under process, madam. We got very good large QTL for sparred chlorophyll matter, which is stay green, which is contributing stay green under drought conditions. Okay. Yet we have confirmed. way of repeating the text points.
Thank you. So we shall move on to the next presentation by Dr. Sharavanan Raju, Principal Scientist, ICAR, IAHR, Bangalore. Please, sir. Okay, okay.
Good evening, respected chair, co-chair, and rapporteur, distinguished participants of this international seminar. At the outset, I wanted to thank or grateful to the organizing committee for giving us an opportunity to present our research conducted at ICR-CTCRI on the application of biochar as an soil amendment to drought-stressed sweet potato varieties and to study their effect on growth and photosynthetic efficiency and yield, tuber yield of sweet potato. So this is my teamwork, and we conducted this experiment primarily with the 3 aims. Yeah. So actually, this actually is a mitigation, drought mitigation experiment. In that, we wanted to use our agriculture residue, which we generate when we are cultivating tuber crops, that is cassava and other tuber crops. They are mostly, the root is the economic part, and we most of the times, except cassava, other planting materials are not used as such. So only 15% of the cassava stems are used for replanting, and the rest of the material is wasted. So we wanted to make use of this residue and make it as a wealth to mitigate the most important problem of our current century, that is drought. So that we wanted to use not as an organic material as such, we wanted to make use as one of the most very promising and carbon positive material, that is biochar. So that we can eliminate the waste, and at the same time we can convert it into a useful product. And moreover, the biochar is not a single application. Only one application will do. For many years it will remain in the soil. So that is the advantage of having a biochar. So biochar has been recommended by FAO also, and many other organizations which are improving the soil health. They recommended this biochar as a very good alternative for mitigating the soil problems whenever the soil is having, poor soils are there and which are having degraded soils, which can be improved with the biochar application, and it is having advantage as it is also retaining the soil moisture and nutrients also recycled. So biochar, when we are converting the agriculture residue, many of the minerals except volatiles are removed, and the carbonic material is retained as a permanent source of carbon inside the soil, and the nutrients are also recycled. So like that we will see in that subsequent slides, how many minerals, how percentage is there in that biochar, and how that is useful to improve the soil health. Okay, so drought stress, as has been my previous presenters have given their due importance to drought stress, so I will not go into that one. It's when a prolonged precipitation deficiency is there for a particular activity or group or environmental sector. This is defined as a drought. So drought has many implications, and there are direct impacts of drought and the indirect impacts. If you see the direct impacts, these are reduce the crop yield, reduces water level, increased fire hazard, and many of these things which may happen in a region or throughout the region also, or for particular crop it can happen. So it will result in many of the social implications, economic implications that has to be mitigated through some means. So this is the global drought map. If you see across tropical regions and even in semi-tropical, subtropical and even other regions also, drought is common and it is occurring historically also. So drought is not unique, and we have to mitigate that one to improve our economy or social wealth of that country or a region. So directly into agriculture management, we can improve the soil structure or reduce or eliminate the tillage so that we can improve the infiltration of water in the soil, or we can build organic matter that can increase the water retention. The problem with increasing the soil carbon is that in a tropical soil, if you add whatever organic matter which is not retained, after a season, you have to replenish it, because due to high respiration and the microbial decay, you will never have the desired level of carbon content in the soil. To maintain that one is a very difficult task. So you have to find out an alternative so that the carbon lost every year has to be either replenished or it has to be retained. There comes the role of biochar. When biochar is applied, it can be retained in the soil for hundreds or thousands of years without degrading, or a small portion of that is lost into the soil as a leach or something due to biotransformation. Other than that, biochar can be retained in the soil for hundreds of years. So this is a very effective way of mitigating the soil, poor soils and also degraded soils, and also to retain the soil moisture and improve the soil quality. So other methods of reducing evaporation through mulch or straw, or those things are regularly you have to do it, and it is also costly. So there only biochar is an agent, which is a very important agent, useful beneficial agent to improve the soil. And this is a sustainable way of increasing soil moisture and also the soil quality. So how biochar can be made is simply any organic matter, not only crop residue, even sewage sludge can be converted into biochar by thermal decomposition, that is in the absence of or in the partial oxygen conditions. If pyrolysis conditions are maintained, biochar can be produced. So this is the biochar. In addition to that biochar production, you can have many other benefits. That when pyrolysis conditions are there, you produce syngas, that is carbon monoxide and hydrogen can be generated, that can be captured, and you can use it as an energy fuel source. And in addition to that, it can also be used as a organic matter for fuel. This biochar can also be again burned. Whenever biochar, if it is in excess produced, we can use it for industries to generate fuel. And this biochar can also be made into material which can also be applied into the soil. So how the biochar structure helps? If you see biochar, it is not that ash we burn in the presence of oxygen, full oxygen presence. When you are pyrolysis conditions, the biochar becomes the organic material becomes ash. So you won't get that benefit of biochar. But in the absence of oxygen or in the partial pressure which is very low, you retain these structures which are very important as the functionality of the biochar improves. If you see this is the ultrastructure of biochar, you retained all the cellular compartments which are retained there. In addition to that, this carbon is not the actual cellulose or hemicellulose or other materials which we found in the cell. So these are polycyclic carbonaceous materials which are very complex. It cannot be easily decomposed by microbes, and the structure will be retained for year after year in the soil. So what the advantage is that it can be a source, or it's a location where microbial growth can be there, proliferate, and the soil and root and other interactions can happen. Fungal mycelia can grow, and all these can be improve the soil quality. Microbial activity, microbial enzymes can increase, so that soil quality can improve, and that is the way the soil physical properties and soil chemical properties changes. And this is the the proximate and ultimate constituent of the biochar. And this I will not go into the detail. You can get anywhere. And this there are stages of biochar in the soil. If you apply the biochar, first 1 month, within the 1 month, it will be dissolved in the soil. It will incorporate itself into the soil particles. So it is the dissolution phase. And after that, the functional groups of the the organic material, which we say that polycyclic organic carbon compounds, they generate functional groups which are essential for the cation exchange and anion exchange capacity of the soil. And this stage will take nearly 6 months, 3 months to 6 months it will take for the functional groups to develop. And after that, aging. Aging means then again this biochar will convert itself into more functionally active form of carbon inside the soil that will retain its quality over a period of time because of the microbial growth and other biological actions of the soil. So this is the advantage of the biochar that I have already discussed. It will be a resilient material and it can generate a lot of functional properties of the soil. So this is how the picture will look like. If a biochar is there, it can bind with the clay and then it can give the clay particles more active sites, and also it can be a fertilizer because it is containing lot of nutrients, and it also increases the leaching loss of the soil because it is filling the gaps in the soil particles, and also microbial growth, and so it is slow release of nutrient fertilizer which is applied. It can act as a slow release material, and moreover it is also a carbon positive. That means that carbon is retained, so you won't get that greenhouse gas emissions which also be reduced, and volatile removal from the soil also can be reduced. So this is how we generate a low cost biochar in our institute, and this is how the waste materials which we got from the cassava stems and other things, they are used for pyrolysis, and this is the double clean, and we got this biochar for use in our experiment, and this is how the biochar yield is there in different materials, and this is the pH of the biochar. And coming to our study, we studied 3 different level of biochar. There is no biochar and 2%, 5% biochar, and irrigation level, that is control and water stress deficit. In 2 varieties of sweet potato we studied, and these are all the parameters we studied in their experiment. And this is how the plants looks like in the control and in the stress conditions, both varieties under maturity stage. And I have studied, we studied the gas exchange parameters like photosynthesis, net photosynthesis, and leaf respiratory rate. And photosynthesis and gas exchange parameters, you can see that it is affected due to water deficit stress. A drastic decrease in the net photosynthetic rate was observed, and the reduction was little bit less prominent when the biochar is applied at 2% and 5% levels. This is the respiratory rate. There also significant changes were noticed. And this is the stomatal conductance. You can see drastic reduction in the water deficit stress compared to control, and the reduction was little bit, we can say that considerable reduction, less reduction was observed when the 2% and 5% application of biochar. Same with transpiration. There was a reduction when the water deficit stress was there, and the reduction was less prominent when biochar is applied. So that means favorably affects the gas exchange parameters when biochar is there. And this is the chlorophyll fluorescence kinetics parameters, which are indicator of plant's performance, and that is the light reaction part of that. Efficiency of the plant can be monitored through this chlorophyll fluorescence. We studied the maximum quantum yield and actual quantum yield of the photosynthesis in addition to photochemical and non-photochemical processes, and this also is favorably improved when the biochar is applied. And this is the photochemical quenching and non-photochemical quenching in both varieties under no biochar, 2% biochar and 5% biochar in 2 varieties with control and water deficit stress. It improved that one in photochemical quenching also. Non-photochemical quenching increased under the water deficit stress, but there was a favorable improvement. This is the total chlorophyll content and carotenoid content. Chlorophyll content reduction was less prominent when the biochar is applied, and carotenoid content was not that significantly. This is the tuber yield of sweet potato, 2 varieties under control and water deficit stress. There was a marked difference in the improvement of tuber yield when biochar is applied. This is the first and dry weight of the tubers under water deficit stress in biochar and no biochar. So in conclusion, and moreover, this is the soil parameters. We studied the soil pH, soil organic content, and soil acid phosphatase, dehydrogenase activity, and nutrients like calcium, potassium content. Application of biochar favorably improved all these parameters of soil quality, and we found that it is very potential is there for improving this tuber yield of sweet potato. And this is the conclusion. Biochar application either at 2% or 5% soil dry weight basis improved the quality and yield of sweet potato, in addition to its plant performance and also soil quality. And this can be a very good alternative, and it can also help us to recycle the agriculture waste generated. And it has been recommended for other crops also, and many initiatives world over, biochar initiatives are there. In India, there is a certain lacuna in biochar application, but this is a very potential approach, where the degraded soils, like in our rain-fed areas and degraded soil where leaching is there, nutrients are lost. This biochar can be a boon when it is properly applied. Thank you.
Thank you, Dr. Saravanan, for a nice presentation. Any questions? What is the approximate cost of that excuse me?
Yeah.
What is that approximate cost of that pyrolysis unit?
Actually, madam, with the available resources only we managed, that is very small investment. You can make it within 10,000 rupees or so. I don't think it won't cost much. These barrels and other things that you can manage.
But lot of crop waste are burned, instead of going for biochar production. What is the reason for that in North India?
Actually, yeah, madam, that is there. In the government support, if it is there, lot of waste can be made into biochar. It is a government supported aid only initially, and a lot of industry can also depend on this biochar. Even sludges, sewage material can also be dried and it can be made into biochar. So this is a very big gap. Maybe it's ignorance, or maybe it is negligence from policymakers
or lack of awareness.
Yeah, lack of awareness, and it is a very potential. A lot of countries are helping to recycle. In Australia, US and Canada and other UK, they even the panchayat or government, city corporations and other things, they fund to generate energy as well as generate this biochar, and international agencies are working. I don't know, in India we have some isolated success stories, but not prevalent.
Thank you. So, any more? Shall we move on to the last presentation? Any more questions? Thank you. Thank you so much. So next we move on to the last presentation by Dr. Vaibhav Lohot, Senior Scientist, Indian Institute of Natural Resins and Gums. Please, sir.
Good afternoon. Actually, I said good evening. Now it's almost past 5. Respected chair, co-chair, distinguished delegates present over here, and students, I welcome you all to my presentation. Till now, everybody talked about abiotic stress, but this is an exception. I am talking about the biotic stress and its impact on the physiology of the plant. So my presentation is on dial changes in leaf starch in response to lac insect feeding on host plant pigeon pea, Cadenus caden. Actually, recently now this INRG has been renamed as National Institute of Secondary Agriculture. So we all know that insect has many faces. From agriculture point of view, they are all pest and harmful, but some are beneficial in nature also. And the best known insects are like honeybee, silkworm, and very lac insect is also there, which is there in the particular region of Jharkhand, Bihar, Madhya Pradesh, and some part of Maharashtra, Odisha also. So basically this lac insect belongs to the family Tacardidae, and it's a specialized insect, and it is a stem phloem feeder, and it produces its a secretion as a protective covering that is known as lac. It is a natural resin, and this lac insect grows on host plants only. So lac is a unique product of insect-plant interaction, and it has many implications from commercial point of view. And other than India, Bangladesh, Myanmar, Laos, Vietnam, and parts of China, they are also found lac cultivation. So this is the insect, lac insect. So first it is in the nymphal stage, then it settles on the plant, then it grows, covering it, it forms a resinous covering over its body. This is the life cycle of this lac insect. Not all the plants are host plants. Some specific plants are there which grows this lac. And one of the agriculture crop that is pigeon pea is an important host for Caryochenensis in northeast region. And our institute shows that this Caryolaca species also grows, and which increases the lac production throughout India. And it can be a good income source for the farmers. As I said, this lac insect is a phloem sap, and this phloem sap mainly consists of this carbon and nitrogen in that, and this mainly composed of sugars, free amino acids, and inorganic constituents. The sugars which are produced during the daytime, this fulfills the nutritional requirement of this lac insect. Now the question here arises, what happens when photosynthesis or sugars are not there in the during night time? So whether this lac insect feeds during night time also? So obvious the question is, we all know that starch is being degraded into the sugars, and the plants are using for all this metabolism during the night time. So we have initiated then study to measure the level of this leaf starch in lac infested pigeon pea plant, and we have at the different lac insect developmental stages, and we have compared with the control. So this is the developmental stages of this lac insect. So the starting point is in October. So first stage is pre-sexual maturity period. So it starts from October and it continues till February. Then there is a fertilization is there. Then female only survives, and it goes after fertilization period and the maturity period. So this extreme photo is small lac insects. Then they will be covered with the resin, and these are the females which are covered with resin. So we have samples we have taken from morning to evening. So 3 samples we have taken in the morning, in the afternoon, and in the evening. And with the standard methodology, we have estimated the starch. So this is in brief. So morning samples, we have plants infected with the lac insect and control in the afternoon also same. And stage 1, stage 2 and stage 3 we have developmental stages we have taken, and we have also calculated the percentage increase or decrease between the lac insect infested and against the control. So we found that this starch is decreasing with a range from 26 to 36 percent in all the developmental stages of this lac insect. And the initial development stage, there is a decrease in the starch was from 21 to 26 to 21% as compared to the post fertilization stage. And again in the maturity stage, there was very less decrease in the starch level, that is 17 to only 20%. And again, to know whether this lac insect feeding at the night, so what we have done is, we have compared the previous day evening sample with the next day morning sample. So it will give the estimation of degradation of starch or utilization of the starch by this lac insect. So this initial stage development, it says that there is an 8% more consumption of starch in the lac insect. So actually in the morning sample there is a decrease in the starch, 8% decrease in the starch we found. So we can say that 8% more has consumption, the lac insect has consumed 8% more lac insect. But when we analyze the sample of post maturity stage, when the insect has fairly developed, so we found that there is a 2.77% increase in the starch level. That means there is a, when that lac insect is feeding on the host plant, it is somehow increasing the starch content in the host plant, so that it will also feed on the host plant and the plant will also utilize that starch. So extra starch it is producing. So coming to conclusion or summary. So this lac insect developmental period, that is from after fertilization till maturity, they are the very critical stages. So initial development stage is not that critical, but once fertilization is happens, these are very important stages. And these initial developmental stages, there is some decrease in the starch we found because this actually post fertilization stage, there is a decrease in the starch it is found. So it means that there is a heavy demand by the lac insect for this starch. And again at the maturity stage, there is a less decrease in starch. That means almost this it has reached to its maturity. So demand for this food or starch, it is very less. And when we seen all the morning samples, we have seen that this starch, there is a decrease in the level of starch in the lac insect infested plants. That means the insect is consuming the starch via, means the insect is converting this starch into sugar, and that ultimately sugar is taken by the plant. So that's why decrease in the level of starch it is found. This also shows that the lac insect is also not only feeds during the daytime, but this lac insect feeds in the night time also. And there is in afternoon samples, when we have analyzed the starch, we are finding this. There is a fast decrease in the starch level. So that consumption rate of lac insect during the daytime, it is very more as compared to the morning and evening time. So this all study will help in nutritional management of the host blood. So when we are managing, when we are knowing the critical stages of the lac insect and critical period of this lac insect, then that time we can give the nutritional management to the host plant so that we can increase the lac cultivation. Thank you very much. Hope I made some explanation, because it is very different things, because insect is having, definitely they are at the biochemistry level or biochemical level, they are making the changes in the host plant. And over the past 5-6 years, I am working on this starch and sugars. So what we found that exactly the scenario for sugar, it is exactly the reverse. We are finding that sugar is increasing rather than decreasing, sugar is increasing. So somehow this lac insect, it's very voracious feeder, so it is not allowing the plants to die. So it is, somehow it is uplifting the sugar metabolism so that it will produce more sugars, so that it will sustain itself also, and it will sustain plant also. Like if some guest is coming to our home, we are making more food. It is like that only. Thank you very much.
So thank you, Dr. Vaibhav, for a very nice presentation. Any questions? So if there are no questions, we can wind up the session. I think one more session is there. Huh?
Thank you, ma'am. Now, I would like to invite Dr. A.K. Pradeep, Assistant Professor, Department of Botany, University of Calicut, to present a memento to Dr. Abida P.S., the chair of the session. Sir, please. Now, Dr. Sinisha, the session rapporteur. Ma'am. Thank you so much, sir. Thank you, ma'am.
Thus, we have come to an end to the technical sessions of the second day of the International Conference on Physiological and Molecular Mechanisms for Abiotic Stress Tolerance in Plants, jointly organized by the Indian Society for Plant Physiology South Zone and the Department of Botany, University of Calicut. Next, we have the oral presentation competitions for the theme Water Relations and Mineral Nutrition under the senior category. I invite the judges for the oral presentation competitions. Judges, please.
Hello. I would like to invite the code number OPWR02. So please come forward. So, a brief announcement for the oral presentation competitors that please keep your presentation under the time limits, that the time is for 8 minutes and the for the presentation and 2 minutes for the discussion. We will ring a bell after 7 minutes to just remind to wrap up the things and keep your mobile off. So last 2 minutes is the question answer. So please start now.
Okay. Thank you, sir. Judges of the competition, esteemed participants of this conference, welcome you all. Today I am here to present the salient findings of my work on sugarcane root feeds in response to macronutrient deficiency. Like we know that the optimum supply of nutrients has the greatest impact on sugarcane growth and yield, second only to water requirement. And the fine balance between the capacity of soil to supply nutrients and the nutrient requirement of the crop or the variety, it is very important. The physiology of that is very important to ensure fertilizer management, that is the right amount, placement and timing, and it also helps to maintain the soil fertility and environmental integrity. When we see the pattern of nutrient uptake in sugarcane, which was done over the years, we can see that potassium followed by nitrogen, calcium, magnesium, sulfur, iron, manganese, zinc and copper in that order. That is the requirement of nutrient uptake in sugarcane. The fertilizers required for sugarcane, it amounts to, not only in sugarcane for all crops, amounts to up to 50% of the operational costs in agriculture. Out of that, only 30 to 50% of the applied nitrogen is taken up and 15 to 20% of phosphorus fertilizer is taken up by the crops per se. So in Australian sugarcane farming system, up to 60% of applied nitrogen was found to be lost from the system, and potassium release rates was also not sufficient to meet the crop requirement in sugarcane. When we see the consumption of fertilizers in India, the high demand for fertilizers, we can see almost 100% increase in fertilizer consumption from 1950 up to 2015-16. So this is because of the variation in nutrient removal and uptake due to the differences in climatic condition, soil fertility and fertilizer practices, and most importantly the area of my research interest, that is nutrient use efficiency among the varieties. So with increasing environmental concerns regarding excessive fertilizer use and emphasis on sustainable crop production, improving the nutrient use efficiency of sugarcane varieties is the need of the hour. So identifying morphological, physiological and biochemical traits contributing to variation in among the sugarcane varieties to nutrient use efficiency is essential. More work has been done in case of water use efficiency and radiation use efficiency as presented by my seniors. So the physiological basis of understanding nutrient use efficiency in sugarcane we have recently initiated in the institute. So what is the relevance of root studies to enhance nutrient use efficiencies? Much work has been presented previously about the importance of roots. We know that roots are the first to experience the vagaries of soil stress, and they are the major determinants of plant growth and development. The heterogeneous availability of nutrient ions in the soil greatly affect the crop yield and quality. And sugarcane being an exhaustive crop for nutrients, there is greater dependence on fertilizers to achieve the targeted crop yields. Hence, assessing the root feeds under nutrient deficiency is essential for improving the resource use efficiency in sugarcane. So, with this objective, initially taking up nutrient use efficiency studies in the field is very much difficult. So, in the later slide, I will show about that. Initially, we started out with pot culture experiments with 5 different popular varieties of sugarcane and treatments, that is control, sufficient supply of NP and K, low nitrogen, low phosphorus and low potassium. The experiment was completely randomized with 2 factors and replicated in 3 batches. We used acid washed gravel chips and modified Hoagland solution maintained at pH 6.8 to 7. Coming to the results, like when the deficiency symptom of the nitrogen, phosphorus and potassium was visible in the plants, that is when we took the observations, that is almost in 3 to 4 month old crop. The root parameters, it was taken through with the help of the root scanner and WinRISO software. the root surface area, we could see that under the stress conditions, it was more, generally it was decreased under the stress conditions, but in some varieties, in varieties like 86032, the root surface area increased under the low nitrogen and low phosphorus, and in the varieties CO09004, the average over treatments was low, and the decrease in the root surface area was highest in case of low nitrogen, low P, and low potassium. The same trend, varietal trend, was found in case of root volume also. But the varietal variation, what we found, once again, yeah. The root volume also, we could find the same trend as in the root surface area, and on the whole, the root volume was highest under control, followed by the treatments. The treatments were all on par with each other. The average root diameter, when we see, the root diameter, it was mostly the variety CO10026, it was found to have a higher root diameter on an average among the treatments, whereas CO09004 had thinner roots, and this variety CO10026, we have found from earlier studies that it is a very efficient variety in case of water use efficiency and radiation use efficiency also. Then the root shoot ratio decreased under the nutrient stresses, and the control showed higher, but the statistical difference among the varietal variation was not significant. Then coming to the specific root length. The specific root length is the ratio of the total root length divided by the root mass, and this SRL is usually an indicator of stress. It increases under stress. Hence we hypothesize that those nutrient efficient varieties will have a lower SRL, specific root length, under stress, hence maintaining the balance between the nutrient uptake. Like we hypothesize that efficient genotypes may not necessarily have higher root length per se. They may be having other adaptations, like under the low nutrient, the expression of high affinity transporters and anatomical modifications may be present in these efficient genotypes, thereby increasing the nutrient uptake. So coming to the specific root length, we could see that in variety CO09004, the specific root length was the decrease in the specific root length was almost negligible when compared to the control. Now the root length density. Root length density, it signifies the exploratory power of the roots in a specified root volume. So that root length density variation also we could see among the varieties. And in general, the highest root length density was found in the control condition, and followed by low P, low N, and then low K. Then coming to the root system architecture, using the root phenotyping board, we were able to get the differences in the root angle, that is with respect to the average root length at the shallow, intermediary, and deep roots, and also with respect to the total number of roots present in these different root angle zones. So with this also we could see from this figure much variation among the genotypes, and we could identify like specific treatment combinations. Then coming to the root anatomical traits, similar to drought conditions wherein we had done earlier root anatomical studies, we could see that thinner root diameter with more metaxylem numbers under the low N condition, and under the low P we could see more root hairs, and this suberized exodermal areas and U-shaped thickenings in the endodermis under the stress conditions. Yeah, okay, I'll just skip this for want of time. Coming to the salient findings, like the varietal variation which may be attributed to the lesser reduction in root dry weight as observed in CO10026 at low N. And the specific traits for enhanced nutrient uptake may be appropriate root distribution pattern, longer root length and surface area, reduced root diameter, low SRL and RLD with greater allocation of photosynthetates to the roots. So with, we had done other physiological parameters also. that is some chlorophyll content and leaf area and canopy temperature depression and metabolite levels and antioxidant potential also. So this is a summary of the whole thing, like the physiological traits influencing macronutrient use efficiency in sugarcane under control condition. In sugarcane especially, as in other crops, we cannot just stop with the control condition experiments. This was just an initial, the first results of the first 2 years. Apart from this, in the field we were maintaining maize crops and sugarcane crop without any fertilizer to deplete the soil nutrients, and we propose to evaluate varieties under the field condition, identify contrasting types, and then study the physiology of it and see the mechanisms what exactly contribute to nutrient use efficiency in sugarcane. So thank you all.
So if anybody have any question, please ask from audience. No question. So one question from my side. What about the soil, what you have used, because you are using the different variety?
Yes.
So why should I trust you that all the soil properties is fixed?
Here I have not used soil, sir. I have used acid washed gravel chips and modified Hoagland solution for growing the plants.
So there is no soil in the pots?
No.
So what is inside?
Which one, sir?
No.
The media used is acid washed gravel chips, and the nutrient is supplied through the modified Hoagland solution. So that is how we can reduce and manipulate the nutrient levels, like low P, maintain the low P, low K and low N.
Okay. In your introduction slide, why
Yes.
you don't cite any literature?
Which one, sir?
In your introduction part, you have not shown any literature background about this particular studies.
Yes, sir. That is because nutrient use efficiency, the physiological evaluation for nutrient use efficiency in sugarcane is very much lesser. Very few works has been taken in case of nitrogen use efficiency in Australia by Dr. Robinson and group. And otherwise, much work has been done in case of water use efficiency and radiation use efficiency, even in our own institute. As far as the nutrient is concerned, we screen varieties based on the total nutrient uptake and not based on the physiological traits. So physiological traits contributing to nutrient efficiency, this is the first attempt, sir.
Thank you.
Micronutrient. Yeah.
Thank you.
Thank you. Thank you all.
Thank you very much. So now next one, CPWR03. OPWR, sorry.
Good evening, sir. I'm audible. Good evening to all. Good evening to Chairman sir and Co-Chairman sir and my dear students and respected senior sirs. Today I am presenting the assessment of genetic variability in finger millet germplasm varying for the drought tolerance. Myself, Pranusha, I am working as a scientist at SAR NBP, National Bureau of Plant Genetic Resources, Hyderabad. In drought is one of the most important limiting factor in finger millet production. Millets in India, major millets and minor millets are available. In major millets, sorghum and pearl millet are under major millets. Under minor millets, finger millet occupies more than 60% production in India. That is finger millet, foxtail millet, barnyard millet, proso, kodo and little millets. Among the finger millet, that is scientific name Melusine coracana, family Poaceae. It's native of the crop is Africa. More than 300 years ago, it come to introduced to India. It's commonly called as Ragi, Mandua and different areas. Area production of these finger millet crops. Major cultivating area is the Africa and Southern Asia. It is a allotetraploid plant, allotetraploid crop, that is 2N is 36. Genome size 1593 MB. In India, major occupying area is the place is the Karnataka, followed by different states, Maharashtra, Orissa, Tamil Nadu. In genetic diversity analysis, I am using the SSR markers. These are molecular markers are essential for the genetic variability existing among the germplasm. Using these morphoagronomical traits, these are the different properties of the finger millet, nutrient composition of the finger millet seed. Calcium is more in the finger millet seeds. That is 3 times more than the milk. These are the byproducts of the, that is ragi mudde, ambali, ragi dosa. Nowadays, these are more familiar to the all diabetes patient. Then, what is the main problem is the moisture stress in different stages of the crop. There is a need to improve the finger millet varieties. These are the finger millet improvement program. In pre-genomic diversity analysis, I am using these morphological studies along with SSR markers. Other markers also use this. This is finger millet improvement program. Main objectives in this study is the effect of moisture stress in different growth and stages, then understanding the physiological biochemical basis of drought tolerance in finger millet, and diversity studies, I am using these SSR markers. Total 30 finger millet genotypes used under this study. But main stress important period is panicle initiation to grain filling stage. This study under rainout shelter and kharif season. These are 30 genotypes along, means 27 genotypes plus 3 checks. These are collected from this JKVK Bangalore. This rain data and yield data, root biomass, this data collected. This is morphological characters, what are collected in this study. Physiological traits and thermotolerant traits and water use efficiency trait are different stress periods, that is 30 days after sowing, 60, 90 days after sowing. Along with some biochemical parameters during the stress period plus control. Superoxide dismutase, then leaf proline content, catalase, along with yield characters. That is mainly, that is number of productive tillers plant. These are the main important characters in this finger millet crop. Along with this quality parameter, that is protein, calcium, iron. Then coming to the genetic variability studies, isolate the isolation of DNA, then running of SES polymorphism information content, then cluster analysis also I did. The genotypic, that is genotypic variability observed among these traits. This GE1013, GE224, GE1034, GE, these accessions are maintain the good crop growth rates and photosynthetic rate. Some correlation studies under moisture stress conditions. This is leaf temperature is negatively correlated with the grain yield in finger millet crop. These studies under the moisture stress conditions, but moisture stress conditions, these accessions are showing the photosynthetic rate, SEMR, and the positive correlationship. Then again, grain yield photosynthetic rate shows the positive correlationship in the under moisture stress conditions. Yield and pad chlorophyll meter readings also showing the positive correlationship. In more than 28,000 germplasm is available in worldwide collections in various institutions, but ICR NBPs are New Delhi having 10,573 germplasms, maintaining the very good conditions at long-term storage and medium-term modules with no gaps. But ICRISAT having 5,750 accessions. What are the genetic variability availability in among the germplasms is good for the, and identification of the specific traits is useful in the hybridization programs for an improvement of new varieties. These are some of the genetic variability studies. These are the total 36 alleles are detected within the 22 SSR markers. These are the alleles range from the 1 to 7, but finally SSR primers that scoreable alleles overall is the 1.64. Then polymorphism content, that is average PIC values in present study within this 30 gene plasm only 0.07. Because of lack of time, I took only 30 genotypes. Then clusters also made with this Upagma method. Mainly cluster 1 and 3, 5 are solitary clusters. Leave it this. These are the cluster analysis. These are the markers I used in this study. These are finally conclusions. Finally, these studies genotypic variability observed in these genotypes for this physiological and molecular tolerance. These are the superior genotypes identified in this study, may be used for the donor source for the development of new varieties, finger millet development breeding programs. These are the few recommendations. These are the thank you.
Thank you very much. So any question from audience? If not, so two questions. In your regression analysis, the R value is very poor. Why? You are showing the R value like 0.6 to 0.82. So should we trust the result? Okay. You presented the SOD activity in terms of gram fresh weight. Do you think it is appropriate? No, I am not asking your lab. I am asking your view. So what could be the best way to present the SOD activity? Leave it. Tell me one big person in this millet research from India. Name of one scientist who is doing very good in this millet research from NIPGR. I can send you the link, a hint. He's very big guy and famous for this millet research. Manoj Prasad, sir. Anyway, thank you. This is OPWR 5. So please, this OPR 05.
Good evening, everyone. I am Dr. Danya. Today I would like to present my topic, morphophysiochemical and anatomic antioxidant responses of Vigna uniculata and Solanum lycopodium under waterlogging condition. Coming to my introduction, we know that water is an essential for life on earth. Without water, we can't survive. Both excess and limited water causes stress condition for plants. Nowadays, climatic changes cause variations in precipitation and flooding events. We know that flooding was mainly two categories. One is waterlogging and second one is submergence. Waterlogging means only the roots are immersed in water. But in the case of submergence, the whole plant partially or completely immersed in water. And due to heavy rainfall and poor soil drainages causes the waterlogging condition for plants. And waterlogging is studied in two phases, waterlogging and post-waterlogging phases. In post-waterlogging phases, most of the plants are susceptible to die because the reoxidation during post-waterlogging condition leads to root system damages, dehydration, drought stress to stem, and also photoinhibition of the desubmerged tissues and increased ROS production. And plants undergo physiological, anatomical, metabolical and morphological changes during waterlogging stress due to changes in internal oxygen level, carbon dioxide and reactive oxygen species. And coming to the methodology part, the collection of seeds are seeds material. The seeds of cowpea and tomato were collected from Kerala Agriculture University, Mannuthi, Thrissur. And potting mixtures are prepared by mixing soil and cow dung powder in 9:1 ratio. And this is my experimental setup. The cowpea and tomato plants are, the seeds are planted in a grow bag and growing up to 20 days. After 20 days, the plants are used for the waterlogging treatment for 7 days. The waterlogging treatment were imposed by standing grow bag in plastic vessel filled with water, and water level is maintained for 2 cm above the soil surface. Control plants were irrigated normally. After waterlogging, both treatment plant and control plant were subjected to morphological anatomical studies. And morphological studies include shoot length, root length, number of leaves and number of nodes. After 7 days, the plants were subjected to recovery for 4 days. The enzymatic and non-enzymatic antioxidants were assayed using standard protocols. And for photosynthetic pigments, the Arnold method is used, and these are the standard protocols used. And coming to my result, in waterlogged plants shows the reduction in shoot length, root length and number of leaves and number of nodes than control plant in cowpea and also in the tomato plants. In both plants, the shoot length, root length and number of leaves, number of nodes are reduced because of the waterlogging treatment. And coming to the anatomical changes, some of the aerogyma formed in waterlogged condition than the control in tomato and cowpea plants. In tomato plants also shows the aerogyma formation in waterlogged condition. And in physiologic changes, analyze the fresh weight and dry weight. The fresh weight and dry weight were decreased in waterlogged condition than control in both plant case, cowpea and tomato. And relative water content also decreased in waterlogged condition than control, and in the case of recovery, it is slight increased than waterlogged condition but decreased than control. And photosynthetic pigment, chlorophyll A, chlorophyll B, total chlorophyll and carotene were studied. In cowpea and tomato, these parameters were decreased in both waterlogged and recovery condition, but in the recovery state, the carotenoid pigments were slightly increased than waterlogged condition, but it's decreased than control. And in coming to the metabolites, the total protein content and the total carbohydrates were higher in waterlogged condition than control, but in the case of recovery, the slight decrease was observed than waterlogged condition, but it increased than control. And same to the total sugar content, the waterlogged condition shows them higher sugar content, but in recovery they slight decrease than waterlogged but higher than control. And in the case of proline, the increment of proline amino acid is higher in recovery than control, and but in the waterlogged condition slightly decreased than recovery but it is higher in control. In the case of non-enzymatic antioxidants, ascorbate, glutathione, both are same result shown. The highest value of the ascorbate or highest activity of ascorbate and glutathione was maximum in waterlogged condition than control. And same as in the recovery state, the ascorbate and glutathione was slight decreased than waterlogged but increased in control. Same result was shown in the total phenolics. The highest values observed in waterlogged condition, but in the recovery stage, the slight decreases shown in the phenolics, but higher than the control in both plants, cowpea and tomato. And coming to the enzymatic antioxidants, the SOD and catalase, the superoxide dismutase and catalase were analyzed. The activity was higher in waterlogged plants, in both plants, cowpea and tomato, and the recovery stage shows slight decrease than waterlogged but higher in control. And same as in the APX also, the waterlogged treatment shows higher APX activity then control, and then recovery. The recovery stage shows slight decreases in the APX activity with then control, waterlogged plants, but it increased in control. Coming to my conclusion, the waterlogging stress has two phases, waterlogging and post-waterlogging condition. Waterlogging stress resulted in the reduction of plant height, root length, number of leaves and number of nodes. Both fresh weight and dry weight were declined in waterlogged plant. The leaf shows yellowing and declined total chlorophyll content carotenoid. In anatomical studies, air engame formation observed. In biochemical and antioxidant activity shows higher in waterlogging condition. And in the case of tomato and cowpea, the tomato plant shows more susceptible to waterlogging than cowpea. Thank you.
Thank you very much. So, any question from audience? Okay, no question. I will ask two questions. Can you, because APX and catalase, both are H2O2 scavenging enzyme. Can you tell me the difference, how, if they are scavenging the H2O2, so why the nature have these two enzymes? What is the difference in their activity pattern?
I think it depends on the stress conditions.
No. This is definitely not related to your entire experiment.
Okay.
I am just asking, what is the difference in the activity of APX and catalase?
Same result there, because they scavenge the hydrogen peroxide and convert it into.
Second question about when you are talking about the water logging and all these things, you measured a lot of metabolites.
Okay.
So, do you think when you plan for the recovery experiments, so how you did the recovery experiments? Can you please explain it?
I am using the waterlogging condition. The grow bag is put into the water condition. Then after the 7 days, the grow bag is take out from that watering condition. Then we just placed in the bare area.
Please continue. Actually, when you present something, after introduction, you have to say what is your objective. What is your objective, and at the conclusion stage, you should say whether you have met the objective or not. I was looking for the objective slide, but it was not there. You are simply going. Okay, I just want to ask one more thing. When you say under waterlogged condition, the total weight of the plants is reduced. But but the result says that you have a lot of sugar content is increasing. So, what is the reason that under a waterlogged condition your sugar is increasing?
Because they try to tolerate that condition. So the accumulation of some sugar, some like carbohydrates, trihalose like something sugar, carbohydrates were accumulated.
You have read any literature on that aspect?
Thank you.
Thank you. Now next one is OPWR06.
A warm good evening to judges and distinguished participants. I am the last speaker of the day, I think. This is my study on characterization of eucalyptus clones for water use efficiency and associated traits. And the reason why we chosen this topic is there are two reasons. One is the eucalyptus known for water absorption. There's a lot of hue and cry for it absorbing more water. We want to address that, and there are a lot of clones which has to be screened for high water efficiency and associated traits. Another reason is that I got a student from Forest College, and that's another most important reason why I chose this topic. Just a brief introduction, not much. That is why water sensitivity, and yeah, speakers, there are many spoke about this particular trait. And this Pasteur models, that's more transpiration, more water sensitivity, and horosynthesis. Horosynthesis it's almost plateau and not possible to increase anymore. Another reason is when that's your intention to increase total dry matter productions, obviously you have to increase either transpiration or WE. Transpiration, improving transpiration is not possible much because you have a problem of water, you cannot increase the transpiration, but WE is a good option. WE, why it is not exploited in many crops is the identifying or the measuring the WEs has been a tough job. It is more gravimeter approach, especially it is more tedious. Another approach we talk about gas exchange, which is not reliable. And third approach now it is coming into carbon isotope discriminations, which is based on isotope studies. So my point is that any improvement in water density can be materialized or can be improved, and that going to impact the biomass intent to the yield. So this is my student work, and the first objective is screen the clones based on water efficiency by gravimeter approach, and this approach has been already discussed by Dr. Ratana Kumar. And the second approach is to gas agent, and third approach, if there is any physiological trait, then that can be correlated to WE as well. So this is this was carried on in Forest College, and this is about details. And we have 15 clones, and we done 8 replications, and we have a lot of clones available from across India and from abroad. And the species there are interspecies also we have received, and we put in that. And just there are morphological different, and the facility in Forest College is very pretty low in the sense we could do only the gravimeter approach, not much facility available. So with the facilities we could able to do this particular experiment. So this screenshot of what kind of the experiment, and we started this experiment like 2 months, and it went on for 70-80 days. And initially we measure initial leaf area at the start of experiment, initial biomass, and then final leaf area and final biomass. And we get a whole lot of parameters, include water efficiency, mean transpiration rate, leaf area duration, and net assimilation rate, all the parameters that we are getting. So this is all the other physiological methods that can be derived from the gravimetric approach. Here this is facilities, and the problem is how to weigh it every day. Now the water can be lost through transpiration as well as evaporation. To have control the evaporation, we have different sort of slides or something. Then we weigh every day. So we measuring the transpiration is always tedious, and we have empty pots to quantify how much water is lost through evaporation. So evaporation minus the total loss, we'll get how much of water transpired individual spots. So we had 8 replications, so still it is not enough. Almost he has to weigh every day 100 parts a day. Almost it takes 400 minutes. And in the sense, again, you have to bring back to the field capacity. How much about the water loss, again bring back to the field capacity, then added every day. So that become cumulative water transpired. So that's the thing. So this is how the experiment was done. Then we also did gas exchange parameters using PPS, and those parameters are listed here. And we use this like so here what I want to show here is the water efficiency. And surprisingly, we thought this particular eucalyptus will take more amount of water, but it is on par with the other parameters. We have done a couple of experiments in rice, which shows the 3 gram of biomass produced per liter of water transpired, and here it is 4 to 2. That's quite a bit of variability we could observe. Point number 1. Point number 2 is it is on par with the cereal crops or the pulses or the rice. So which means it is not taking more water as everybody think, and that initial conclusion. But we need to do it. The only thing that we have is this is a pot culture experiment. I understand that's a problem of pot culture experiment. Now we have started the field experiment. We are now designing for lysimeters. We have sent a proposal to the DST itself, but even then the particle experiment, but accuracy of particle experiment is very good in terms of quantifying the transpiration. So that's the thing. So we measured all the gas experiments, and that And we also calculated the water efficiency in terms of gas experiment by intercellular CO2. And there are two methods, intrinsic water efficiency using A by GS. A by GS is my conductance, assimilation by conductance. Again, instantaneous A by transpiration, assimilation by transpiration. And we correlate with the gas exchange parameter water efficiency with the gravimetry water efficiency. But unfortunately, we could not get relationship between the gas exchange water efficiency with the water efficiency from the gravimetry, which says in the single level water efficiency may not correlate with the gravimetry water efficiency. So this is a Z distribution wherein we can group the genotypes. For example, this x-axis we have a mean transpiration rate, y-axis we have a water efficiency. The first one says there are less water efficiency and more MTR. Second group is high water efficiency and low MTR. So third category is high MTR and low efficiency. Fourth category is low water efficiency, low MTR. So we have to have water efficiency also high and transpiration high if the irrigations are high. So we were able to group them into high conductancy, low conductancy. High conductance based is more transpiration types. So high capacity based on more transpiration. So you have a high WE based on high photosynthesis, not based on low transpiration. And the one genotype which we are receiving is high capacity types, which is based on more photosynthesis. So we have calculated, measured all the other parameters, and we expected these particular genotypes to be performed in all the parameters. It's not so. Some of the parameters are very good, and most of the parameters is not so. So and we also correlated with the other correlation curves using WE and mean transpiration rate and water efficiency with the net assimilation rate. More net assimilation rate, more water efficiency. So conclusion from me is that's quite a bit of genetic variability for WE, and this is an initiation that in the first time we have done this eucalyptus, and we wanted to continue in terms of in the field. And the particular clones are performing extremely well in the drought, and we have to check it in the field. And the third one is we are able to group them into conductance types and capacity types. And there is no relation between the water efficiency in the gravimetry and gas exchange. Thank you very much.
So the open for the question, please. Any question? Okay. Actually, you have done this in a pot. No? What is the age of the plant?
Age after 2 months.
2 months.
From 2 months, from another 3 months, almost 90 days.
Okay. Do you think that assessing the water use efficiency of 2 months old plants and extrapolating that to the fully grown tree is reasonable?
No, the proportion to photosynthesis transpiration has not changed with the age. In the many literature also, we have checked it. But as I said, now we are trying to do it in the field level using 6-month-old seedling to complete integration using lysing meters. So for which this lysing meter is essential, then we are, there are some problem, but the rate of photosynthesis transpiration may not change with the age. And this is just extrapolation, I understand, sir.
Because the field condition is something different from what you're giving in the pot condition. So I think probably
we have started the experiment. Yes, that's the point. We need to do it unless we do it in the field. And but this an accuracy in terms of measuring water efficiency is very good in the pot. And always they say that it's absorbed more water, and it is not so. For 4 grams per liter of water is same method of this.
What about the plant material? Are they homogeneous?
Yeah, it is completely homogeneous.
Homogeneous. So the root is homogeneous?
Yes. It's all same age, same, all the genotypes are selected with the same leaf area, with the number of leaves are same, and root types are same. No, we have taken all the care to see all that.
So which among the systems of water use efficiency you have conducted here, which one do you think is more appropriate or accurate?
Gravimetry, sir. Gas exchange, for example, we are done for rice, and there is a correlation between gravimetry WE and gas exchange WE in case of rice. But in this case, there's no correlation, and again we need to check that. And there's one more approach called carbon isotope discriminations, and we are sending the sample for isotope studies. We need to check with that whether this correlate with the gravimetry WE or gas exchange WE, something like that.
So, as a continuation of this program, how do you plan your bigotry study? What age?
No, we are from the 6 month old to complete. This at least 3 to 4 years we need to study this. Unless then we don't get the results. Field, we have a lysimeter. A lysimeter like we are designing, from that how much water lost, and that's how we study. Another approach is like moisture sensor. We are using moisture sensor because exact quantification or transpiration is always tough in the field, because a lot of evaporation and a lot of the soil material is not homogeneous. And we need to have a control conditions, and that seems tough.
So
for us.
what is the maximum capacity of tree size possible in the lysimeter with regard to eucalyptus?
No, what we are planning is 2 meter by 2 meter, but again we can't do up to.
No, the age of the tree. What is the size of the?
No, that will be very huge.
How long it can be in eucalyptus?
It is 15 years. 15 years. Yeah, 15 years. 5 years. Normally they go for cutting by 15 years.
Do you have any such idea to prolong this?
No.
That only a short-term study.
That will be yes.
Okay. All the best.
Okay.
Sir, I have a query.
Please.
Bhuminathan, you said lysimeters. What is the size of lysimeters? How many plants you can accommodate in that?
No, one to two in individual lysimeter. Now we are designing with the company people, and they are saying it will be very expensive. Now we are, that it is good for the annual crops, but for Yeah. Yes. Isotopes, that will be better. But whether isotope studies not correlate with the many crops, again we need to check for eucalyptus.
We checked with Dr. Shesha.
I spoke to him. We are in discussion. Yes, but there's no study for eucalyptus. So we have sent the sample and we got to check correlate with the.
What the roots that you have you any done on root studies?
No. This is just we measured root biomass and correlate with the WE, but that's no correlation.
No, especially in eucalyptus, that root is double than the shoot. There are some reports like.
How root and shoot?
Root shoot ratio. Root ratio is double than the shoot ratio, like that.
No, here it is 1.5, not double, 1 to 1.5 ratio. Yeah, but there's a more root in case of eucalyptus.
If it is a long-term experiment, can you accommodate them?
It will be tough. Yes. Thank you, sir. Thank you so much.
Thank you, participants, and thank you, judges. We have come to an end of the second day of the International Conference on Physiological and Molecular Mechanisms for Abiotic Stress Tolerance in Plants, jointly organized by the Indian Society for Plant Physiology, South Zone, and the Department of Botany, University of Calicut. I thank all the delegates and participants for their active participation. The University Park is open for the participants. We encourage the delegates and participants to enjoy your evening at the park. The dinner will be arranged in the Calicut University Park. All are requested to reach the Calicut University Park by 8 p.m. for dinner. Thank you.